Test microfono radio.
Sì, 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 sì. Va bene, non importa, tanto solo... Ok.
Complimenti, ho visto molte persone, bell'evento.
Abbiamo fatto il nostro meglio. Devo dire, se non c'era Claudia e Antonella, tutto questo non era possibile, professore, sicuramente.
Abbiamo fortunatamente dei gruppi pieni, tra l'altro pieni di giovani che ci stanno sostenendo. Welcome everybody. Thank you to be here to join this meeting from different countries, and we are really happy of this big interest that you show. Here we have our rector, Nathan Levialdi-Giron, and we are really happy and proud to have him to open and to give greetings to let's start this conference at the best.
Thank you very much. Dear guests and colleagues, good morning. With great pleasure, I welcome all of you at Tor Vergata University. Let me thank the organizing committee chaired by Professor Massimo Bottini for inviting me to open this important international scientific event that arrived at its sixth edition will honor us with the presence of authoritative researchers and biologist experts from all over the world. The wide and interesting program aims to bring together young and senior experts in the field of extracellular vesicles and stimulate discussion on the state of the art and the challenges in the field. On the first day, you will face the very last discoveries on the research field of extracellular vesicles, which has gained an increasingly central role in medicine and biology over time for the identification of new early biomarkers of disease, such as, for example, tumors, infections, and other complex pathologies, and the development of innovative personalized therapies. I am glad to see that young researchers, our rising stars, will have the opportunity to present their research and that the best work presented will be awarded, giving them the right recognition and stimulation to continue researching. Another compelling aspect of this event is the School of Extracellular Vesicles and Nanoparticles. On the last 2 years of the Congress, participants will take part in theoretical and practical lectures with the Laboratory of Experimental Medicine of our university. This experience will offer them the opportunity to apply their knowledge, deepen advanced technical skills, and directly compare with the latest research methodologies in experimental medicine. For sure, the field of extracellular vesicles is very complex, and there is still a lot to discover and study, but I'm confident that through significant events like this conference, enriched by the valuable contribution of nationally and internationally renowned speakers, our university has successfully fulfilled its mission to foster the exchange of ideas and knowledge, as well as to disseminate groundbreaking scientific advancements. So I now leave the floor to my colleagues and all the distinguished guests who will speak today, and I wish you all a very fruitful day to all of you. Thank you very much.
Thank you so much. I don't know if Professor Bottini wants to tell something, and I know that the Professor Giron has to go.
Grazie, professore. Grazie, professore.
Okay. Even if we couldn't finalize the registration, maybe we can start for the morning section, and then during the coffee break, who didn't take the things needs and just to not to go
there is no
because
yes.
we are a little bit late.
Little bit late.
So this is good for you.
Now, unfortunately, we have to start today with the sad news that yesterday one of our colleagues in our department passed away. She was extremely young, and therefore, Valentina didn't make it yesterday. She was recruited like less than 1 year ago in our department as assistant professor. So I would like to have 1 minute of silence for her, because that news was just heartbreaking for us. So please, 1 minute of silence. So silence. Okay. Thank you very much. Okay, so we can start, the event today. First of all, I would like to thank all of you, for being here, for having accepted our invitation to be here and to talk. I would like to give a very quick overview of the conference. We will start with a keynote speaker that is from the Bernamese Professor Milan. Then we will have two sessions. One session, the first one will be about media vesicle, the second one about matrix vesicle. Then we'll keep the conference, we'll keep going with the conference, will be the presentation of the sponsor. The sponsor, will have 1 hour to present the instrumentation, and during the buffet lunch will be the poster exhibition. Now, we will start again after lunch with a new keynote speaker, Professor Radeglieri, is the vice president of Evita, will give a keynote talk, and then we will have a fourth very short session about media vesicles, and then finally, the rising stars. So the young investigators, young students will present their research. We're very, very happy. And then we will be the awards. So the commission will we evaluate the posters and the talk, and we will give awards. Now, we would also like to thank the societies, the International Society for Extracellular Vesicle that sponsored and our event. They endorsed us. The Italian Society for Extracellular Vesicle, and today we have the vice president with us, and the Journal of Extracellular Biology. The book of abstract will be published on JX Bio. And finally, last not least, a special thanks to the students from a lab, from Claudia's lab, the students from the medical engineering that help us, and the other, that were very important, for the success of this event. So thank you very much for being here, and now I'll let Claudia to introduce the first speaker.
So it's a pleasure to me to introduce the keynote speaker, that is Professor Joseph Louis Milian. And he received his PhD in medical biochemistry from University of Umeå in Sweden, and then he performed his postdoctoral period in Copenhagen, and then in Center Bernard Prytz at the Medical Discovery Institute. In 1986, he's appointed in the faculty, and from 2008, he has appointed in Stanford as a Stanford investigator at the Sanford Children's Health Research Center in Human Genetics Program. His research focused on the mechanism and initiation of the skeletal or dental mineralization and the ectopic calcification of soft tissue using the power of mouse genetics to understand the pathophysiology of rare genetic disease. In extracellular vesicle is focused on matrix vesicle involving initiation of mineralization in cartilage, bone, and other vertebrate tissue. And this is a pleasure to have you here to
Thank you very much for the kind introduction.
Thank
And thank you for the invitation to be here with you. I'll try to make it short, just do an overview of what has been of interest of mine for many, many decades, which is the mechanisms of initiation of skeletal mineralization. The system we have chosen to work with are the hypertrophic chondrocytes of the growth plate. These are cells that essentially bleb this type of extracellular vesicles called matrix vesicles, and that is where calcium and phosphate come together to initiate calcification. And these vesicles were co-discovered by Clark Anderson in the US and Hermano Bonucci here in Rome at La Sapienza in the early 60s. And these are images from their work. You see that they are essentially entrapped by a double layer, membrane layer. And this is where calcium and phosphate come together for the initial seeds of mineral, of hydroxyapatite deposition. Now here I also introduce the presence of pyrophosphate, inorganic pyrophosphate, two molecules of phosphate linked together by an oxygen group. This is a very potent inhibitor of initiation of mineralization. And these are images from Massimo Bottini's work, actually with us in La Jolla, showing the crystals beginning to grow within the matrix vesicles by atomic force microscopy. Then these crystals grow beyond the confines of the membranes, puncture through the membranes, destroying the matrix vesicles, and then propagates the mineral onto the collagenous matrix. Throughout this entire process, pyrophosphate is there to control the rate of growth of the crystals and to slow down this process that needs to be done at a precise rate of growth. We spend, this summary slide represents about 25 years of work, dissecting one by one the enzymes and the transporters that are involved in this mechanism. So the initial crystal within the matrix vesicles are formed by two mechanisms. One mechanism generates phosphates within the matrix vesicles, and this is by the action of SMPD3 that takes sphingomyelin to form phosphocholine. Phosphocholine is acted upon by phospho-1 to create phosphate within the matrix vesicles. That is one mechanism to generate phosphate within the matrix vesicles to initiate this seed crystal formation. Another mechanism is to take phosphate from the outside of the matrix vesicles via phosphate transporters, and this phosphate is generated perivesicularly by the action of alkaline phosphatase, tissue non-specific alkaline phosphatase, an enzyme that I work with for about 50 years now. The main substrate for this is ATP, perivesicularly, but also pyrophosphate. And a crucial function of this enzyme is to destroy pyrophosphate, to hydrolyze it, so this essentially keeping the concentration of pyrophosphate at a low level to allow mineralization in the matrix of the bone to calcify. Outside in the extracellular matrix, you also have the presence of osteopontin, another very potent inhibitor of mineralization, just like pyrophosphate. And osteopontin exists in two forms, phosphorylated and dephosphorylated. Phosphorylated is an active inhibitor of mineralization. Dephosphorylated does not work as an inhibitor of mineralization. So alkaline phosphatase also acts on phosphoosteopontin. So alkaline phosphatase regulates the phosphate to pyrophosphate ratio as well as the osteopontin to phosphoosteopontin ratio. Very important function in the extracellular matrix. Okay? And these are the images that shows initiation, progression, and propagation onto the extracellular matrix. That's a summary of about 25 years of work in that particular schematic. Here I show you images generated by Clark Anderson, who again co-discovered matrix vesicles, showing you what happens when you lack alkaline phosphatase. You have initiation of mineralization. Here you have mineral within the matrix vesicles, but no propagation. This is what a normal matrix would look like. All of this black material here is hydroxyapatite deposited onto the collagenous fibrils. Here you see lack of propagation. However, initiation is there because you have the phospho-1 mechanism generating phosphate inside. When you knock out phosphor-1, you still have initiation in the vesicles because now you have the phosphate generated outside the vesicles coming in through phosphate transporters. So both of these vesicles still initiate, but they do not propagate well. That's an example of how these matrix vesicles function in bone mineralization. This is probably the best experiments I've ever done in my life, and that is a double mutant where you lack alkaline phosphates and phosphor-1 together, and you have zero calcification. The skeletal enlage is there, the cartilage is there, it just does not calcify. And this is very important for what I will finish up my talk with, because if you knock out these two enzymes, you prevent calcification. And this could be useful to prevent ectopic calcification in a variety of conditions. And I'll come back to this. Together with Pietro Giancaglini, sitting here in the audience, we have tried to also develop a biomimetic system, trying to put one enzyme at a time into proteoliposome system to see whether we can reproduce in vitro in synthetic nanoparticles the function of all of these enzymes to be able to use them for nanomedicine applications. And this has been quite crucial work to help us confirm what we see in vivo in our animal models. So working with in vitro, the proteoliposome systems that he's a specialist on with the animal models has been really very informative for us. There are things we still do not understand about these matrix vesicles. First of all is how are they generated? We will hear from Dobrava Napirala that phosphate is involved in the biogenesis of matrix vesicles. We know that phosphor-1 is involved. This is also worked by Massimo Bottini here. When we knock out phosphor-1, the vesicles that are produced are less in number. They are smaller. Here you see the diameter and the volume is smaller, and they do not have mineral inside. So phosphor-1 has a role in the biosynthesis and the production of matrix vesicles, but probably also SMPD3. Cassie Shanahan in the UK has shown that inhibiting SMPD3 also affects matrix vesicle biogenesis. So all of these things are probably engaged in the biogenesis of matrix vesicles. We still do not understand very much about how these matrix vesicles attach to the collagenous matrix and propagate the mineral that was initiated inside the vesicles. How is that mineral transferred onto the collagenous matrix and propagated? But some inklings are coming now. This is a large collaborative study together with colleagues from Lyon, René Boucher, Saida Mebarek, who is here with us, Massimo Bottini, Pietro Giancaglini, Ana Paula Ramos. The idea here has been, could these vesicles be cloaked by a functional protein corona that could affect the ability of these vesicles to attach to the collagenous matrix? And indeed, we now have a paper essentially impressed in JBMR+, showing that this is the case. These are matrix vesicles, native matrix vesicles attaching to a collagenous matrix, and in the presence of ATP, they mineralize the matrix. Here are vesicles shedded that we have essentially denuded them of all this cloaking protein corona, and they do not attach to the collagenous matrix, and they do not mineralize the matrix. And when we add those protein corona proteins back to these matrix vesicles, they regain the ability to bind to collagen, and they regain the ability to mineralize the matrix. Something that is very interesting to me is the fact that alkaline phosphatase activity on the surface of these vesicles is affected by the presence of the protein corona. These are native matrix vesicles. When they are shaved, denuded of the protein corona, activity goes up. In my view, it means that we're unmasking active sites, exposing more enzyme to the environment. And when we add the protein corona back to the vesicles, we reconstitute partially the activity. What this indicates to me is that we're not adding the proteins in the right order, something that perhaps in nature happens as these vesicles are being bled from the cell. They are beginning to be exposed to a certain type of protein corona, and they need to be added in a certain order for these vesicles to function physiologically. Here we are messing up with the right orderly addition of these proteins, but more to look into. But this is just the first paper in this direction, which I think is very, very exciting. Now, what I've told you so far is that the concentration of inorganic pyrophosphate is crucial to maintain physiological skeletal dental mineralization. And the enzyme that is crucial for maintaining that concentration is alkaline phosphatase, tissue non-specific alkaline phosphatase, and pyrophosphate has been called, appropriately so, the body's water softener. Essentially, we are inundated with pyrophosphate in our body so that we do not calcify throughout our body. Pyrophosphate is what prevents our soft tissues from calcifying. Now, when you have too much pyrophosphate, then you are suppressing physiological skeletal mineralization. When you have too little pyrophosphate, now you have ectopic, inappropriate calcification of our soft tissues. So crucially important to maintain that concentration controlled. And I will show you now two examples of these diseases. Hypophosphatasia, which is a lack of alkaline phosphate that leads to accumulation of pyrophosphate, and how we have dealt with that issue. And then I'll show you two examples of inappropriate soft tissue calcification in the form of vascular calcification and also age-related macular degeneration in the eye. So hypophosphatasia, lack of alkaline phosphatase, leads to accumulation of pyrophosphate, accumulation of phosphosteopontin, and this is what these children look like when they are born. They essentially die within 6 months of age with very poorly mineralized skeleton. How can we prevent this from happening? How can we target the missing alkaline phosphate to the sites where it is needed in the extracellular matrix to essentially cleave pyrophosphate and allow calcification to proceed? Well, the trick came working with a company in Canada, Innovia Pharmaceutical, that had a little trick up their sleeves. Adding a polyaspartic acid motif would confer mineral binding properties to the enzyme. So this was a recombinant enzyme. There was an FC region of immunoglobulin to make purification easy, but this was really the business part of the molecule. Polyaspartic acid motif, with injecting the enzyme, it would go to where mineral is, cleave pyrophosphate, allow calcification to proceed. And this is a beautiful example here, looking at teeth enamel. When you look at wild-type sections, you see the alkaline phosphatase activities at the cellular layer. Here is the amyloblast, positive for alkaline phosphatase, and here you have odontoblast, positive for alkaline phosphatase. When you knock out this gene in mice, it's totally blank. There is no cellular activity of alkaline phosphatase, but when we inject this mineral-targeted form of the enzyme, it goes to the enamel, to the mineral layer. It is active in the extracellular mineral layer, not at the cellular level. So we're not correcting the defect in the matrix vesicles and in the cells. We're simply putting the enzyme where the mineral phase is to downregulate pyrophosphate concentrations, okay? Beautiful example of how this enzyme works. And what happens is exactly what I said. This goes to the extracellular matrix. It essentially reduces the pyrophosphate to phosphate ratio, reduces the phosphosteopontin to osteopontin ratio, allowing proper mineralization to ensue. Well, this therapy was approved in 2015. It is essentially saving children's lives. This is what the hands of this particular 1-month-old baby look like before treatment. After 24 weeks of treatment, you see the incredible improvement in the calcification of the skeleton and teeth. So this therapy is saving lives. However, there are problems. These kids need to inject themselves daily for life. And sometimes they develop this injection site problems, and sometimes discontinue treatment, which is not really good for their skeleton. Together with Professor Shimada and Miyake, we've been working for years trying to see whether we could use AAV vectors to deliver this mineral-targeted form of the enzyme. And indeed, in mice, a single injection to deliver this enzyme fixes the problem for life. For humans, it may be more than so. It may be once a year, maybe once every 5 years, but there is an opportunity for AAV8-mediated therapy to at least resolve that problem that these children have, which are the daily injections. We just presented this work at ASBMR in Toronto a few months ago, where we have done all the dosing studies, both in models of early onset hypophosphatasia, late onset hypophosphatasia, and it works beautifully. Now, this is ready for prime time. A company needs to pick this up and take it to the clinic. But our work, I think, preclinical work, I think, is done for this approach. Now, we developed another approach together with the support of another company, and that is what would happen if we inhibit ENPP1, the enzyme that produces pyrophosphate at the cellular level and at the matrix vesicle level. And what would happen there is that we're essentially affecting the ratio by inhibiting this enzyme, which is crucial for the synthesis of pyrophosphate. And we presented that work at the SBMR, and it shows promising results. So again, one example of how we can affect an activity that is proper at the matrix vesicle level. And now, just to finish up the other part of the story, which is there are two types of vascular calcification, intimal and medial. Intimal is atherosclerosis, medial calcification, end-stage renal disease, diabetes, obesity, aging, and then some rare diseases. Generalized arterial calcification of infancy, pseudoxanthoma elasticum, arterial calcification due to a deficiency in 5' nucleotidase, CD73, which is also called calcification of joints and arteries. These are all very rare diseases, but very interesting because these are experiments of nature that allows you to probe a mechanism physiologically. And for a pharmaceutical company to go into a new treatment, it's always easier to go with a rare disease and then apply off-label a treatment to a common disease and to do clinical trials with a common disease. So rare diseases are very valuable also to develop novel treatments. We know that upregulation of, all of these diseases, what they have in common is upregulation of alkaline phosphatase ectopically at sites of calcification. And we tested how significant that would be with two different mouse models. We overexpressed alkaline phosphatase in the medial layer of the arteries with the Taglin-Cree mouse, and in the intimal layer of the arteries with the Tie2 Cree promoter, and the consequences are dramatic. Simply overexpressing alkaline phosphatase in the medial layer, the aorta solidly calcifies, completely calcifies. Cardiac hypertrophy, short life, increased blood pressure. Essentially, this is a severe model of generalized arterial calcification of infancy, and the mice die prematurely with severe calcification of the vasculature. The same happens if we overexpress the enzyme in the intimal layer of the arteries. You have here the heart arteries calcified as well. So we know that it's very severe consequences of having alkaline phosphatase overexpressed in the vasculature. So this indicates to us that alkaline phosphatase is not only a protein biologic to fix the problem in hypophosphatasia, but also a therapeutic target to try to inhibit this to prevent vascular calcification in all of these rare and common diseases that I mentioned to you. So together with scientists at our institute, we essentially did a HIT screening for inhibitors and modified by medicinal chemistry. We went from a HIT to a pharmacological inhibitor, SBI-425, a very potent pharmacological inhibitor, and we showed that indeed we can prevent calcification, we can reduce the calcium load in these mice, we can reduce the heart size without affecting physiological, skeletal, and dental mineralization, which is crucial. This enzyme is crucial for those physiological needed events. We should not touch the enzyme in the skeleton and teeth. We should touch it only in the vasculature when it is inappropriately overexpressed, and it does so. We licensed this compound to a company who chose to work with a rare disease to try to bring it to the clinic. And the rare disease they chose was pseudoxanthoma elasticum. This disease is caused by deficiency in ABCC6, which is an ATP transporter, the substrate that is then going to be used by ENPP1 to produce pyrophosphate. And what happens in this disease is that you have pseudoxanthomas, meaning deposition of hydroxyapatite in the skin. These are not lipidic depositions like in xanthoma, but pseudoxanthoma is hydroxyapatite in the skin. You have eye issues, you have eye calcification, this peau d'orange, this kind of problems, and also vascular calcification. So we licensed the compound in 2015 to Daiichi Sankyo. They insisted on spending 2 years repeating everything that I had done in our preclinical models, which was good for them and good for me. Everything cleared out. And then they initiated phase 1 clinical trials for tolerability 2017 to 2019. Then COVID hit. There was a big stop in all these clinical trials. Phase 2 were now completed as of 2023, and now we're engaging phase 3 patients for phase 3 clinical trials. So probably, if this inhibitor goes further, it will be used to treat pseudoxanthoma elasticum, these rare diseases, but of course the hope is that they will be used for more common indications. And while the company was doing the clinical trials, we continue to do preclinical work in our laboratory. One of the diseases that are, of course, of interest is chronic kidney disease. Which is end-stage renal disease. And with Takashi Tani in Japan, we have shown that indeed it can suppress inappropriate calcification of the medial layers of the vasculature in chronic kidney disease. We've also worked with David Mang in Lyon to see whether this inhibitor could be useful for the treatment of atherosclerosis, and indeed it does a beautiful job in preventing microcalcification, which is one of the bad events in atherosclerosis. Large calcifications may be protective for plaque rupture, but microcalcification are really a bad indication of atherosclerosis. So these are the potential large indications where such an inhibitor could be used. And just to finish up, more recently, we started collaborating with Francesca Marazzi on age-related macular degeneration. Now, there are no good models of age-related macular degeneration. Instead, we're using surrogate models. And the surrogate models are the ones I already mentioned to you. ENPP1 deficiency, which causes generalized arterial calcification of infancy, ABCC6 deficiency, which causes pseudoxanthoma elasticum. So we're interested in looking at the degeneration of drusen. Is this mineral in the eye which eventually leads to blindness? Is this caused by matrix vesicles forming inappropriately in retinal pigment epithelial cells or in the choroid vasculature in the eye? These are the mechanisms that we need to explore. We've shown in the past that we could develop also inhibitors of phospho-1 to break down or to impede the formation of phosphate within the matrix vesicles, as well as interfere with matrix vesicle biogenesis, and that prevented in vitro calcification of cells. If we combine the phospho-1 inhibitor with a TNAP inhibitor that we developed, we now have essentially an increased effect. And what I think is happening here is a double whammy. You're essentially inhibiting phospho-1, potentially inhibiting generation of phosphate inside the vesicles, potentially inhibiting the formation, the biogenesis of matrix vesicles, and then by inhibiting alkaline phosphatase, you're doing all of these that I already mentioned to you. You're essentially increasing the pyrophosphate to phosphate ratio, you're increasing the phospho-osteopontin to osteopontin ratio, suppressing propagation. So with this dual phosphatase strategy, which could be a phospho-1 TNAP strategy, could be an SMPD3 inhibitor with TNAP strategy, you could suppress biogenesis of matrix vesicles and propagation of mineral onto the extracellular matrix. And with that, I'm going to leave you. I tried to make it as quickly as possible to catch up with time. Happy to answer any questions for you. Thank you very much.
Thank you. Thank you. Really amazing presentation. There are some questions from the audience.
Yes, please.
Thank you for your presentation. Very impressive.
Thank you.
So really impressive. I have a question.
Yes.
Because if I understand this well, this matrix vesicle have an intrinsic enzymatic activity.
Correct.
Right?
Correct.
Thank you. And
It's working.
the phosphocoline is a substrate.
Yes.
So you think if you modulate the phospholipids or the lipid composition of these matrix vesicles, you can also affect these enzymatic activity?
Yes.
Because you use also artificial liposomes, so maybe this could be in the simple way analyzed or checked.
Right, right. In fact, we have here the expert on how to modulate phospholipids in nanoparticles, like Pietro Ciancaglini here. modulating the composition of lipids in vivo, that really more depends on diet than anything else. I don't know that we could affect artificially matrix vesicle, because that depends on what we ingest, how the membrane behaves, and that changes based on your diet. So I don't know that we can artificially modulate that, but certainly in vitro that happens. And Pietro has been playing with phospholipid compositions looking at this issue. But I don't know, I mean, we could essentially target SMPD3, the enzyme that physiologically acts on these lipids. But whether we can modulate the lipids medicinally, I don't know that is possible. Honestly, that's beyond my expertise. Yeah, please.
Okay, just a curiosity, because it was an exciting idea that protein corona is not a chaotic addition of molecules, but a precise addition of what is needed. Who is orchestrating the addition?
Good question, and I don't have the answer. Presumably, since the matrix vesicles are blebbing out of the cell, depending on what ECM they see, what extracellular matrix they see, the protein corona is bound to be different. And this is what I'm interested in. Is the protein corona different, potentially it is, in chondrocytes versus osteoblasts versus vascular smooth muscle cells? So as soon as these vesicles are blebbing out, they are sensing their environment, and some protein corona may be adhering in some orderly fashion. So we still don't understand very much biogenesis. We still don't understand the position of the corona. Lots of things to look into, but I'm excited about the possibility that the different protein corona may be modulating the activity of these enzymes so crucial to control mineralization in the different organs: vascular smooth muscle cells, intimal cells, osteoblasts, osteocytes. Lots of things to look into. I think it's exciting.
Yes, I believe that this biology of the extracellular vesicle is very complex, and so we have a lot of pieces to be had, information to be had, and we hope that today we will heard a lot also from different disease contexts, so and more than characterization of the extracellular vesicles. I believe that we are starting the better way.
Okay, thank you.
So thank you so much.
Thank you. You're welcome.
Thank you very much. So we can start with the. Thank you very much, Professor Milan. Thanks. And by the way, so there will be the keynote speaker in the afternoon, the vice president of EVITA, that will tell us something more about the protein corona, and there will be an event in February. I think that she will tell us something more about this event in February that she's organizing. So let's move to the session 1, media vesicle. It is my great pleasure to introduce Professor Dini, my dear friend, Professor Dini, that is a full professor of cell biology and nanobiotechnology at Sapienza University. She works on nanobiotechnology spanning from synthesis to characterization and nanotoxicity, and currently she's exploring the role of extracellular vesicles in differentiation, cancer, and metabolic disorders by using such techniques as electron microscopy, tomography, optical imaging, correlative microscopy, histochemistry, and molecular biology. Today she will tell us something about the macrophage-derived extracellular vesicle, altered polarization of recipient macrophages, and skeletal muscle homeostasis. No, this is not.
No.
This is not your
It's
time?
always the same mistake. Doesn't matter.
I'm so sorry,
We
Lucian.
can just move to the presentation. So the title you can.
They didn't change it.
Presentazioni.
Sorry, extracellular vesicle mediate glioblastoma.
Doesn't matter.
Luciana,
I will introduce myself, and this is the
introduction.
reason why we love so much.
The introduction was okay.
The introduction was perfect because you read what I wrote.
Always lovely.
No, it's okay. So this is the interest, the actual interest, trying to understand the role of extracellular vesicle in the brain tumor that is glioblastoma. First of all, I have to thank Professor Bottini and Matteucci to be here. And this is a sort of mission impossible to speak after such a beautiful presentation that we listen just now. Okay, so start with the few words regarding glioblastoma multiforme. That is a still incurable tumor with only 15 months as average of survival due to a lot of typical characteristics of the cancer cells, mainly that they are heterogeneous, very resistant to all the different types of therapies. So actually the therapy include surgery, chemotherapy, and perhaps radiotherapy all together to help patients. What we already know is that there is a peculiar microenvironment of glioblastoma tumor in which there are a lot of different types of cells. The different types of cancer cells, but also macrophages in different activation, one of M2 that can promote or reduce the growth of cancer cells. And again, all the astrocyte, oligodendrocyte, microglia, together with cells that are coming from the blood and the new vessels that are produced. So in this environment, now we have to consider also the presence of extracellular vesicles that are released from the all different types of cells. So there is like a Roman traffic of extracellular vesicles that are touch each other and have a contact with the other cells. We know that extracellular vesicles, as EXTRACELLULAR has established, have mainly the different sizes, small and large, because all the other characteristics are still in the discovering of the pathway, but we know essentially there are two different pathways. One is through cytoplasmic formation of multivesicular body, and the other one is through the budding. But as you can see, we have a lot of different types of budding. So this is in line of what we have already discussed and listened, that each type of cell decides which is the best production of extracellular vesicles. So which are our goals? Just to understand if the extracellular vesicles can shape the tumor microenvironment, and if extracellular vesicle can have an influence on the therapeutic treatment, and if this is the case, at which extent extracellular vesicle can influence the treatment, are also modulate the macrophages that are in the tumor microenvironment. So there are still a lot of questions and only few answers. According what is asked from the EXTRACELLULAR guidelines, we start first to analyze the presence of exosome and multivesicle extracellular vesicle microvesicles in situ. Then we established a method of isolation, and then we went to the characterization of morphology, size, and cargo of extracellular vesicle produced by 4 different types of GBM cells. We found, of course, because all cells have exosomes, and with the aid of FM, we analyzed the presence of budding not only on the cell surface in its whole, but by analyzing the different distribution in the nuclear and perinuclear area. After this characterization, we went to isolation, and we choose ultrafiltration plus size exclusion chromatography, and we went to the analysis at the electron microscopy to measure the size distribution, and what we found is a mess. Each type of GBM cell produce vesicles in a different amount and in a different size. Again, we start to analyze the role of these extracellular vesicles, and first of all, the sensitivity of the resistance of the different type of GBM cells. Then we went to the characterization of extracellular vesicle produced after the treatment with the temozolomide, that is elective drug at the moment for this chemotherapy against this tumor. And then we assess the role in some of the function of this cell. One is the migration rate. And what we found, these are the example of one sensitive and one resistant cell line that we could, after 28 hours of treatment with the drug, maintain the cells able to produce extracellular vesicle that could be isolated to analyze after the chemotherapy treatment. We confirm our resistance and sensitivity by analyzing the level of induction of apoptosis, and not only, but also ferroptosis, because temozolomide as a drug has an influence in the iron metabolism of the cell. And what we obtain is the confirmation that U87 are very sensitive, and only the resistant cells have an increase of the receptor for the transferrin that is necessary for the induction of ferroptosis. We isolated extracellular vesicle from GBM cells treated with temozolomide, and as a general results, we obtain that after the temozolomide treatment, the number of vesicles was increased, and was increased also the size. But very surprisingly, we found that by using cryo-EM, that is the only way to have these pictures, that there are a multilamellar structure. Perhaps there is something that is changing the generation of extracellular vesicle to modify in such dramatic way the vesicle itself. So these are the main data that we obtain. And again, we also analyze the topographical distribution, and we saw that some of the cells after the temozolomide treatment increase the perinuclear distribution of microvesicles of the budding. Others, they reduce, they modify in the nuclear part of the cells. By a preliminary analysis of the content of the proteins in the vesicles, clustering by the function, we saw that the cell migration and cell death cluster genes associated with this function were mainly affected, up and or down regulated. So with this idea of our tool in analyze the role of extracellular vesicle, we decide to treat GBM cells with extracellular vesicle isolated in absence of temozolomide treatment or in presence. But also we decide to see if the effect can be maintained or could be different when we took vesicles from one type of cell given to the other, to see the ability to spread the signal over the different complexity of the tumor microenvironment. And first of all, we analyze the internalization, the rate of migration with wood healing assay, and of course with cell death markers for apoptosis and ferroptosis. It's not easy to see, but here there are some vesicles that are fluorescent labeled. That means that even when we use cell and vesicle of different type, there is internalization. And this is just to show you what we are now studying, is that also macrophages are affected by the presence of this extracellular vesicle and start not completely to differentiate, but at least to have some idea that the environment is a little bit different. Okay, so we analyze the migration rate once we gave the vesicles from untreated and treated cell of the same species in a cross incubation. And what we found is that the U87 were the faster in closing the gap, that when there was the presence of the extracellular vesicles from treated temozolomide cells. Conversely, the resistant cells, they close in a long way the wound, and only okay, it's a little bit more different. Up to 24 hours, there is a decrement in the migration rate when are facing with extracellular vesicles, but after 24 hours, we don't know why, there is a rapid increase in the closure. So like a migratory rate increased. When we went to analyze the effect of on U87 cells, the vesicle from the resistant, we found that there is a decrement in the closure, both with the temozolomide treated and untreated extracellular vesicles. The same was with U251 cells, in which there is a decrement, but a decrement after 48 hours to speed up the closure. Considering all this data, we realize that the extracellular vesicle from sensitive cells increase the migration rate of both cell line, but were able to decrease the migration rate when derived from cells that were treated with temozolomide. So what the cells is elaborating by the presence of the drug is able to put in the vesicles and communicate to other cells. The same, we analyze the treatment on cell death, and what we found that only for U87 there is a significant increase in the apoptotic markers and in the ferroptotic markers, but only when, in only one condition, when the sensitive extracellular vesicle derived cells were given to the sensitive one. So they signal the sensitive property of their own. Okay. At this point, we stay on the way of our study. So we know something that we have to go further, because this is just the summary and I don't want to read, but there is a big communication. I can just anticipate that they can interact with the immunogenic cells, not only with the macrophages, but also, for example, with dendritic cell to modulate their activity. And what we are now thinking is which is the major player in this effect, in orchestrating all these effects. Are proteins? We don't think so. Are lipid? Perhaps are the highest player. Are nucleic acid? Why not? Because the vesicles are full of mRNA, so they are working a lot using this pathway. And this is the small stuff for this for this job. That is my lab, and the next speaker will be Stefano Tacconi, and we'll go on with our study of extracellular vesicles.
Thank you, Luciana. Great presentation. Is any question from the audience? Yes, please, Agnieszka.
Thank you. That's a very interesting study. I was interested in your vesicles in vesicles, those multilamellar structures that you showed. And why I'm asking, because we study osteogenic cells, and occasionally we see similar structures, and we always discarded them as artifacts of ultracentrifugation. But now with your study, it's like I'm going to go back and revisit that thought, because clearly they may have been different kind of vesicles with different biological functions. Is that what you think?
Yes, this is exactly what I think, and I think that extracellular vesicles can, when are multi-structured, can have two different biogenesis, because we can have the one who are concentric, okay? So that is not fusion, that is not due to centrifugation or whatever, or at least not only. But the other, the one I show you, are vesicles. Some are concentric, then are included in a large one and another large one. So could be a sort of internal phagocytosis, I don't know, but are completely different. And what we are doing is try to find the percentage of these figures in the different conditions. That is not so easy, because the number in cryo-EM are not so big as in other assays.
Thank you.
Yeah. More questions for the public? Can I, no, just one comment, question for Luciana, because I had the same question of Dobrava. Could be that the cell are basically shedding mitochondria, because mitochondria inside vesicles, they can be shedded through different type of extracellular vesicle, like with single bilayer or multiple bilayers. When I saw those pictures, but this doesn't seem to be, because those are cryo-EM images, so they don't lie, basically.
Okay. With cryo-EM, you can see all this procedure not just with the isolation of extracellular vesicle, but with a much more technical, difficult analysis of the whole cells. In any case, in our experience, we analyze the formation, not of course with the cryo-EM, of amphiosome that are a fusion of the two internal pathway, the autophagic pathway and the multivesicular body. I don't know if this is the case, or when you are producing migrasomes, you can pack the vesicles that are moving in a different way. In the case of the amphisomes, you can have a part of damaged mitochondria, but at that point it's difficult to discriminate between the autophagic or extracellular vesicle pathway. But
Thank you very much. So we move to the next speaker.
Okay, we move with Stefano Tacconi from Sapienza University. His work on method isolation, characterization, and study of biological activity of extracellular vesicle in different in relation to the disease, and also for the application in biotechnology. And he presented lipotoxicity, a new role of lipid cargo in extracellular vesicle from lipid-associated macrophage. No.
No.
Stop passing.
Thank you for giving me the opportunity for do this talk to the organizing committee of this conference. So I'm really interested in the study of the role of the lipid composition of extracellular vesicles, in particular about the lipotoxicity, which is associated with, this is not me, but obesity is associated with a lot of the other collateral alteration, like, for example, dyslipidemia. So during obesity, you have an increase, for example, of circulating free fatty acids in the blood. These circulating free fatty acids can be internalized from adipocytes, so they start to produce triglyceride, and when this excess is too much, they start to degrade triglycerides, so to produce other free fatty acids that can be, of course, driven to the liver and the skeletal muscle, but also taken by other neighbor adipocytes, which are start to accumulate other triglycerides, and in the end, you have a process that is called adipocyte hypertrophy. There are several mechanisms within the adipose tissue just to prevent this process, and one of these are macrophages. In particular, in my work, I'm strongly interested in studying this specific population of resident macrophages, which are called TREM2 macrophages. These TREM2 macrophages, also named as lipid-associated macrophages, so LAM macrophages, generally they produce a crown-like structure within the adipose tissue, so around the adipose tissue, and their important function is to internalize free fatty acids accumulating in formal lipid droplets, just to prevent the toxic fat accumulation within the adipocytes. These TREM2 macrophages are specific for different surface and intracellular marker, in particular this protein, surface protein, that is fatty acid receptor, that is called TREM2, and other like PLIN2, FABP4, CD36, and so on. In this very interesting paper, they found that if you remove this type of macrophages within the adipose tissue, you increase the weight gain, the glucose intolerance, the fat accumulation, the adipocyte hypertrophy. So they have a protective role for the adipose tissue. But what about extracellular vesicles that are released from this type of cells? So we have built this type of model by using THP1-derived macrophages that we have filled these cells with fatty acids, palmitate and oleate. So we observe first the accumulation within the cell, so lipid droplets, as you can see here, by night-red staining, also by electron microscopy. And by performing T-layer chromatography, we observe a strong accumulation of triglyceride in these fatty acid-treated macrophages. So they are able to accumulate lipid droplets and triglycerides. After we checked for the expression of specific marker that characterize the TREM2 subpopulation of macrophages, and we observed that all the markers TREM2, PLIN2, CD36, and FABP4 and ABCA1 that are specific from TREM2 macrophages, they increase after the treatment with FFA. And this type of macrophages have a more anti-inflammatory phenotype. As you can see here, there is an increase of secretion interleukin 10. But what is characteristic for this type of macrophage is not only the expression of specific marker, but also the metabolism. In fact, they increase the expression of diglycerol acetyltransferase that can enzyme the synthesis of triglyceride, but they are not able to use this accumulated lipid by beta oxidation and respiration. mitochondrial respiration. That is very characteristic for this type of macrophages. You can discriminate, for example, this type of macrophages from, for example, foam cells from the vessel. What about EV? First of all, by just conducting transmission electron microscopy analysis, we observe that some of the lipid droplets are strongly connected to the plasma membrane. And performing scanning electron microscopy, we observe a very heterogeneous population of extracellular vesicles. It was really interesting for me is to see that after the treatment with FFA, you reduce the production of refractosomes. Refractosomes are the last part of this large microsome that are produced during cell migration. Also, by same analysis, we observed that these cells treated with FFA, they produce a very large budding vesicles, and they seems like bigger like 1 micron, so like lipid droplets that are pushing on the plasma membrane. You can see here they here are going to be released, and here these huge large particles that are totally released in the extracellular environment. In fact, if you perform the size of these vesicles, they have a range diameter between 800 nanometer and 1 micron. So they are very, very large. We confirm also this type of phenotype in human-derived macrophages, so it's obtained after differentiation of monocytes, blood monocytes. And if you treat these macrophages, you can observe also here lot of lipid droplets that are pushing just the plasma membrane of these cells. Also, in human macrophages, when you do the staining of lipid droplets, we observe the presence sometimes of these round, large particles just outside the cells. But was also interesting for me to see other way of communication. In particular, macrophages, they are able to produce these long tubes that are called generally tunneling nanotubes, and they are able to transfer lipid droplets to other recipient cells. So lipid droplets, they can move, they can be transferred from one cell to the other one. You know, okay, this is a lipid droplet, so it's very important for the structure of the lipid droplet, particular surface protein that are called prin-2 proteins. Differently, for EV biogenesis are very important these microdomain on the plasma membrane that are enriched in tetraspanin that are specific protein of EV for EV formation. In particular, for large EV budding for the plasma membrane, very important is the CD81. So we try to combine these two markers, so PLIN2 CD81, they can co-localize. So the release of lipid droplets can be also associated with this tetraspanin-rich domain. So we do the labeling of lipid droplets with PLIN2 antibody without any permeabilization. So we are just seeing on the surface of the cells the presence of these lipid droplets. So you can see here the cells have a lot of lipid droplets, but just one part of these are labeled for PLIN2. And what we observe that PLIN2 co-localize always with this group of lipid droplets that are going outside. It's not very visible here, but here you can see the CD81 raft domain, and there is a budding that is here, budding droplets just going out from the cells. So we moved to isolate EV from our experimental model by differential centrifugation to obtain the large and small EV population. We, of course, performed the classical TEM analysis to see the morphology of large EV fraction, small EV fraction, the presence of EV-associated markers, CD81, CD63. Also the size distribution by dynamic light scattering, and what we observed that was very interesting, that only in the large EV fraction we found, of EV isolated from FFA-treated macrophages, we found a second peak around between 600 nanometer and 1 micron. So very large particle that we also analyzed by transmission electron microscopy. We performed the lipid profile of this EV by nuclear magnetic resonance, and also here we found the presence of triglyceride and diacylglycerol only in the large EV fraction from FFA-treated macrophages. In order to specifically demonstrate that fatty acids can be incorporated in triglyceride and they produce EV, large EV, that can be filled with lipid droplets, or they can release directly lipid droplets outside, we perform this experiment where we use C14 radiolabeled palmitate together with the mixture of fatty acids to induce the accumulation of radioactive triglyceride within macrophages. After we isolate this EV, this radioactive EV, and we perform thin layer chromatography, and we so determine the level of radioactivity just to be sure for the presence of triglyceride within EV. We found that only in the large EV fraction were present triglyceride, and not in the small. This EV can be internalized. They can transfer triglyceride on recipient cells. So we use this EV to treat and recipient macrophages, which we inhibit the acylglycerol transferase, that is the key enzyme for endogenous triglyceride synthesis. We also perform thin layer chromatography, and also in this case, radioactive tag was present only in macrophages treated with large EV. So tag can be incorporated in large EV, and this can be transferred in recipient cells. We also analyzed the effect in terms of activation of these macrophages. So we observed that the treatment with large EV increase the same TREM2 marker that we observe in donor macrophages. So there is a sort of transfer of the phenotype mediated by these lipids. So, just in conclusion, macrophages treated with FFA, they produce a lot of EV, a very heterogeneous population of EV, including also this type of microsomes. But what was interesting for us, that they are able to release, and we don't know yet, but I hope soon, if they are just extracellular lipid droplets released in the extracellular environment, or are lipid droplets which are enclosed in very large particles from the plasma membrane. But this, in both cases, these EV are internalizing recipient macrophages, which modulate their activation toward the same phenotype. So I want to thank you, all the people that are collaborating with me, in particular, of course, Professor Dini and Dr. Sophie Rom from University of Lyon, Thank you, all of you, for your attention, and if you have questions, I'm here.
Thank you. So some question from the audience?
Thank you, Stefano. Very great talk. I have a couple of questions. The first one was if you, maybe you're looking at it now, if you see the triple leaflet, phospholipid leaflet in your vesicles. So because if you have lipid droplets enclosed in vesicles, you see
just one.
Ah, okay.
Okay, no. You're right. To discriminate between extracellular lipid droplets, you mean enlarged EV with the multi-layer.
Yeah, you should see 3 layers of phospholipids, like the one from the lipid droplet and the one from the extracellular membrane, let's say the membrane, okay, double layer.
Yes, just in 2 days I will be in Pavia just to perform cryo-EM. Yes. Just to see if we can, in this large EV population, specifically see these droplets with single layer, so they are extracellular lipid droplets, or 3 layer, so lipid droplets containing a large EV. So we are going to, and also what I want to do is just to optimize the protocol of the isolation. So on large EV, there is a specific protocol to isolate lipid droplets. So we are going to isolate lipid droplets by gradient centrifugation from the large EV fraction to see if we can separate the 2 populations.
Okay, thanks. And the second one, just a curiosity on a technical challenge. So how many vesicles do you have to analyze to get the lipidomics by NMR? So how many vesicles do you need to have? Do you know, like
a lot. If I remember well, 100 microgram.
Okay.
100 microgram of protein of EV.
It's a lot. Yes.
That means 90 million of cells. You isolate EV from 90 million of cells, 100 million of cells.
Okay, we have to move because we are a little late.
Thank you.
Thank you.
So next speaker, I hope that we will say the right title this time. Yes, 2 on 2, huh? Not completely. So the next speaker is Professor Claudia Maria Radu. She is an assistant professor at the University of Padua in the Department of Medicine. Her research focuses on the study of the role of extracellular vesicle in the onset of coagulative diseases and the potential use as a biomarker in diagnosis and therapy of pathologies by means of flow cytometry and confocal macroscopy. I'm going to use this one. So her title today, she's going to talk about the high sensitivity flow cytometry for the analysis of extracellular vesicles, new developments in the research and clinical application. Thank you, Claudia.
Dear colleagues, good morning. Many thanks in the scientific organization and to Professor Claudia Matteucci and Professor Claudio Bottini for this invitation. From the cells, flow cytometry from the cells to extracellular vesicles, the big cells to the small EVs. EVs, you can see here some cells that release some bits, some EVs, extracellular vesicles. We go from the cells that have a big size to something more small, very small. The apoptotic bodies, the microvesicles, like platelets, remember that have 3 micrometers of size. And then we go to exosome 50 to 150 nanometers, like a virus. Remember SARS-CoV virus. What is the term to say extracellular vesicles that you have to use now? Now we have to use extracellular vesicles because it's a generic term to say all the secreted cells Vesicles that characterized by a phospholipid bilayer membrane and can don't replicate because you don't have any nucleus, and then have the size. You have remember the size, nano micro-sized particles, and released from all cell types after cellular activation, injury, or apoptosis. The nomenclatures now it's correct that we know that have exosomes that coming from the inside of the cells outside, for the compartments of the inside of the cells, by exocytosis and the multivesicular bodies. I have a very small size, 100 nanometers. Then we have ectosome, microvesicles or microparticles, the old nomenclature. That means cells that coming from outside of the membrane after injury or apoptosis, but have a fragment of 100 to 1 micron, 100 nanometers to 1 micron. And the last is the apoptotic body that is very big. We have different society that works in the extracellular vesicles for the definition, the nomenclature, how to isolation, the characterization. We have some database that provides to collect for all the groups that work with extracellular vesicles to explain what have, to found what have inside, to what is the DNA, the cargo of these lipids, the membrane composition, etc. This is of International Society of Extracellular Vesicles is all the task forces. Any groups have specific rules in rigor and standardization collection of data. The last minimal information for studies of extracellular vesicles guideline gives some information for the research, how to work, what is the limitation of detection of extracellular vesicles, the isolation, and what is now the definition of extracellular vesicles. Small extracellular vesicles and large extracellular vesicles, because when you have writing a paper, you have to write a paper, you have to make attention of the nomenclature that you use. Small EVs means under the 200 nanometers diameters, large EVs means up of 200 nanometers of diameters. This is the recommendation by the society. And what about exosome and ectosomes that you know that you use? We know that it's from the inside or outside of the membrane. The problem is you can use this, but you have to explain what is the markers for internal or external AV's composition. And you have to be sure that what you see is exosomes or ectosomes. Oops. Blocked. Okay. And why this? Because from 2014 to 2023, we have some differences. We have some recommendation, because now the focus is that we have other types of vesicles, like the lipoproteins, the exomers, all the other lipids vesicles, non-vesicular extracellular particles. And why to study extracellular vesicles? Because we know that the membrane have some composition of lipids. Inside we found genetic materials that can regulate some process of proliferation. Remember the metastasis of cancer. And have inside transcription factors, cytokines, and crosstalk, one cell crosstalk with the other. Microvesicles go from the cells to another recipient cells, but remember, from cells to organs. That is important of the vesicles is as effective biomarkers for cancer, because if you see all these types of cancers have different composition of inside of proteins, of inside DNA, genetics information, microRNA, DNA, and it's important because we use as biomarkers, that is indispensable tools in clinical application for the disease, for the progression of the disease, for the metastasis. And not only, we can use as promising carriers for drug delivery, for control the tissue regeneration, for the angiogenesis, for suppress apoptosis, and others. When you see the constellation, you use a telescope. And when you have to detection of EVs, what happens? We have different systems. We can use differential ultracentrifugation. We can use nanotracking for the count, to understand the size distribution, the phenotype, the flow cytometry. But all these things have some positive and negative parts for the detection of extravesical. We can have a good recovery, but or loss recovery. In base, if you have if you make ultracentrifugation, you can lose some EVs. Or if you use chromatography, you can obtain isolation of some needs, other you can lose. What do we have to do? We not have an optimal separation methods. We not have a perfect quantification. We not have genetic markers for all these extracellular vesicles. To centrifugation, you can lose, not lose, depends of your population of EVs that you want to detect. And important is to provide all the indication in the papers what is the limit of detection of your instrument and what your instrument can see. And what we have to do, try. This is my opinion, try. And maybe you find the correct way. For the standardization, from the past to now, we have more sensitivity flow cytometers, but now we have again the problem of the standardization of the calibration of EVs, and then remember that most of cytometers can not go under the 500 nanometers of detection, and then the light scattering theory that for the calibration you use some bits, and it's not the same to use really extracellular vesicles. Now, say used for the detection the fluorescence signal and staining with CD61 is a marker of platelets EVs with the side scatter, because platelets EVs is the most extracellular vesicle circulate in our bodies. And this is a best practice for the flow cytometry detection. You have to go with a side scattering and CD61, and then you can gate for your EVs, your gate. But you remember that you have a general information about the sites. The problem is, what about under 500 nanometers? That is the most EVs that circulate in our bodies, in all the fluid liquids, in the synovial liquids, in the urine. The quantitative information, we have the problem. You can use nanotracking, but see not only the extracellular vesicles with a bilayer membrane phospholipids, but found only with a single phospholipids. And with some polystyrene bits, we can use the megamix from contain different sizes of bits, and you can optimize our gates. But remember that it's polystyrene bits, that it's not the same to use really EVs. It's, the refractiveness is more higher, and maybe we lost some information about the small EVs. And Cytoflex is one of the cytometers of the family, the basic, that have a solid state lasers and advanced photodiodes, and is equipped with a violet size scatter that can trigger for understand the noise for our extracellular vesicles. You have the band pass. It's like a confocal microscope. You can see in specific the emission of your fluorescence. And if you use the gigamix, that is the mix of these types of polystyrene bits, you can see all the different population with a different diameter of these bits, 100, 500, 900 nanometers. And if you use a size scatter without the violet, you can see that it's difficult to distinguish the different population of bits. The same, if you want to design a gate, you can use these bits to design a gate. It's a reproducible gate, 1 centimeter for the other, because the standardization focus in the standardization of the detection of the gate for analyze the extracellular vesicles. And this is one way to use this type of bits. If you see, you have difficult to see the smaller EVs. The same, if you go to see the fluorescent intensity, you see the difference bit from the noise is very distant if you use the violet size scatter. This is a paper in some patients with hospitalization with COVID, and you see the different population of EVs in the plasma. This from endothelial cells, because you know that with COVID patients, we had the problem of activation of coagulation and thrombosis and circulating microthrombosis, and you see the different population of EVs bearing tissue factor, that is initiation of coagulation, and when start, we have hypercoagulability of the patient and we have thrombosis. And for the cytometers to the confocal system, we see that really these vesicles are present with tissue factor expression, not only, but we assay to a marker of COVID. And in the confirmation coming not only from the microscope, normal microscope, but from electron microscopy, that you see the dimension of EVs, but nothing else. You don't have any information, but electron microscopy. And with Cytoflex SRT, we want to try with the same system, have the same system like Cytoflex, to try to see if you is capable to isolate these EVs. We have the possibility to isolate this and to see what happened. Yes, we can isolate because have the same system with the Cytoflex normal, have the band pass, have a nozzle for 100 nanometers, and you can design again your gate like this, and you can see the fluorescent intensity. If you use the violet size scatter with FITs or other markers, we can isolate your EVs. And we try to isolate for sorting 1-2 population of EVs. And we see here after the isolation. It's normal, it's not possible to isolate one EVs because you not have any information about one EVs, and it's impossible. But using these bits, you can isolate some population of EVs. And, but I want to see if it's possible to isolate one EVs, one polystyrene EVs. Remember, the refractive index is more higher than the normal, but I try. I try to see if it is possible, and it is possible, of different size. But to be sure, I go to see in the microscope, and I have the confirmation that I have only, in my droplet, only 1 bit of a specific dimension. Can do it. We use this system to see what happens in the patient with COVID virus infection, and we isolate SARS-CoV spike 1 and with calcine together, because calcine is something that gives me information about the inside of my extracellular vesicles, that are real extracellular vesicles, because it was fluorescent only when exterases of extracellular vesicles cut this fluorescent calcine. And after the isolation, we take these extracellular vesicles and we put in the human coronary cells, vascular cells, and we see the expression of tissue factor, the activation of these vascular cells. Not only we found the spike 1, that means that the virus go around with extracellular vesicles. And now we have Cytoflex Nano in our labs, and is for very small bits. We can arrive to 40 nanometers. you use the polystyrene beads to see the sites that it's possible to see in these cytometers. We have, remember when you have go to detect some extracellular vesicles, you know, to have to use more antibodies. Here you can use 3 or 4 antibodies, but the same for the other cytometers. My opinion is 1, 2, 3, not more to 4 antibodies, because you have the problem also of the spheric of the antibodies, yes. And have 5 size scatters. We try again the possibility to see the different population of different beads, have a sensitive monitor report, go very low with the extracellular vesicles detection, and we use borosilicate glass tubes for the extracellular vesicles. If the system is very clear, very cleaning, you have to clean it very well, and the sample flow rate is 1 microliter per minute, means very, very low. And the other things is very precise instruments, have to be very well cleaning, and to have more patients, more patients, because need very cleaning, back flush from one tubes to the other tubes, and need calm, very calm. We try again with the polystyrene beads, and we found that have this possibility to see very low. Look, the noise is very low noise. You have 2 types of violet scatter. The violet scatter 1 and 2 is very sensitive, but if you see up to 300 nanometers, you have the problem to see these extracellular vesicles, because it's a nano, it's a baby cytometers, and see only under the 300 nanometers very well, but not more than. And we try to see rainbow calibration particles, and we see that it's very, very easy to use, and the different population between the different one-to-one population with different sites. And we try to use AISON columns to purify it. This is PBS, and this is our gate for extracellular vesicles. And after the isolation, we obtain this. But we tried without isolation in a patient with thrombocytosis, and you can see very well the difference in the small extracellular vesicles, the blank, and with calcine in the patient, only with calcine. This is another patient with the same thrombocytosis. It's the second patient. You can see the CD41, the gate that I proposed before to use. The CD61 or 41 is for platelets extracellular vesicles, and you can see the difference with the blank and after the CD41 staining. What is the problem in the extracellular vesicles? How to detect real extracellular vesicles means the bilayer of extracellular vesicles. Now we have a new ACO dyes, is some dyes with a particular composition, is oligoelectrolytes that go inside the double phospholipid membranes, in the membrane of the double phospholipid membrane, and give the possibility to understand if really we have extracellular vesicles with a double bilayer. And this is the results. We make our gate, and we use ACO 1490, is a dye for this laser, and we obtain the positive. And after the positive in ACO dyes, we go to see the staining in the violet for a scatter, the staining of extracellular vesicles with a gate with polystyrene beads, and after annexing phi and calcine, we obtain the double positivity. Now we can say, yes, it's really extracellular vesicles with a double lipids membrane, phospholipids membrane, because it's a conjugate oligoelectrolytes that go inside the bilayer of phospholipids. This is a new dyes for extracellular vesicles. Remember, casein for the really extracellular vesicles, because inside the extracellular vesicles we have esterases, the ACO dyes for the bilayer lipids. And for the problem of the polystyrene beads, silicone beads, that we have the refractive index that different with the really extracellular vesicles. Now we have the Cellarcus. I not have information about this because I don't use it. It's in the Italian airport now, and I stay here for 1 week, and I hope to obtain it. And this is a kit that have inside really phospholipids, artificial phospholipids, that with a refractive index the same of extracellular vesicles. This is an example of SRT that we use for isolation in patients with diabetes and with carotid plaque, fibrous or calcified, and we see the difference in these patients of the contact of mineral with next generation sequencing. That to say what is important to understand what have inside our extracellular vesicles. And the same with urine. This is the last experiments in the urine in the Fabry disease, and you found increase in soluble interleukin-6 receptors in these extracellular vesicles, important for the prognosis of these patients. And you use artificial extracellular vesicles, some vesicles nanoparticles, with the cargo with an mRNA, is a fluorescent mRNA to see in the mouse model the clearance of these extracellular vesicles. And we see when use the mouse, we found in all the organs the extracellular vesicles, and we see what is the time of clearance in the different organs. And now EVIS is important not only for as biomarkers for different disease, but for the vaccines, remember the COVID vaccines, and for the cargo of different therapeutic drugs inside for the different disease pathologies. And with this, I want to thank my young staff of our labs that work with extracellular vesicles in Padua. Thank you for your attention, if you have any.
Thank you, Claudia. Very interesting presentation, and also very informative, because in this day we have a lot of people that arrive and maybe is not expert in extracellular vesicle and want to know a lot of things about this, and we try to be more informative as possible.
I have a presentation of 60 slides, but I say no, because it's impossible. If you
and we have to take the coffee, we are late also. But anyway, I have some comment, but I would like to know if someone wants, okay, if there is some question.
Thank you for this overview. I have just a curiosity. So you measure the limit of detection of the Cytoflex Nano by using beads, right? And it's 40 nanometers. But
for the nanoflex?
Yeah,
okay.
For the nano Cytoflex. But ever you try to understand which is the limit of detection in terms of nanometers using, for example, the Rosetta bead system?
Is the same, the Rosetta beads, yeah, we can use this. You see a presentation of the Beckman Coulter of the company. We use Rosetta is the same like the Gigamix, because it's the same polystyrene beads. And you have the same results. But the problem is not the same, that the refracting index, I have other slides, but the refracting index from Rosetta from NITS, and this gigamix is that is polystyrene beads or the same with the silicon beads. The refractive index is more higher than the real extracellular vesicles. That is weight from Celarcus, because it's not in commercial now, because it's phospholipids, really phospholipids with different sizes, and we can use to understand the really low detection of these instruments.
Thank you.
Here.
Just a curiosity, this instrument is able also to produce a size distribution of EV, because, for example, with Rosetta calibration system, by using the refractive index, you can reproduce after the size distribution of EV.
Yes, we use Rosetta, the same the NITS and the other beads, and the gigamix. You can reproduce the same identical gates, the same identical information with the 3 cytometers. With all the 3 cytometers, Cytoflex, Cytoflex SRT, and
Nanoflex.
And Nanoflex.
Oops.
We use the 3 cytometers, and we check, but I can't see all this, and we have the same results. That is important for the standardization of the system of extracellular vesicles.
Of course.
Of the gates to analyze. But I say that is polystyrene or silicon beads, because we use silicon beads, and we need something really near the refractive index of these extracellular vesicles.
Thank you.
Okay, thank you. I have a lot of questions, but we
we
need to stop
cut
them
if you
because
want.
we are late of half an hour, and but we will have to go on this discussion. We can take a break. I don't know if we can take if 15, 10, 15, not more, because we are late with the...
We're a little bit late, so please.
Thank you. Sorry, so we have to start. Okay, we'd like to start with the second part on matrix vesicle and invite Professor Zleka Kiliskę from Polish Academy of Science of Poland. Her research interest is on vesicular transport, median matrix vesicle, annexing and calcium homeostasis, bone mineralization, vascular calcification. And the presentation is the difference between media and matrix vesicle as a possible diagnostic tool to monitor the progression of cardiovascular and skeletal disease.
Hello everyone, and I'm Agnieszka from the Nansky Institute, and I want to thank organizers to inviting me for the meeting. Today I want to focus on a difference between the media and matrix vesicles as a diagnostic tool for the cardiovascular and skeletal disease. As a background, I want to mention that when I told about the mineralization process, I think about the physiological process which is present in the mineralization competent cells and pathological possible in vascular system. And it means that the cells should differentiate or transdifferentiate depending on the situation to produce the minerals or some plaques. And of course, different enzymatic systems are engaged depending on the changes of the calcium to phosphate ratio or some inflammation. The same was mentioned by the Professor Milan, but today I want to focus not about the PI turnover, rather protein and calcium turnover. We are a specialist in Nansky Institute in our lab about the calcium and phospholipid binding proteins, annexins. We hypothesize that vesicles may be a membrane domain microdomains models. They have a lot of lipid markers of rafts and a lot of protein markers, for example, TNAP and annexins family. Annexins are the phospholipid and calcium binding proteins. They engaged in a membrane composition, and today I also want to focus on phetuin A as a typical inhibitor of the mineralization. The vesicles may modulate the mineralization process by the decreasing of the bone mineralization, which are involved in osteoporosis or bone cancers, or may increase the vascular calcification, leading, for example, for arteriosclerosis. We used different cellular models of the mineralization in our lab. We have 3 human cell lines, HFOB line, which is a model of human osteoblast and physiological mineralization. We also use the SOUS2 cells, which are the human osteosarcoma cells, as a model of pathological cancer mineralization, and also human coronary artery smooth mass cells, which are the human vascular smooth mass cells, and model of the pathological arteriosclerosis model. We culture them with the different conditions by the 21 days in resting and stimulated state. Stimulation for me means that we add special osteogenic factors like ascorbic acid and beta-glycerophosphate, so sources of the calcium and phosphate. And we also want to modulate this mineralization using inhibitors like apigenin, like K201, and some inhibitors of the TNAP, like levamisole and MLS. And we isolate them. We isolate the vesicles, media vesicles and cellular vesicles, matrix vesicles using the collagenase digestion, and also separation by two-step ultracentrifugation. And we also use three potential modulators. Why apigenin? Because this is a plant flavonoid present in the green part of the plants. We eat it every day, and from the earlier papers, it was known that they may neutralize free radicals, may prevent oxidative stress, and may complex different metal ions, and they are very useful in our diet and may modulate the apoptosis and osteoblastogenesis, and not so many information about influence of the flavonoids, for example, apigenin, genistein, or naringenin present in fruits, how they influence on the mineralization of the bones, how they influence on the mineralization in soft tissues. And together with this unknown modulator, we compare the results of known from the literature about the calcium channel regulators, for example, K201, or inhibitors of TNAP. So we used MLS. Inhibitor of the calcium is calcium-dependent blocker of the SERCA system. It regulates calcium levels in cardioprotective as a cardioprotective has cardioprotective properties. But MLS is a specific inhibitor of the TNAP without any influence on other phosphatases or phosphodiesterase, and this influence is uncompetitive to the phosphate donor substrate, and has different inhibition mechanism. So our results present that that after stimulation by the 21 days, we have increase of the number of the cells and production of some minerals. After the apigenin treatment, we think that we have even more mineral, but using the calcium and phosphate inhibitors, we have the problem with the cell growing and mineralization. So next, we checked the presence of the minerals. So as you see, flavonoids even increase the production of the calcium salts. And of course, the activity of TNAP is also increased, but depending on the isolation of the type of the vesicles. We use this protocol. We collect the media, and after two-step ultracentrifugation, we have cellular vesicles, extracellular vesicles, or media vesicles, depending on the literature. But when we use collagenase treatment through the 3 hours and 3 steps, we have matrix vesicles named MVs or MAVs. It means collagen-attached vesicles. And depending on the vesicles, we see that extracellular vesicles, we have not so strong effect of the inhibitors of the calcium system. Of course, apigenin only, we have some signal going down when we use the TNAP inhibitors. But for the matrix vesicles, we have much stronger influence. Then we look through this type of cells on the colocalization of the annexin and phetuin, which may be inhibitors of the mineralization, and how they colocalize with the TNAP. We use fluorophores and yellow colors on the pictures mean that they colocalized. We calculate the percent of the colocalization, and after the stimulation of the cancer cells, we have a little degrees of the colocalization between the annexin 6 and TNAP in cancer cells, but no effect of the inhibitors of calcium and phosphate. But when we stimulate the cells, we have influence of the apigenin much stronger than other inhibitors. We also use the nanotracker to see how the vesicles look like. So after the apigenin, we have no effect on the mean size of the vesicles, but we have very high level of the increase of the number of the vesicles. But when we use the inhibitors, we have influence on the size of the matrix vesicles, and of course we have much more number of smaller vesicles. Then we want to focus on the structure of the vesicles and minerals inside the vesicles. So we use transmission electron microscopy connected with the X-ray microanalyzer to see the structure of the mineral. We want to see phosphate ions and calcium ions, so we use this system in the Nensky Institute. It's possible to do transmission electron imaging, also X-ray analyzing of the ions, and tomography of the vesicle. So we have three systems in one column. So we have a traditional TEM-SEM in the middle part. We have tomography equipment and EDS equipment. It means that when you put the electron beam on your sample, one electron is kicked out of, and some X-ray energy is emitted. And as the results, we may see images of the extracellular vesicles and matrix vesicles, and you see that the matrix vesicles from the same cell line, they are bigger, they are full of mineral, and we may do the ion spectra calculated for components as we wish from the Mendeleev table. For example, we have increase in phosphate comparing to the extracellular vesicles and the calcium, and we may calculate also the ion maps, for example, for phosphate on the red and calcium green color, and we may calculate the calcium to phosphate ratio. Of course, we may see also the electron microscopy tomography. We may put the antibodies to the vesicles connected with the nanogold particles. For example, here we have annexin 6 with the 10 nanometric gold particles, and TNAP with the smaller one, 5 nanometers. And here you see the circles when we may found colocalization of both proteins. So in control situation, we have vesicles full of the mineral, even with the needles going out from the vesicle. A lot of colocalization. After apigenin, we have huge needles, but not so many places with the both proteins together. But when we treated the cells and vesicles with the calcium inhibitor, we have almost empty vesicle, only a nuclear core of the production of the mineral. Completely different situation we have in matrix vesicles using the TNAP inhibitor. We have probably nano drops of the mineral inside. They are full, but still no colocalization. So we put our minerals to the Fourier transform infrared spectroscopy with our collaborators from Lyon, and we may see if we have calcium to phosphate ratio changes. So after stimulation, we have decrease of the control cells and cancer cells in media vesicles, but increase in matrix vesicles, and only this type of matrix vesicles, they have similar structure as pure hydroxyapatite control. Other situations mean that we have artificial calcium phosphate minerals not similar to hydroxyapatite. And as the summary, I may say that the mineral formation was stronger affected in matrix vesicles than media vesicles, using both inhibitors for the calcium transport or phosphate transport, and maybe a pigeonine is a modulator, disturb only the intracellular distribution of both proteins, annexin 6 and TNAP, influence mainly in matrix vesicles on the colocalization, and both activators, I mean annexins, and inhibitors, phetuin, may influence on the mineralization, and they are recruited to the TNAP enriched membranes, and we think that both the structure and the calcium to phosphate composition of minerals were typical for hydroxyapatite in matrix vesicles, but in media vesicles we have artificial calcium phosphate. And as you see on some published papers, we thought that annexins may be stimulators in this situation, and apigenin may act double pathways, may block colocalization of the annexin with the TNAP, because the annexin may be attached on the inside part of the membrane with the some phospholipids, and after the signal may flop from the to the outside part of the membrane, or apigenin may directly stimulate the hydroxyapatite production. So thank you for your attention, and I also want to thank my collaborators.
So thank you, Agnieszka, for your presentation. Any questions for Agnieszka? Yep.
Thank you for your presentation. I have just a question, a curiosity. There is a way, a procedure to, because if I understand well, you have a mixed population of EV and matrix vesicle. There is a way to discriminate or a protocol to isolate differentially these two populations?
As I mentioned in a scheme, different additional ultracentrifugation for the matrix vesicles.
High speed, 250,000 G.
Higher speed.
Okay.
Or, of course, the collagenase treatment. When I remove the extracellular vesicles, I have more pure population, but of course, with matrix vesicles, it's possible to isolate also the multivesicular bodies. So why I add one more step? Okay. Yes, if I come back, it will be the protocol here. Yes, so I add one more step to isolate the matrix vesicles. So extracellular vesicles, I not use any collagenase.
Okay.
So they are attached to the collagen.
For example,
and I have some more marker because I not focus on this scheme because Professor Milano said a lot, but for the annexins, I have annexins only attached to the phosphatidylserine inner membrane, but annexin 5 and 6, they only these two create the calcium channel, and after the stimulation, only annexin 6 and 5, they go outside of the membrane. So I may separate them from the other vesicles. So the annexins may be the markers of the matrix vesicles, only that one which they create the calcium channels.
So maybe also by sorting using this marker.
All extracellular vesicles, media vesicles, they contain only annexins inside attached to the phosphatidylserine. But these two, they go out and attach to the phosphatidylcholine, what means that you have accumulation of the hydrogen ions and lower pH.
Okay.
Because they are pH dependent. So this is, I stain the vesicles mainly matrix for annexin 6.
Okay, thank you. Very clear, thank you.
Okay, just a curiosity. The first is a technical curiosity. I see that you use osteogenic media, but without dexamethasone. This is specific aim, or?
I use the dexamethasone only on for vascular smooth muscle cells. For bone cells, I never used because it's increased the level of the calcium and magnesium ions, but annexins are calcium and magnesium dependent. So if I use the dexamethasone, it will be high expression of the annexins. So I resign from that. But I know that for vascular smooth muscle cells, it should be because they transdifferentiate better than bone. But for bone cells, cancer cells, I always use the media without any calcium and magnesium.
Okay. And the second one is if you tried the same experiment with, in particular with epigenin, so I can understand, in primary cell culture, in human primary cell culture from bone.
We plan to do it next time.
This was a curiosity.
Now we finished some cardiovascular disease grant, and now we want apply for the second one using the epigenin more.
Thank you.
Okay, so we have to move on to the next speaker. Thank you very much, Agnieszka. So next speaker is Professor Dobrava Napirala, that is Associate Professor at the Department of Oral and Craniofacial Sciences and Center for Craniofacial Regeneration at the University of Pittsburgh. Her research focuses on the molecular regulation of the biomineralization processes in skeletal dental tissues, and she's interested in phosphate signaling, biogenesis of matrix vesicle, and molecular networks of the TRPS1 transcription factors. Without doing any mistake, her talk today is matrix vesicle secretion and molecular composition is regulated by phosphate ions.
Close enough.
Thank you.
Hello, everyone. We have to read the one. I think we will read the words in each slide because.
So this is the motif of today, basically.
I would like to start from thanking the organizers for assembling this fantastic meeting. So far, the morning session has been very inspiring, and I have great time. And thank you for inviting me to talk about our research. So my lab is interested in phosphate ion, and this is a biocompatible form of phosphorus. And you already heard about this ion today. It's a very important molecule for biomineralization, for formation of mineralized tissues in human body. Before I go to the project, I would like to acknowledge people who contributed to this study. Most of the work was done by Sandeep Chowdhury, who is now working for a pharmaceutical company. Unfortunately, academia lost a great person. But also many other people who contributed to this project over the years, our fantastic collaborators and the sources of funding. So mineralized tissues, we have 5 different mineralized tissues in human body. 3 of them are in the tooth, and 2 are skeletal, mostly bone, but also very unique type of cartilage, hypertrophic cartilage. And what's common for these tissues, why we call them mineralized, because other than organic components, they have an inorganic component, mineral. And that mineral is deposited in organic extracellular matrix in a highly organized way. And probably you heard many times the term calcification, which basically means mineralization. What I don't like about the term calcification, it highlights the role of calcium in mineralization, when indeed it's calcium phosphate crystal hydroxyapatite. So because I study phosphate, obviously I will be using the term mineralization. And phosphate is critically important for formation and health of mineralized tissues. If we have phosphate insufficiency, our bones and dental tissues are hypomineralized. That makes bones softer. They cannot withstand mechanical forces. They break easily. It causes rickets, which is the problem with development of endochondral bones. Causes undermineralization of teeth, which makes them more prone to dental caries. On the other hand, if we have too much phosphate, that causes excessive formation of bones and dental tissues, but also ectopic mineralization, meaning in the places where it's not supposed to occur. And we already heard today about vascular calcification, but also calcifications in the tendon. So this really illustrates how important the phosphate availability is for the formation of mineralized tissues. And phosphate is not just a structural component of those tissues. It's not just a hydroxyapatite. Phosphate ion is a bioactive molecule. It's a signaling molecule. When in case of osteogenic cells, and I'm using this term as an umbrella term for the cells that produce mineralizing extracellular matrix. So in these cells, high phosphate levels, they induce osteogenic differentiation of progenitors, or once those progenitors differentiate towards osteogenic phenotype, it induces formation of mineralized extracellular matrix, in part by regulating gene expression, but not only. And we know this property of phosphate from our in vitro studies, what Agnieszka mentioned or just talked before. What we do in vitro when we want to study mineralization, we just treat competent cells, mineralization competent cells, with ascorbic acid and a source of phosphate. That's it. That is enough to induce mineralization. However, we have to keep in mind that other than mineralization, phosphate has also basic biological functions, mostly supports proliferation. In many other cells than osteogenic cells, excessive phosphate causes apoptosis, but also in case of vascular smooth muscle cells, causes their transdifferentiation into osteogenic phenotype. So this highlights the role of phosphate as a signaling molecule, which is critical of driving mineralization process. In the local cellular environment, phosphate is generated by 2 enzymes, which Jose Luis already highlighted earlier this morning, tissue non-specific alkaline phosphatase and phospho-1. Both those enzymes use different substrate to generate high levels of phosphate ion within cellular environment, and that action of those enzymes is critically important for mineralization. So our interest from studying role of phosphate in biogenesis of matrix vesicles came from the study of Jose Luis, which he kind of mentioned this morning, but not that particular set of data, when they analyzed the mice with compound deficiency of phospho-1 and TNAP. One of the observations they made is that in mineralized, in dentin, one of the mineralizing tissues, the deficiency of those 2 enzymes results in loss of matrix vesicles in the extracellular matrix. So that observation made us think that phosphate is important for biogenesis of matrix vesicles. And just to quickly remind you that matrix vesicles is a unique population of extracellular vesicles. It's called matrix vesicles because they basically reside in extracellular matrix. That's a basically physical localization
of those extracellular vesicles, which makes them matrix vesicles. But also they have very unique function. They support initiation of the mineralization process, and that function is executed through a unique molecular composition of those vesicles. They are highly in phosphatases that support generation of phosphate, TNAP and phospho-1, annexins, which provide calcium, and also phospholipids, which serve as source of phosphate for intravesical mineralization. Also, they have been defined as vesicles of the size in about 80 to 200 nanometers. So how we analyze matrix vesicles? So this is overview of our experimental approach. We use in vitro approach with osteogenic cells that we grow on the dish. And we use, to analyze vesicles, we use two experimental conditions. Control, which we also call non-treatment. Those cells are grown in standard physiological phosphate levels, which is 0.2 millimolar concentration. Or we grow those cells in osteogenic medium, which is contains 10 millimolar phosphate. Osteogenic cells like high concentration of phosphates for mineralization. And we either add ascorbic acid or no, depending on the assay. So we grow those cells for 24 hours, and then we collect conditioned media or extracellular matrix. And I'm sorry, something showed up here. We isolate vesicles from extracellular matrix by enzymatic digestion, because what Agnieszka already mentioned, they're attached to collagen, so we want to release them from the collagen. And we call the vesicles isolated from the media extracellular vesicles, and those isolated from extracellular matrix vesicles. We use differential ultracentrifugation to purify those vesicles, and we did not do any different cutoffs because we wanted to collect all populations of vesicles which end up either in medium or in matrix. And then for characterization, we use nanoparticles tracking analysis just to size and count them. We did protein analysis using mass spectrometry and gene ontology analysis to get some ideas about the molecular functions of the proteins in those vesicles. So just to start to highlight specific features of our cells that we use to analyze matrix vesicles, we use 17-A11 cell line, which is a committed osteogenic cell line. And by committed osteogenic cell line, I mean those are the cells which are already highly expressing genes which are required, or proteins that are required for osteogenesis. We do not need to differentiate those cells. They are already high in activity of tissue non-specific alkaline phosphatase. They're high in two major transcription factors important for osteogenesis. And once we stimulate them with osteogenic medium, they rapidly, within 6 days, undergo mineralization. And this one is a proper physiologic mineralization with formation of hydroxyapatite. And what's important for our analysis of matrix vesicles, those cells produce actually a lot of matrix vesicles. So even in unstimulated state, they produce around 1,000 of vesicles per cell within 24 hours. So it is a lot. But once we stimulate them with osteogenic medium, they rapidly increase production of matrix vesicles. And you can see that within 12 hours, they already ramp up to around 6,000 vesicles per cell. So this is a really good model to study matrix vesicles, and that's why we stick to those cells. But also what you can see from that graph, analyzing the production of matrix vesicles over time, that those cells are already equipped to produce matrix vesicles, most likely without the changes of gene expression. Most likely they already have those proteins which are required to make matrix vesicles already ready to go and when stimulated with osteogenic medium. So as our first step, we compare how phosphate changes the populations of vesicles produced by those cells, and we use a very simple characterization by size. So what you can see here, that from trying to use the mouse, in matrix vesicles, in the unstimulated state, most of the vesicles are in a smaller size, which are below 80 nanometer. However, once we stimulate those cells with osteogenic medium, they decreased by half contribution of those small vesicles and increase production of vesicles which are the size around 80 to 200 nanometer. This is their size which we predict for matrix vesicles. So already we see from that very crude analysis that stimulation of the osteogenic cells with phosphate specifically increases the population of matrix vesicles, those which are supposed to participate in mineralization. There is different situation in the vesicles isolated from the media. In the media vesicles, phosphate doubles the population of the small vesicles, those in size 40 to 80 nanometers, and reduces the population of vesicles with 100 to 160 nanometer size. So there is differential effect on the populations of vesicles, depending whether we isolate them from medium or from extracellular matrix. And because we assume that each cell produces variety of different type of vesicles, we wanted to see whether the vesicles produced by the cells, they have characteristics of exosomes or matrix vesicles. So here is a very simple, this is a Western blot analysis when we analyze proteins from the matrix vesicles, comparing with the original cells, and we compare to exosomes from the plasma. Didn't work here, but from melanocytes, which produce quite a lot of exosomes. So what you can see here, that HSP70, which is one of the exosomal marker, was absent in the vesicles isolated from our cells, regardless whether there was no treatment or osteogenic medium, although the cells themselves, they do express that protein. Another marker, CD81, was also absent in matrix vesicles. On the other hand, the vesicles were highly positive for annexin 5, tissue non-specific alkaline phosphatase, and phospho-1 enzyme. So already that crude analysis by Western blot of the composition of matrix vesicles shows us that they're most likely different than exosomes. So then we were interested in figuring out which one of the osteogenic factors that we use to stimulate matrix vesicle production actually stimulates matrix vesicle release. And since the osteogenic medium is a very simple medium, it contains only, well, it's in addition to growth medium, contains only ascorbic acid and phosphate, we compared the release of matrix vesicles when we use both of those molecules as opposed to phosphate only and ascorbic acid only. So what we can see from this graph, that ascorbic acid only does not increase production of matrix vesicles. However, phosphate only stimulates cells production of matrix vesicles to the similar level as both factors together. And here is just a nano-size site analysis of the size distribution, showing there's no major change in the shift of the size, and cryo-EM analysis of the vesicle morphology. So what we see from these results is that phosphate is the factor that stimulates the release of matrix vesicle from osteogenic cells. So now, knowing that phosphate is a bioactive molecule that changes the physiology of the cells, we wanted to understand whether phosphate also changes the molecular composition of matrix vesicles, because we know that extracellular vesicles carry the information about the pathophysiological state of a cell of origin. So here is the summary of our proteomic analysis of 4 groups of extracellular vesicles. And again, just a reminder, no treatment means physiologic levels of phosphate, or phosphate treatment means supraphysiologic levels of phosphate, those that stimulate mineralization. So what you can see from that Venn diagram, that there is a high overlap of common proteins detected in all 4 groups of proteins. However, each group also has a good number of unique proteins, with the most common unique proteins in the matrix vesicles stimulated with phosphate. The least unique proteins in media vesicles stimulated with phosphate. When we did the gene ontology analysis of the molecular function of proteins in those 4 groups of vesicles, we really didn't see any differences at the superficial level. All of those vesicles were enriched in proteins with catalytic activity, which indicates the enzymatic processes occurring in those vesicles, or at least information about the ability to carry on the enzymatic activities. And relatively low abundance of proteins with receptor and signal transduction activity, which kind of makes sense because there is not much physiological response we can expect from matrix vesicles, which don't have the gene expression machinery. Now we have a closer look at the changes that phosphate induces in the composition of matrix vesicles specifically, and media vesicles. So within the group of matrix vesicles, there were, with no treated cells versus treated, about 2/3 of the proteins were the same in both groups. However, many of them differ in the level of expression. Almost 40% of the proteins were significantly changed between those 2 groups, and in our analysis, threefold difference was considered significant change. From the proteins that were mostly dysregulated or differentially regulated by phosphate, the biggest category were proteins with catalytic activity. So phosphate mostly changes the metabolic processes that can be regulated within the matrix vesicles. There were also changes in proteins with binding activity, structural molecules, and transporter. It's a different situation within the media extracellular vesicles, where fewer proteins that were common for non-treated versus phosphate-treated cells, only 16% was significantly changed. And with those proteins, most downregulated were with the enzymatic activity and binding activity, while the most upregulated was closely structural molecules, catalytic activity, and binding activity. Now we also wanted to have a little closer look at the unique proteins for each group. And before we analyze those proteins, we removed, out of the total proteins identified in each group, we removed the proteins with the low count, because we assumed that different mixed populations of extracellular vesicles, but we didn't want to include the proteins which were counted only once or twice in the whole group, to get a better understanding of what's the most significantly changed by phosphate. So what you can see from this table, that phosphate mostly changes, again, the protein composition of matrix vesicles. Up to 21% of all proteins identified in that group were unique proteins, not identified in other groups of vesicles. So now going back to the functional analysis of the proteins that were changed by phosphate, those which in matrix vesicles specifically, those which were downregulated by phosphate mostly the most downregulated were the ones associated with exosomes. So if you recall, very first data that I showed, that phosphate treatment decreases the contribution of small vesicles in the matrix, that may reflect basically that there is fewer exosomes in the population of matrix vesicles from cells treated by phosphate. Also, phosphate decreased the contribution of proteins involved in the actin cytoskeletal organization and establishing cell polarity. What phosphate upregulated in the matrix vesicles, and that was a little bit surprising for us, the number one group was cellular response to interleukin. The way we interpret that is one of the activities of phosphate. Phosphate is heavily pro-inflammatory. So if we think that this may reflect the pro-inflammatory function of phosphate. However, the next group of molecules that was changed was heavily associated with the biosynthetic processes. So we think that this corresponds to the function of phosphate of supporting extracellular matrix formation and mineralization. Those proteins might be involved in production of large amount of extracellular matrix proteins and also glycosylation of those proteins. So that's what's different between those four groups of vesicles. What was similar, and that was highly similar, all groups of matrix extracellular vesicles secreted by those cells, regardless whether treated with phosphate or not, were highly positive for tissue non-specific alkaline phosphatase. And in all four groups, that was one of the most abundant proteins. And this is just a confirmation of the proteomic and mass spec analysis with Western blot. So this analysis tells us that tissue non-specific alkaline phosphatase can be considered a marker of matrix or all extracellular vesicles are released by osteogenic cells. And that might be useful in the future when we study, for example, vesicles in the circulation or body fluids. If we want to study the origin of the vesicles, we think that tissue non-specific alkaline phosphatase could be a good marker after verification in vivo. So just to summarize what we found out in our studies, first of all, that extracellular vesicles in the media and those in matrix are very different populations of vesicles. They're all high in enzymes and low on proteins involved in signaling. And they're all high in tissue non-specific alkaline phosphatase, which makes it a marker of extracellular vesicles released by osteogenic cells. We have shown that phosphate is a very potent stimulator of release of matrix vesicles and also changes protein composition of extracellular vesicles with stronger effect on the matrix vesicles than media vesicles. Thank you, and I will be happy to take questions.
Any questions for Dubrava?
Thank you. Really interesting presentation. I had just a curiosity because from your proteomic analysis, so you saw a decrease in proteins that are related to exosome biogenesis and formation in matrix vesicles after phosphate treatment. So you think that maybe that you have more information about other possible biogenic processes, maybe from the proteomic analysis that you have performed?
Yeah, this is a great question. So we are still analyzing those data because that's what we are hoping for, that with the proteomic analysis we will be able to pinpoint the biogenesis pathway. However, we found a little bit of everything. And that's what was disappointing because we don't have a clear answer. So yes, the exosomal markers were down, but we have a little bit of cluttering, a little bit of apoptotic proteins, but not all of them, just few of them, but it's still significant. So we have proteins involved with formation of microvilli. So we don't have a clear answer, but we have to analyze those data more carefully.
Could be interesting, for example, if you block the formation, for example, the exosome of the budding from the plasma membrane, how it affects the release of this matrix vesicle.
I'm sorry, can you repeat that?
There are many inhibitors, for example, to block the exosome biogenesis, or for example, the budding from the plasma membrane. For example, the ones that act on the budding, they work on cytoskeleton to block the budding outside.
We did that, and again, I don't have those data here, but we did those with few inhibitors. Each of them inhibits a little bit, none of them blocks completely. But yes.
Thank you.
Yeah, that's what we're hoping. We're really hoping that with this analysis, we'll say, yeah, this is the way. But unfortunately, we're still kind of like, we don't know.
Thank you.
Thank you.
Is other comments? If I can do a comment too, if I can,
sure.
Say something. I have 2 comments, and then I would like to say something to Stefano. Matrix vesicles are not only the one that are able to mineralize. Matrix vesicles, just extracellular vesicles, are able to bind to the extracellular matrix. People that have been working on matrix vesicle, they have been focusing just on mineralization, but no one ever said that matrix vesicle. So what Dobrava found actually goes directly to that kind of direction. There are subpopulation of matrix vesicle that self-release constitutively just to do other things that are not mineralization. And exactly this, for sure. Why not? Cells can release also. But what I actually will be puzzled, like on your data, is that with inorganic phosphate decrease the bind activity of the vesicle. It should be the opposite.
So those are the. It decreases the. Actually, it's not decreases. It changes the composition of proteins with binding activity.
That's interesting.
So the way I interpret that it basically changes what those vesicles bind to.
And
can bind to. Yes, and I thought that was
the other
actually
thing
interesting.
is the active cytoskeleton, that goes down, but the glycoprotein synthesis goes up. This is basically, is the opposite, because glycoproteins like mucin, we are trying to study it now, are related to the budding, because when increase the density from mushroom to brush, they induce the blebbing, and therefore, the active cytoskeleton goes inside, and depolymerization stimulates the release of the blebs. So the 2 things, they should be on the same direction, not the opposite.
Actually, we are thinking completely differently about it. If you think about the phosphate, it was like, okay, debate. If you go back to the phosphate function, it stimulates the formation of extracellular matrix as well as mineralization, not only matrix vesicles. So one of the interpretations that I have, which still remains to be proof of or disproof, is when we stimulate cells in phosphate, that induces protein synthesis pathway to produce enormous amount of proteins which go into extracellular matrix. Production of extracellular matrix is a very heavy protein producing process. And many of those proteins are highly glycosylated. So another pathway which goes up is glycosylation.
It's not really, not related to the plasma membrane, but what?
No, I think it's to the production of extracellular matrix.
Okay.
Again, I might be wrong.
No. It makes sense. It makes sense.
So thank you very much.
Thank you.
One moment, please. Excuse me. One moment, please. There is another question.
Okay. Faster, because we are 1 hour in late.
Okay, so maybe 1 hour late?
Yes.
I will try to be.
45 minutes.
If I may, I want to ask you, Dobrava, about the concentration of the calcium when you increase the phosphate ions to 10 millimolar.
Calcium stays the same. We don't change calcium.
Okay.
The only thing which we change is the phosphate. That was short. Thank you.
Okay, thank you. I introduce Professor Owen Davis. He will present on matrix vesicle insight to composition prospective therapeutic application. He is an engineering regenerative medicine expert, and he works establish a multidisciplinary research group on the investigating on EVOS in regenerative medicine drug delivery with the primary focus on skeletal application. He is a board member of the board of different several associations that work on AV, and also on the editorial of JEV and tissue engineering journals. Thank you.
Okay, thanks very much. So my name is Owen Davis. I'm from a place called Loughborough University in the UK, which is very difficult to pronounce if you're not from the UK. We can call it LU, or it's about an hour outside of London. So my research group focuses on the application of all types of extracellular vesicles in regenerative medicine and bioengineering principally. And today I'm going to present some of our work on matrix vesicles, some insights into their composition and their prospective therapeutic applications. And some of the things we're finding are slightly different to the previous presentations, so we'll see how we go. So in terms of the need, there's a distinct need, in my opinion, for new regenerative treatments for bone regeneration. So cellular therapies, there was huge promise in the stem cell field. However, MSC therapies have proven quite costly, difficult to scale, and I think we're lacking relevant critical quality attributes related to potency. There are existing medical devices available, such as the infused bone graft, for example. However, they typically rely on supraphysiological doses of a single growth factor, such as BMP2, and have received numerous there should be animations in here, but it's
Gianluca, we have also the PowerPoint presentation, because it's on the PDF.
Yeah, it's.
Okay.
I can go with a PDF, but it might get a bit confusing because there's a lot of information.
I think if you have sent or give to the
I did send.
Okay, so we can.
Sorry to delay you further. Sorry, my fault. I clicked on the wrong one. So there's so we believe that EVs of multiple descriptions could be used as a somewhat intermediate between more complex cell-based therapies and more reductive single growth factor-based approaches to drive a more physiologically relevant response. The one thing to note is that in practically all EV studies, certainly those in preclinical testing and clinical trials, the EVs are derived from a fluid. So that could be a biofluid, such as platelet lysate, for example, or from cell culture media, with perhaps the number one cell source being stem cells. However, as we've heard for most of the day, we've known since the late 1960s that within bone and hypertrophic cartilage that matrix vesicles represent a primary site of early mineralization. I'm referring to them as matrix-bound vesicles in this talk because we'll be comparing them with media vesicles, and the abbreviation for media vesicles I've used is MV, so we're using MBV for matrix-bound. They were identified as early sites of ECM mineralization way back in the late 1960s. They've been observed in a range of mineralizing tissues, and they drive their effects through an enrichment in channeling proteins for calcium and phosphates. We've heard the annexin proteins, for example. They are enriched in phosphate hydrolyzing enzymes, such as tissue non-specific alkaline phosphatase, EMPP1, phosphor1, etc. And they're also enriched in a variety of lipids, such as phosphatidylserine. However, we're interested in using these therapeutically in the future, However, we have some questions related to their biogenesis also. So the majority of studies to date suggest that these matrix vesicles are ectosomal, so they bud from the plasma membrane and are not exosome-like in nature. But we wanted to try to understand this using single particle imaging to try and prove this further or disprove. And we also wanted to, secondly, look at the potential therapeutic applications relative to media vesicles. So our rationale for looking at the biogenesis of these was based on a publication by Irving Shapiro back in 2015, which was, I think, somewhat controversial at the time, where they suggested that perhaps matrix vesicles were in fact anchored exosomes. So what we did was we don't traditionally isolate our extracellular vesicles using ultracentrifugation. We use tangential flow filtration with size exclusion chromatography, but we wanted to keep the process as similar as possible between the two, between the media vesicle isolation, so the MVs, and the matrix vesicles, the MBVs. So the only distinction here is we have a higher centrifugal force for the isolation of MBVs, and we employ a collagenase digestion step also. And these are being isolated from mineralizing bone marrow MSCs over a period of 3 weeks. And what we first found when we conducted Western blotting was slightly surprising to us, was the presence of markers that you may associate with endosomal biogenesis, so ESCOP pathway, for example, such as ALEX, TSG101, and CD63. I think CD9 is perhaps more, the literature suggesting is perhaps less of an exosome or endosomal-like marker, where CD63 seems to be reoccurring as more endosomal. When we performed liquid chromatography mass spectrometry on these matrix-bound vesicles, we were interested to look at their overlap with published databases. So these are open access databases, Exocarta and Vesiclepedia, where you can look at the top 100 proteins. And I think the thing to note here is that these top 100 proteins are not typically from matrix-bound vesicle studies. They're from secreted vesicles. So we were interested to know how many of these proteins were present within our MBV fraction. And we found 60 to 65% depending on the database. So not conclusively ectosomal or endosomal, but showing some overlap with common top 100 proteins. And when we cross-reference these further, so there's a good, it's a little older now, but it still stands, and I think it's a good publication by Guillaume Van Niel's group out of France. And you can see that in the top panel here we have exosome biogenesis, or endosomal, and the bottom we have ectosomal. And I've highlighted our proteins that we identify using by proteomics on the diagram in yellow. And you can see we're neither suggesting that we're isolating a pure ectosomal or endosomal population, but we are showing some considerable overlap with the endosomally derived material. So I know what we're all thinking. You could be isolating a lot of heterogeneous material from the matrix, some of which may be endosomal, some of which may be ectosomal. So what? So what we did next was to look at a single EV expression using super resolution microscopy. So we used the ONI system, and I think there's a talk by ONI later in the day. I don't work for ONI. That's I'm not plugging them. What we did was we looked at the single EV level, and we were interested to look at a common marker of matrix vesicles, so tissue nonspecific alkaline phosphatase, or TNAP. And we wanted to look at co-localization with markers of what we would be indicative of endosomal biogenesis. They're not exclusive to endosomal biogenesis, but they are indicative. And I think there are a number of publications coming out recently by Clotilde Thierry's group and again Guillaume Van Niel's group, which suggest that CD63 is a good marker of endosomal biogenesis. So it's implicated in cholesterol sorting into the endosome, for example. And what we find is approximately 40%, at least it declines over the period of isolation, but at day 7 we find approximately 40% of our MVV population co-expresses TNAP with CD63. We find a few, about 5 to 10% of our MBVs are TNAP only expressing, and we find a significant portion are CD63 only expressing. So there's considerable localization of both CD63 and TNAP at the single particle scale. And we took this a step further, and we decided to, formulated a platform, so immunoprecipitation protocol, where we used immunomagnetic beads, which we conjugated with TNAP, and then we pulled down the TNAP positive fraction, and we proved that this worked using Western blotting. So you can see that in the TNAP positive fraction, we have a lot of TNAP positive MBVs. And we also have the expression of other markers that you might expect, such as Onexin-5. But what was interesting was when we looked at the expression of other proteins after we'd enriched for the TNAP-only fraction, then we found a considerable upregulation, almost exclusive expression of endosomal proteins such as ALEX and TSG101, for example, were completely enriched within the TNAP-positive fraction. So the conclusion from this piece of work was that we can't say for sure, we can't comment for sure on the biogenesis because we would need to track intracellularly, but there's certainly a sharing of markers that you would consider to be endosomally derived within our MBV population. And we are currently progressing a full omics study of these TNAP isolated MBVs also. So I was hoping I'd have that by today, but I don't, unfortunately. So that can be maybe for next year. So the second question that we had was, does source matter? So we have an interest in MBV biogenesis, but fundamentally we're interested in the most effective and efficient population for driving a pro-therapeutic response. So we'd previously shown that you could apply media vesicles from pro-osteogenic MSCs and also from osteoblasts to drive mineralization in model systems, and I don't have time to present that data today. But we were interested in understanding whether we could enhance the pro-therapeutic effects, perhaps by switching to matrix-bound vesicles. So we did exactly the same thing, where we isolated our MVs and our MBVs from mineralizing MSCs, this time over a period of 2 weeks. And what we found was that we could isolate significantly greater yields of MBVs. And this is interesting from a therapeutic perspective, I think, because I think the binding to the matrix is acting as almost a mechanism of enrichment for these particles. So if you're looking for a platform which is scalable, then perhaps releasing these from the matrix is a good way forward to derive high yields relative to media vesicles. We were, interestingly as well, also we found through collaboration with Seda Mabrek, we were looking at incubation of our matrix and media vesicles in a solution of calcium chloride and sodium monophosphate. And when we did this, we found that both could actually produce an hydroxyapatite-like mineral phase. You can see from the transmission electron microscopy images to your right that we were seeing the presence of far more electron-dense particles within our matrix vesicle fraction than our media fraction, but they were there nonetheless. When we subsequently went on to look for proteins that would be representative of mineralization and calcium channeling, for example, we found a considerable upregulation in EMPP1 and TNAP. We also found considerable upregulation in Onexin-2 and Onexin-5 within our matrix-bound vesicles relative to our media vesicles, as you might expect. When we looked at alkaline phosphatase or TNAP content within both vesicle populations, we found up to 13 times greater alkaline phosphatase content within our matrix vesicles. However, when we looked at the activity and we normalized for TNAP between these two vesicle populations, we found that both were active in processing TNAP. When we subsequently looked at again using super resolution microscopy, at the expression of, in this instance, a cocktail of tetraspanins. So this was CD9, CD63, and CD81 in conjunction with TNAP. We found that, again, this was upregulated at the single particle level in our matrix vesicles. However, we still found the presence of these dual-labeled particles within our media vesicles, suggesting that they are there, just at a much lower concentration. And we did the same for TNAP and annexin A2, and again, we found a similar pattern, where we had significantly more within our matrix vesicles. However, they were nonetheless present within our media vesicle population. And this was leading us to think that perhaps, so it may be incorrect, and I'm open to questions afterwards, but our theory was that maybe the extracellular matrix was, these vesicles were being released, and some of them are endosomal. They express TNAP, they express annexin proteins, some of them express CD63 also. They become associated with the matrix, and they become enriched in the matrix. And if you're using relatively low resolution techniques to compare the two, you're missing the expression of pro-mineralization markers perhaps in your media population relative to your MBVs, because they've not been enriched within the matrix. So we wanted to test the matrix binding capacity of these two vesicle populations. And to do this, we seeded our media and matrix vesicles on a collagen type 1 hydrogel, and also on a decellularized, demineralized bovine bone matrix. And we found, you can see from the graphs there, that so you can see from the graphs there that we found considerably more matrix vesicles binding to the matrix. So this is interesting. From a therapeutic perspective, it's interesting because the matrix vesicles retain their ability to bind to a matrix. So if you're thinking of sustained delivery using biomaterials, then this is an important factor. But the other interesting thing is that relatively few, less than 10% of our media vesicles were able to bind. However, if you compare that data with the number of media vesicles which expressed TNAP and annexin proteins, you're also looking at around 10% or fewer. So perhaps it's these vesicles, and we need to do a bit more research here, but perhaps it's these vesicles which are relatively low within the overall media vesicle population that are binding to the matrix and perhaps driving some therapeutic effects. We also looked at this graph here. We also seeded our media and matrix vesicles within a collagen type 1 sponge, and we embedded them subcutaneously in rodent models, and we looked at their release over a period of 30 days. Initially, we were looking for hours or days, and we ended up going on for 30 days, and we had to draw the line there. But we were finding release was sustained over a period of 30 days. There were fewer MBVs released more quickly than media vesicles, so the media vesicles were released more quickly, and the matrix bound were retained within the matrix. So again, suggesting that if you're looking for a sustained delivery mechanism using biomaterials, then vesicle choice comes into play here. And lastly, we conducted a study where we seeded our media vesicles and our matrix vesicles in stem cell cultures. These are 6-well plates containing stem cells, and they were fed either type of vesicle over a period of 3 weeks. And you can see from the images that mineralization was significantly enhanced within our matrix vesicle administered populations. However, again, this may simply because we have more matrix vesicles binding to the matrix and being retained and then driving a mineralization response, whereas we have far fewer within our media population. And what we're next looking to do is to perhaps normalize for TNAP or a mineralization marker within our media vesicles and look to see whether we can standardize this, because it could be a somewhat unfair comparison at present. So, in summary, matrix vesicles or matrix-bound vesicles, our matrix-bound vesicle preps contain a higher proportion of pro-mineralizing vesicles with enhanced ECM binding capacity. We were finding, at least in our study, that these vesicles expressed markers that you would commonly associate with endosomal biogenesis, so they do have some relationship with exosomes, at least in our hands. And my final thought, I feel a bit Jerry Springer saying that, but my final thought would be that these matrix-binding vesicles could represent a subpopulation of mineralization-competent media vesicles that become bound, retained, and enriched within the matrix, and then become subcategorized as matrix vesicles. So I'd just like to thank everyone who did the work from my research group, particularly Evie Angleri, she's finishing her PhD next year, and she's very talented. So if anybody's looking for a postdoc, I'm trying to keep her, but she might want to go somewhere else. And thank you to the funders and the collaborators, and thank you for your attention.
Can I ask a quick question first, because this is something that we are doing, and actually, why, something that really I'm not able to, why in the high resolution, super resolution images, you have TNAP on one side of the vesicle and the tetraspanning on the other side of the vesicle?
Yeah, we don't. It's not like that in every single image.
Oh, okay.
So I just showed a select image, but it's not like that all the time. But I don't know whether there's some polarization in their expression at the single EV, or we don't know yet.
I think that this is very interesting because this is also another contest in flow cytometry. We are not able to find markers that are unique for all, and in combination with our markers, different kind of FOS of different size also can express the marker or contain the marker, but at the same time they don't have the classical marker that are meant to be some that identify FOS. So it's like that. Probably we have a very complex situation in which we have several kind of FOS, and we still have to understand also the function of different subset.
Yep, 100%.
So this is one point that we have to take account during our investigation.
Yep.
And if I can add something, the definition of mineralizing matrix vesicle, they have first to bind to collagen, and then they have to start producing organic phosphate or to accumulate calcium inside. So maybe for a mineralizing matrix vesicle, we must look for two biomarkers together, one of binding to collagen, another one of production of mineral, like TNAP or like phosphol-1. So that the two, if there's only one, like TNAP, not necessarily is because of they have to mineralize. An organic phosphate might be used for other things, of course, mostly for mineralization.
I agree. For us, it was we came at this backwards because we're really not, I don't want to say not interested, but we're fairly pragmatic. We want to isolate the most efficient EV that can drive a pro-mineralizing response, and we don't mind whether it's matrix bound or not. And that was what prompted the investigation. It would be easier for us actually if it wasn't matrix bound, because they're simpler to purify at larger scales.
Correct.
That was very interesting practical application talk. My question is related to what Massimo said and what you commented about what we define as matrix mineralization competent or matrix vesicles. Because if I understood correctly, what you said at the end is that you think that matrix vesicles are those media vesicles which get trapped in matrix. I would think opposite, and that relates to what Massimo said about biological function of those vesicles. They have to drive mineralization of the matrix.
Yeah.
So I was thinking rather, again, I might be wrong, that TNAP positive or mineralization competent vesicles which are in the media are those which escape from the matrix. Because then really it's about the thinking whether they do have matrix binding capacity or not. And that's related to what Massimo said. Again, mineralization process occurs in the extracellular matrix. It's not at random locations in biological fluids. So when we think about getting vesicles for therapeutic purposes, driving mineralization, I think it's important to consider matrix binding capacity as well.
I think the thing that was prompting me to that hypothesis was the fact that we were finding around, slightly fewer than 10% of our media vesicles binding to a matrix when we seed them directly. And we were also, after recovering our matrix vesicles or our media vesicles from a mineralizing culture, finding around 10% or less TNAP positive and exim positive vesicles within that prep. So the way we were looking at it was, let's try and isolate these before they bind to the matrix.
I think it's a great approach, exactly, yeah.
But like I said, might be, I don't know, I might be wrong.
Thank you.
Hello. Could you try to down-regulate annexin 2 expression or overexpress, and to look how it influences on a binding between the vesicles and how it influences on a binding between the vesicle and the matrix? Why I asking? Because annexin 2 only increase the fusion between the membranes when it is double connected with the S100 protein. So maybe why you have more binding for your vesicles from media? Could you check the expression of the annexin when you overexpress or downregulate?
Yeah, we certainly could. It's a good, valuable point. Thank you. We could try.
Okay, I'm sorry, we have to move on to the next speaker. Just 1 hour late. So the last speaker of the session is Maria Letizia Benolazzi. She is an associate professor of biochemistry in the Department of Neuroscience and Rehabilitation at the University of Ferrara. Her research focuses on the molecular mechanism of osteogenic chondrogenic differentiation, developing 3D models with human primary cells and designing nucleic acid-based drugs and innovative delivery systems. So her talk is decellularized Wharton jelly as a source of matrix-bound nanovesicles, new frontiers in the treatment of intervertebral disc degeneration.
So, good morning, and really thank you to the organizer for this opportunity to share our very first step in the world of matrix nanovesicle. And this project, these are the results of a profitable collaboration that span from north to south in Italy, and in particular from Padua, Michela Pozzobon, and Bologna, Istituto Orpedici Rizzoli with Gina Lesignoli, and also University of Foggia with Tommaso Colangelo, and the Professor Roberta Piva Lab that is in Ferrara. That's the research group to which I belong. And this project stems from an epidemiological observation. The 5.5% of the worldwide population is estimated to have asymptomatic disc degeneration. And despite the several therapeutic approaches, the intervertebral disc degeneration cannot be reversed or cured. In this context, in our lab, these are our principal research interests. The study of molecular mechanisms that are on the basis of the modulation of discogenic differentiation, and also the discovery of markers to improve prognosis or diagnosis, and also the degeneration of intervertebral disc as an inflammation, as implication. And so the employment of leaf plant extract to improve the behavior of intervertebral disc cells, and the purinergic signaling, in particular P2X7. And also the last but not least approach in tissue engineering, so stem cell-based therapy. And the project that I'll tell you today that is relative to the use of the cellulite Wharton jelly as a bio-inspired scaffold. Just a premise, something about the intervertebral disc anatomy. The vertebral disc is a fibrocartilaginous joint between two vertebrae. It's an epoxy tissue, and is characterized of an inner part that is really rich of proteoglycan and collagen type 2, the outer part that are organized collagenous fibers, sorry. And there is a big issue because during degeneration there is a loss of extracellular matrix homeostasis, but first of all the discogenic cells lose completely the phenotype, and in particular some of the markers that are in this table. And there is an increase in collagen type 1, the metalloprotease activity. So the aim of our project is the restore of the discogenic phenotype of the resident cells, the recruitment of progenitors, and also the improve of extracellular matrix. Wharton jelly. Wharton jelly is a part of the umbilical cord, is a perinatal tissue. There's no ethical issues in the use of this part of the umbilical cord. And Wharton jelly is noteworthy a source of mesenchymal stromal cells, but also Wharton jelly is very rich of extracellular matrix. And we observe that Wharton jelly contains several structural and also bioactive molecules which are very similar to the SEM of intervertebral disc. So we start with decellularization process with the heterogeneous enzymatic treatment, and we obtain the VJ. On the other hand, we need a good cellular model. And thanks to the collaboration with the University Hospital in Ferrara, in particular Professor De Bonis, we have the possibility to collect human intervertebral disc biopsies. So we proceed with the isolation of cells, and we culture in vitro until passage 2 or 3. And we observe that by immunocytochemistry that during the cellular in vitro expansion there is a decrease in discogenic marker, so this resembling the degenerated phenotype. As first publication in this context, we treat intervertebral disc cells with the VJ, in particular in a 3D culture model, and we observe just after 2 days the increase in SOX2, SOX9, and also TRPS1. So we can observe that the VJ was able to improve the behavior of intervertebral disc. And starting from these results, and thanks to a collaboration with Professor Nastruzzi in Ferrara, we realize hydrogel, so the combination of alginate and gelatin, and also the VJ in a shape of millicylinder. And we try to combine with intervertebral disc cells in vitro, but also we realized such as an ex vivo EVD model in which scaffold, thanks to a transwell system, was combined with intervertebral disc biopsies from patients. And we observed for 3 different patients an increase in discogenic marker, in particular FOXO3A and also SOD superoxidismutase 2, that's a target of FOXO3A, that's a transcription factor, and also TRPS1. We arrive at this point, and so we wonder if the biological effect of the VJ was the result of growth factors or cytokines, or why not vesicles. So we impact the literature, and we impact in particular the world of matrix-bound nanovesicle. And we choose as inspiration the protocol of Professor Badigalak in Pittsburgh, but we realize our own protocol. And so we start with 10, so 100 or 200 milligrams of the VJ, and that were treated with collagenase, but also hyaluronidase, depending on the particular type of this tissue. And after serial centrifugation, we obtain a good amount of particles. So we start with the characterization by NTA, and we observe a medium range diameters of 150. And also we check by electron microscopy, and we observe an intact bilayer membrane, and also we verify the markers, the surface markers, and in this cell we observe CD9, CD63, CD81, but also CD41B and also CD29. We thought that's a memory of the parental cells. And we start with the first series of experiment in macrophages, and there is a rapid uptake of MEVs in this kind of cells, but also there is a reduction in LPS-induced nitric oxide synthase in a dose-dependent manner. In some case at high concentration that's similar to the treatment with dexamethasone. This is very good results. But our goal are EVD cells. So first we try if EVD cells could introduce MBVs, and we observe also this good results after 4 hours after the use of lipophilic dyes as also calcine. But also we observe that after 7 days there are no, the cells were very viable, and also there's no change in the spindle shape morphology, and these are also good results. Go on, and then we know that intervertebrate cells didn't retain high proliferative, are not proliferate so much, and so we try the effect of MEVs in their ability to of wound healing. And we try, and after 48 hours we observe a good results, and also that MVVs could significantly repair wound healing and the scratch that we applied, and so MVVs could induce the migration of EVD cells. And at this point, we would verify if MVVs could alter the phenotype of the cells. And we observe after 7 days actually an increase on SOX9 and TRPS1, but no effect on collagen type 2 and also FOXO3A, but there's good preliminary results. And this is really preliminary results. So we verify by NGS the microRNA content of this vesicle, and among hundreds of microRNA that was analyzed, we observed 7 different microRNA that are common for this 3 preparation. And by Mirtar base, we observed, we check also the target, only target with strong evidences, but the target of some of these microRNA. And also we observe NANX2 and also collagen type 1 and also metalloprotease, so we can potential sentence that MVVs contain microRNA that has potential antifibrotic and also pro-discogenic properties. So as a conclusion, we have many work in progress and open questions. The first is MEV's protein cargo, so growth factors, cytokines, we have to study this, and also the biologic effect. So this is sort of chicken and egg paradox, MVVs or a pool of secreted protein from our digestion. And also the analysis of MEVs corona, but we don't know if the treatment with collagenase and also hyaluronidase can disturb this corona. We think so, but maybe. And also intervertebral disc is physiologically an hypoxic tissue. So we would like to verify the effect of MEVs and also MEVs production in an hypoxia, and in particular we have the possibility to use this instrument in our lab that can provide this condition. So this is us, this is our lab, and we are very, very open to any collaborations. Please come to Ferrari, there's many things to see and also good food. Okay?
Maria Letizia, thank you very much. Quick questions?
I have a comment. Just to add the other aspect that we have to follow during the investigations, also the condition, like for example hypoxia. So probably in under different stimulation, after different microenvironment condition, we can probably drive different production of AVS. So this is another aspect that is very interesting.
Yes, in our project we had also the possibility, it's not this for this talk, to check the EV's production from intervertebral disc. So this result also on MBVs for microherniata argo. We have the same result for intervertebral disc cells, and also the content of microRNA in EVs and also in cells. So next step is the same analysis in hypoxia.
Thank you. So we have, if it's okay, we can go on to discuss in the during the post-test session, the lunch. And we go on with the section of the sponsor.
Okay. So we start with the, actually session 3 is from our sponsors. So the first sponsor is ONI. So Alison Fuji will tell us something about the nanoimager characterization of extracellular vesicle using the ONI nanoimager super resolution microscope. Thanks, Alison.
All right, so welcome. My name's Allison. I'm a field applications scientist representing ONI, visiting from San Diego, California. And what we're going to be talking about today is characterizing your extracellular vesicles using the ONI nanoimager super resolution microscope, and that's this guy here spinning around. So a quick background about how the nanoimager can help your EV characterization is that ONI has accessible technologies in terms of easy to use and easy to set up hardware. We have a collaborative platform in terms of usable cloud-based software that allows you to perform your analysis and share your data with your colleagues. And we also offer bioware, such as the EV Profiler Assay Kit, which is an end-to-end solution that's going to allow you to have an optimized protocol for imaging your individual EVs. So I'm going to skip most of our introduction to EVs because I'm sure this crowd is very familiar with them, but essentially they are these small particles that contain different molecules such as DNA, RNA metabolites, and lipids. They can be used for diagnostics and therapeutics, and for this reason, they are thought of as a budding field worth investigating in the biological and medical sciences. So, single particle EV characterization is a challenge, and that is because EVs are really small. So there's distinct different biogenesis pathways to produce EVs. They're not all created equal. You get a very heterogeneous population out, even from a clonal cell line. They have diverse molecular contents, and then when uptaken by recipient cells, there is a diverse fate. So they might end up in different organelles or having different functions, depending on the EV and depending on the recipient cell. So the challenge to look at individual EVs is we need more resolution. This image here is EVs which have been stained, and then we're watching them being uptaken into a cell. But what we can see here is that while they're stained, they just look very blurry and pixelated. And that's because the EVs themselves are too small to get high resolution when you're doing standard microscopy. So let's talk about how to get that more resolution. When we're looking at this scale of biological structures, going all the way from small to larger, such as multicellular structures like small organisms or organs or multicellular structures, conventional microscopy like standard fluorescent microscopy, phase contrast, and confocal are going to work very well for your samples. But once you get into the subcellular size range, such as organelles, smaller cells like bacteria, or even smaller particles like EVs and viruses, you no longer have the ability to see those really small structures with high resolution unless you get into single molecule localization microscopy, which is what the ONI nanoimager allows you to do. And the reason for that is because there is something called the diffraction limit of light, which essentially means that there's no physical way for you to see structures that are smaller than about 200 nanometers. So how can we achieve more resolution? Let's take a quick look at what we can do to start with. So on the left, we're looking at a diagram of an extracellular vesicle. We have a nice lipid bilayer. We have some proteins and we have some nucleic acids. We can actually label all of these and look at it with a fluorescent microscope. However, because of that diffraction limit of light, we end up seeing a structure that looks blurry, something like this. So while these structures are labeled, you don't get that detail out. However, when you use a super resolution technique, such as D-Storm, you can actually get that very small structure resolved and take a picture of it where you can see those details coming to life. So how do we break the diffraction limit? Let's start here looking at one single fluorescent molecule. This fluorophore is only approximately 1 nanometer in size, and let's look at it now with a fluorescent microscope. What we are going to see is not that molecule itself, but rather a large glowing signal called a point spread function. And this is going to be much larger than the molecule itself, but we can determine the spatial position of this molecule by fitting a Gaussian curve to this point spread function and selecting that very center of the signal, and that happens to also be the point of highest intensity. So it's actually pretty easy to locate where this molecule is, but it becomes more problematic when we have multiple fluorophores glowing in close proximity to one another. So what we can do then is use a single molecule localization technique like D-Storm to turn our fluorophores off and allow them to blink back on just one at a time. So whenever we have one individual signal by itself, we can localize it. So we will put a localization down right there at the center. At another point in time, another fluorophore will blink and we can localize that one. And at another point in time, another fluorophore can blink and we can localize that one. And then over the course of a few minutes, which is a few thousand frames, we can stitch together our high resolution image made up of these individual localizations. So what does an SMLM experiment look like? SMLM meaning single molecule localization microscopy. It's going to take us from our standard microscopy image on the left to our high resolution SMLM image on the right. And these are our EVs stained for CD63, CD9, and CD81, looking blurry, though you can see the fluorescence appear there. And during the actual acquisition, what we see is this field of blinks, where these individual fluorophores are stochastically blinking off and on. And then our final stitched together image over here shows our EVs, where you get a better feel for what's a true EV, what might be background noise, and really be able to define individual molecule or individual vesicle by individual vesicle. So let's get into how you can use the nano imager to help you on your journey to visualize your single particle EVs. And that will be using our full solution workflow EV profiler kit. So everything will be included, that all the reagents needed for sample preparation. And then you will acquire your sample images using the nano imager, but doing an automatic function. And then the analysis can also be performed automatically. So your sample preparation includes validated reagents and protocol. Your acquisition has automated image acquisition with real-time quality checks, so you can watch while your data is gathering whether or not you have good images or whether you might need to change something, so you don't waste too much time. And our analysis has robust built-in analysis that will auto-complete and spit you out a report at the end of your experiment. So here's an example of the interface of the auto EV acquisition. So the field that we're seeing here is actually a zoomed out image of all of your fixed captured EVs on the surface. And on the right, you're seeing the live acquisition. So all those blinking dots are the individual fluorophores on your EVs being localized. So your result is going to be something like this. This is your raw data. And let's zoom in on our EVs. And now we can see multiple different EVs. The software will go through and cluster these. So it's going to determine which signals it wants to group as part of an individual EV and which signals it wants to exclude as background noise. So each individual EV will be analyzed and you'll get parameters for every single one. So if you compare the EV on the left to the EV on the right, you can see they have a slightly different area in terms of size. They have a different length, and they have a slightly different shape in terms of their circularity. And then you can see also the biomarker abundance difference between these two. So the one on the left has a higher abundance of CD9, while the one on the right has a higher abundance of CD63 and CD81. So this is important because when we look at one single population of EVs, they can be very, very heterogeneous. You have large ones, you have small ones, you have ones that are triple positive for all of the markers. You have ones that are single positive and double positive, and different clustering patterns of your proteins as well. So here is another example of the analysis that you will get out when you perform the auto EV acquisition. So your EVs are some They're presented in a mosaic here where you can scroll through and look at representative images of your different subspecies of EVs in your population. And you also get this summary page. And let's zoom in on that summary page. What you'll get is a result for how many EVs you have. You'll also get this biomarker profile. So, for example, this salmon colored bar here represents the triple positive subpopulation for all your EVs. So that's represented 41% in this case. And then the ones in the middle are double positive for 2 out of 3 of your markers, and the ones on top are just single positive for only 1 of your markers. And this is 3-color analysis, so in this case it was CD81, CD63, and CD9, although you can choose to change which markers you're probing for. You'll also get a size analysis, so this histogram here will show the total population size distribution. And each individual subpopulation, you have a box and whisker plot showing the spread of sizes and the average size of the population inside each subpopulation. So the single positives, the double positives, and the triple positive. So that was looking at the EV Profiler Kit, which are fixed, and looking at individual EV characterization. However, the life cycle of EVs is a bit more complex than just looking at them outside of cellular contexts. So we may want to look at more than just the EVs by themselves, but we may also want to investigate biogenesis and cell delivery. So the advantages of D-Storm, which was the technique we were just looking at previously. So it's really great for characterizing molecular signals and understanding the heterogeneity of your EV population. But there are limitations here. So this does not provide insight into dynamic EV behavior and cell EV behavior where they come together. So D-Storm's a fixed technique, but you may want to use a live technique. So let's look a little bit into that. Here's an example of real-time monitoring of EV release. So in this case, CD63 as an endosomal marker, there's a construct here of CD63 that was tagged with fluorine, which is a pH-sensitive fluorophore. So we can actually follow the movement of these vesicles from the multivesicular body inside of the cell, and where you see these bursts of fluorescence is actually when the exosomes are being released to the outside of the cell where they're encountering the higher pH as opposed to in that endocytic compartment. So you can look at this chymograph here on the right, which is going to show the movement of these individual particles and that moment of EV release into the extracellular fluid. You can do longer time lapses. So if you're interested in uptake of EVs, here's an example. We have our lysosomes stained in red here. And throughout this 60-minute time lapse, you can see the time ticking up here. We're watching our GFP positive EVs accumulating within the lysosomes of these cells. So we can see with the merge, we're getting more yellow and more green towards that end of that 60-minute time lapse. We can also follow the movement of particles. So in this case, we're visualizing vesicle biogenesis in real time. We're tracking the particle transport and the pathway that it makes within the cells. And this is using the integrated tracking software that comes with the ONI Nano Imager. And you can track this in terms of time and also speed. So that's what we're looking at here when we're looking at this final pathway merged from all of these individual particles moving over time. All right, so the ONI Nano Imager makes super resolution easy for you. If you look on the right, this is the actual benchtop setup. It's easy to use because it doesn't have a whole lot of extra cables and lasers to connect and wires and things like that. So you can get started on it quickly, and you can use it on a standard laboratory benchtop. Here's some examples of some institutes that are using our technology. And yes, where will your curiosity take you next? Any questions?
Thank you, Alison. Any question for her? I have a comment. When I was in San Diego, Tony told me that you are preparing a nanofluidic system, and that I can give you. We're discussing with the CEO in San Diego that, for instance, this kind of machine, one potential target and application could be to see in real time the formation of the protein corona on extracellular vesicles. So as soon as they will be ready with this kind of machine, if you introduce like a single protein with a single fragment, right, you can directly see how they, the distribution of protein corona is something even nanomedicine was never investigated. So in nanomedicine, people have a lot of questions still open up about protein corona, and these questions have been transferred to the world of extracellular vesicle. So the same things that we don't know about protein corona of synthetic particles, the same. So this is just great. Any comments and questions? Hey, so thank you very much, Alison, for your time. We appreciate it a lot.
Thank you, everyone.
Okay, next presentation is from Beckman Coulter, is Tommaso Rondelli. And we'll speak about the application in flow cytometry.
Good morning to everybody. My name is Tommaso Rondelli. I'm part of application team for South Europe for the flow cytometry part for Beckman Coulter. And today I'll talk you about our solution for EV research. As you can see from this slide, we are, as Beckman Coulter, on this market since the 70s, when the instruments were very big, with a few lasers, with a few colors. And now we have very small instruments with multiple lasers, multiple colors, with a great technology inside. And what I would like to highlight about this slide is around 2010, the Beckman Coulter was owned by Danaher, and Danaher started to invest a lot on flow cytometry. For example, acquiring the technology behind the Cytoflex, which is the technology behind all our system today. I put this slide just because I know that not all of you are familiar with the flow cytometry, so just for this reason. What is flow cytometry is a technique used to analyze physical characteristics of the cells and antigen expressions. Basically, briefly, we have a laser or multiple lasers that will hit the cells or the particles, in your case. The cells are put in a row in a laminar flow, and fluorescence is generated, and the fluorescence will pass through filters and then through detectors. At the end, we have a signal, an electrical signal, and a digital converter. And at the end, we get graphs like those dot plots. Basically, a flow cytometer is formed by three parts: a fluidic part for sample delivery, an optic part for the excitation and then the emission collection, and an electronic part for digitizing the signal. What about flow cytometry and EVs? Okay, as you can see in this slide, the publications are increasing, but today flow cytometry is still not the first choice for EV research. Why? Because it's challenging, at least until now, it's challenging. Okay. The first reason is the limit of detection. As you well know, the particles, the EVs are small, and the traditional flow cytometry can detect from 100 nanometers, the better flow cytometer, to 15, 20, 50 microns, so cells. So in small EVs, the antigen density is reduced. Then the fluorescence is low, so you need a sensitive instrument to detect it. Are there any standards? Yes, there are. For example, there is the MI flow cyt EV framework. This is a framework for to standardize the flow cytometry experiments in terms of sample stainings, in terms of detection, in terms of how to measure, to design an experiment, and how to report an EV flow cytometry data. As Beckman Coulter, we provide, we are a well-established company that provide flow cytometry solutions in terms of instrument, in terms of kits, antibodies, in terms of lysis reagent, in terms of quality control, everything. Our first solution for EVs has been the Cytoflex, which is represented in the left side of this slide. The Cytoflex was launched in 2015. It was the first instrument based on the APD technology. It's a very sensitive instrument. And why is good at EV studies? One, because it's very sensitive. And two, because there is a third size scatter, okay? In addition of the classical forward scatter, the classical size scatter on blue laser, there is a violet size scatter. And the violet size scatter is fundamental to resolve small particles from the background. As you can see in this slide here, we have a mix of bits. On the left, these bits are analyzed with the classical forward scatter and the blue size scatter. And as you can see, the smallest one, the 80 nanometer bits, are not resolved from the background. The 100 nanometer bits are not resolved from the 80. The 200 are not resolved from the 100. Okay. On the contrary, if you use the violet size scatter, you can easily resolve the 80 nanometers bits from the background. You can resolve the 100 nanometer bits from the 80s, and so on. Okay, you have a great resolution. But now we moved a step forward with this instrument, the Cytoflex Nano, which was launched in 2024 at the beginning of this year. The question we try to answer is this: can flow cytometry be further adapted for EV research? Yes. The answer is yes. How? By building an instrument completely dedicated to this topic. So with an optic system, a fluidic system, an electronic system, a software completely built and designed for this aim. This is our new instrument, the Cytoflex Nano. Is the first flow cytometry that can detect particles at least as small as 40 nanometers polystyrene bits. It's a very consistent instrument in terms of performance and data quality. It's flexible to design the EV experiments, and it's built on the same principle of the Cytoflex platform. So if you are familiar with the Cytoflex, you are basically familiar with this instrument because the software is basically the same. Some technical characteristic, the instrument has 4 lasers, the blue one, the yellow, violet and red. It has 5 side scatter, 2 5 scatter on the violet laser, 1 on the blue, 1 on the yellow, 1 on the red, and it can detect 6 fluorescence parameters. This instrument allows you to detect populations that are undetectable with traditional flow. Okay, this is an example. Here we have a mixture called on the left called the nanovis. Okay, it's a mixture of bits from 44 nanometers to 1000 nanometers. This instrument has 2 violet side scatter. One is more sensitive, which is the violet side scatter 1. By using this scatter, you can resolve 44 nanometer bits from the background, and you can clearly resolve even the 80 and the 100 from each other. Okay. With the second violet side scatter, you can resolve the higher bits. Okay. And here on the right, we have another example with silica bits. And we have a mixture of bits from 50 nanometers to 100, okay? And we have bits of 50, 55, 60, and as you can see, we can clearly resolve bits, the 50 from the 55, the 55 from the 60, which is very impressive. As I said before, the instrument has more than the side scatter, 5 side scatters, and more side scatters means more performance. This is an example. Here we have commercially EVs, okay, without any staining. As you can see, if we try to analyze these particles with just the violet side scatter 1 and violet side scatter 2, okay, we cannot resolve anything, okay. But if you use the violet side scatter with the red, or with the yellow, or with the blue, you can resolve the population, okay. And it's clearly seen with the histograms, okay. You can clearly see the peak of this population, and it's clearly visible. This is another example. Here we have GFP commercially available exosome. You can resolve them from the background here with the blue laser, okay. And then we try to stain these particles with 3 different CD81, 3 different colors with 3 different lasers. And as you can see, you can clearly resolve them from the background. Okay. Keep in mind that with the traditional flow cytometry, the characterization would be incomplete, as only exosome down to 100 nanometer can be detected. And with this instrument, you can fully characterize the exosome. And the workflow contains new checks to ensure the instrument consistency and data reliability. We have 4 new automated processes, which are a sensitivity monitor to monitor the sensitivity and the resolution. We have a volume monitor to monitor the volume left in tube. We have a baseline monitor to monitor the sample line debris and clean if needed. And then we have an onboard enhanced clean, an automatic sample line cleaning using a buffer inside. Last but not least, this instrument works paired with a third software called FCM Plus. This software can convert the size scatter to diameter. So at the end, you can get information regarding the dimension of your particle directly in the graphs. So in summary, our solution for EVs are 2. The first is the Cytoflex. With the Cytoflex, you can analyze EVs from 100 to 1 micron, 100 nanometer to 1 micron, and you can analyze cells. It's a traditional flow cytometry. And on the left, you can see the Cytoflex Nano, a flow cytometer built just for EVs, with a limit of detection at least as low as 40 nanometers. And this is my last slide about the Flex family. On the left, you can see the Cytoflex, the first one launched in 2015. Then in 2020, we launched the DXFlex for clinical. In 2023, the Cytoflex SRT, a sorter, a benchtop sorter. And now we launched in 2024 the Cytoflex Nano. All of these instruments shares the same technology based on APD detectors. Thanks for the attention. If you have any question, I'm here and later at the booth as you want.
Thank you, Tommaso. There are questions for the audience? Professor Lanuti?
Hi, thank you very much. My question today is always the same. Did you measure the detection limit with beads, right?
Yes, the beads.
Have you ever tried with exosomes or Rosetta beads or some other?
And not me, but I can ask to my colleague that directly follow this launch of the instrument. I can ask him, and if you want, I can tell you his answer.
Thank you.
You're welcome.
Okay, we can go on and take the opportunity during the lunch process section to make more questions.
Please, stop by
the
the booth
sponsor.
of the sponsors. So we had just 1 hour late. Let's move on with the next. So Mayra will present eyes on iron, scalable EV isolation, single particle characterization from ISON. And so Mayra is presenting today. Thanks, Mayra, for being with us.
Okay. Okay, so good morning to everyone. I'm Mayra. Stefan has the flu, so he's sorry. Today I walk you through the ISON technology for the isolation and the characterization of extracellular vesicles. So the whole idea of ISON is to give the possibility to isolate extracellular vesicles from any kind of sample, so plasma, cell culture, urine, milk, and so on, and then to characterize the extracellular vesicles and maybe to concentrate them for future studies. Starting from the purification, ISON uses the side exclusion chromatography. Okay. And this technique separates the extracellular vesicles from proteins or dyes or lipid nanoparticles from liposomes. And we have 3 different resins, so the 20 nanometers, which maximize the particle recovery, but with a little bit of contamination by proteins. 70 nanometers, which maximize the purity of the final sample, and 35 nanometers just in the middle. This is the elution graph, so we can see the purity of our eluted sample. Just a second. Okay, and the protein contamination. So, as you can see, the 20 nanometers, so the light violet, has a little bit of contamination by proteins thanks to the size. While the 70 nanometers has the maximum purity thanks to the exclusion of protein-like size particles. But we have the minimum recovery. 35 nanometers, as I told you before, the blue one is in the middle. So comparing this technique to other purification technique, we have a significantly difference in the purity of our final sample, and we don't have a very different quantity of final product. We have different sizes of columns based on your needs. So we start from 150 microliters to 100 ml, but we can go on to maybe 50 liters and so on. So the idea, the protocol is different from the cell culture media and for the plasma. In the first step, with the cell culture media, we have to do a centrifuge to remove the cell debris and dead cells. And then we can use the columns from ISON to collect a purified sample. With the plasma is a little bit different because there is just a centrifuge, and then we can use the columns. Especially in the case of cell culture media, we can start from a very huge volume of liquid, and then we can go, thanks to centrifuge and so on, to a smaller volume of the column. We can do the isolation manually, or we can use the same column and the automatic fraction collector to make it automatic. And this improves the reproducibility thanks to standard protocols, but at the same time the process can be easily customized. So this is the automatic fraction collector. It turns on the color of the column resin. So in this case it will be turns on with fuchsia because the color resin is 70 nanometers. Then the rotational carousel automatically advances once the selected volume has been collected. And from the screen we can use the standard parameters, or we can change it, like the number of fraction, the default buffer volume, or the volume for each fraction. And by avoiding manual pooling, we can decrease the risk of contamination and also of errors. Going on to the quantification, ISON uses the tunable resistive pulse sensing. So we have a nanopore, this one. It is like a funnel. So each particle, one by one, passes through this pore thanks to a pressure and a voltage applied. And in this case, we can obtain this graph. So each spike, the size of each spike is the size of the particle. The frequency of the spikes is the concentration of our samples. And then the speed to return to the basal current is the zeta potential. So this is the XOI, so we put our sample in the upper chamber. The sample needs to be diluted in an electrolyte, and then we apply a pressure and a voltage, and the current starts. So every particle, one by one, passes through the nanopore and creates an interruption of the current. And from this interruption, we can obtain data on the size, the concentration, and the zeta potential. So the range of particles analyzable is between 50 nanometers to 11 micrometers. And in the end, we have the histogram for size and concentration, or for size and zeta potential, and an Excel with all the characteristics for each particles. We have different pore size based to your size samples. And this is the software, so we can see the basal current here and the spikes underlined by fuchsia dots. So the sample is analyzed at 3 different pressures to have the most accurate analysis. This is a paper published in 2021 where they're wondering which isolation technique is the best. So here we have a comparison. We can see that a DLS technique like the NTA and nano-FCM both missed one population, while MALS is unable to distinguish individual population. TRPS, our technique, is clearly and accurately identified and size all 4 populations. Another important thing is the reproducibility. So while the nano-FCM does just 1 repeat, the NTA and the TRPS does 3 repeated runs. The percentage of error in an NTA is around 20%, while with the TRPS we have around 1%. This is another paper published in 2017 about the zeta potential. And in this case, the pulse technique is unable to distinguish different populations, while the TRPS, another time we can say that it can distinguish 2 different populations. And at the end, we obtain an histogram with different population. In this case, for example, we have the empty lipids nanoparticles with lower negative charge than the mRNA-loaded nanoparticles. Just a quick explanation about the concentration kits. So both of them target specific proteins on the particle surface, but the first one needs a centrifuge because it's with PEG, while the second one is with magnetic beads. So please contact us, don't be shy. We have a booth outside so we can speak easily, maybe in Italian. Outside. Thank you.
Any questions? If you agree, I would like to move to the last because basically we are completely invading the time of the lunch. So is it also your interest?
Massimo, I don't know if you're more worried for the lunch, but we have the days we stay together. Don't worry, we will. Okay. NTA, More Than Size and Concentration by Ingrid Bloss for NTA.
Hello, my name is Ingrid from Particle Metrics, and I want to show you a little bit about our NTA system. So first of all, is this working? Not, okay. Okay. If you look at the papers that are published that are using NTA as a method, a lot of them are focusing on size and concentration. So this is a tried and trusted machine to use for size and concentration analysis of EV samples. However, NTA can also do a lot more, especially our zeta view systems can also measure zeta potential, can identify subpopulations. Of course, you then can do fluorescent measurements, and with our latest models, you can also do colocalization experiments. So how does it work? You have 1 up to 4 lasers coming from the top, illuminating the sample, which is in a cell. And at right angles, you have a video camera with a magnification, and it is taking a video of the sample. And the camera and the laser are automatically focused and are migrating through the cell and taking measurements at 11 positions. So the handling is straightforward and very quick. You inject your sample, you see straight away on the screen your sample, and then you can perform your measurement, and the whole thing takes about, let's say, including injection and afterwards cleaning, maybe 5 minutes per measurement. So it's really quick and straightforward. So why are we doing this 11 position measurement is because we want better statistics. If you look at only 1 position, you will maybe analyze something like 30 particles, which is not really giving you a good representation of your results of your sample, because you don't know if this is the real size distribution. However, if we add more positions here, we get a much better resolution. We get much more particles that we analyze, and we can better trust this histogram. If you look at position 9 here, you see a big white thing on the screen. The system has an automated outlier control, so it has some statistical algorithms which will compare the results of the individual positions against each other, and in that case it will suggest to you that you maybe discard position number 9 because there's something that is different from the rest. However, we leave the decision to you. We can just suggest that there is something different, and you can look at the results, and you as scientists can decide on your own if you want to include or exclude these positions. The concentration is measured by particle counting, so we know the size of our field of view, and we also know the focus depth of the camera and the laser, so we can simply count the particles, and this works at a concentration from 10 to the power of 6 up to 10 to the power of 9 particles per milliliter. It's a linear relationship between the concentration that we measure and the concentration that is actually in your sample. It would also work with a smaller concentration of 10 to the power of 5. However, then you have very few particles to analyze. So, but just in case you have a sample which has not a high concentration, you can also go for it. The size is measured by analyzing the Brownian motion. Okay, the video is not working? No. Okay. Then you come outside and look at our booth. You can see the Brownian motion of the particles. They are sort of jumping around a little bit, and the smaller the particles are, the quicker this Brownian motion is. And the system is detecting this by following a particle over a consecutive series of video images and taking the so-called mean square displacement. And with the help of this, you can use the Stokes-Einstein equation, where the radius, the particle radius, is then the only unknown feature, and you can calculate the hydrodynamic diameter of the particles. If you inject your sample, you see a lot of particles, and the first thing would be, okay, these are my EVs. On the other hand, in scatter mode, you cannot really distinguish what it is. It could be basically anything. It could be a liposome, it could be a salt precipitate, or it could be an EV. But you will not know with just analyzing size and concentration. So the next logical step would be to go for fluorescence marking. In fluorescence, if you have particles with a biomarker, you will slide a fluorescent filter in front of the camera, and you will then be able to distinguish them between the rest of your particles. So you can calculate the ratio between the total number of particles and the particles that are bearing a biomarker. Here I have an example from practice. This is a membrane staining of an EV sample, and in this case you can see that only 85% of these particles, that's the orange curve, are bearing the fluorescent marker or are having a membrane in this case. And the blue curve is the scatter, so you see that you have approximately 15% of stuff in your sample which doesn't even have a membrane. And now still you know, okay, I have membranous stuff in my sample, but I still don't know if it's really an EV. And if I want to go one step further, the next thing would be to go for antibody stainings. In this example, our instrument with 4 lasers has been used, and we did 4 stainings with different antibodies in order to be able to see how are these antibodies present on my sample. And you can see that the yield is much lower with antibodies because you have a lot of stuff in scatter, and depending on the origin of your EVs also, you probably know that the presence of antibodies is also not always the same. So in this case we have a lot of CD41 with this sample, and we almost have no CD9 in the sample, but you can now distinguish and you can see that probably only the particles that are bearing these antibody markers are EVs, and the rest is something else. When it comes to fluorescence marking, a lot of fluorescent dyes bleach very quickly. So if you look at this orange curve here, you will see that over maybe 5 seconds, it's losing almost half of its fluorescing power. So it's really important if you work in fluorescence that you are using a system which is really quick. And with the help of our anti-bleach technology, we can reduce the accumulation time to about half a second. So if you compare with the standard NTA, which needs 20 up to 30 seconds to do a measurement, you see that you will lose more than half of this power here. And with our anti-bleach technology, we are much better here. So if you look at the green curve in this, on the left-hand side of this graph, you will see that with our anti-bleach technology, we are able to gather almost all of the fluorescence. Our new system is also equipped with a colocalization functionality. So if you are doing a double staining of EVs, for example, then you have a certain amount of single stain particles and you have a certain amount of double stain particles. And of course, it's interesting to you to see how many % are bearing both markers. So you want to know the double positives of your example, and this is possible with our fluorescence system. With our colocalization system, sorry. In this example, this is an example from our lab where we worked with commercially available double-stained and single-stained silica beads in order to prove if the system is behaving in a linear way. And what we did is we made different mixtures. We started with only single stained up to 100% double stained particles. And we can see that, the relationship is almost linear even in the very high double stained samples. And the colocalization works for all laser combinations that we have. The one we are selling the most is 488 and 640 nanometers, which makes a lot of sense. The 488 is a classical for scatter measurement, and you have a lot of distance to the 640, so you will probably not get any crosstalk between the two wavelengths. And also for these two wavelengths, you have a lot of dyes at your to choose from. But now you have seen an example with beads, but what you are really interested in is how is it working with my samples. And so I'm going to show you one example from practice where we did a triple staining from MSC-derived exosomes. These have been created with PEG precipitation, which is not a very pure or not a very good way of purifying the sample, but for the experiment that they were needed for, it was okay because it's quick and easy to do. We made a triple staining, 2 antibodies. One was CD9 with Pacific Blue. The other one was with Alexa Fluor 488 CD41. And the third one is a membrane staining with cell mask Deep Red. And if you look at the graph here, you see a lot of stuff in scatter mode again. And only 30% here are bearing the cell mask. So this is due to the PEG purification method, because you have a lot of other stuff still in your sample, so it's not a very super pure sample here. But still, you have around between 12 and 13% of both antibodies here in this staining. And what we did is we made several stainings where we varied the sequence. We threw in first the antibodies and then the membrane staining, or we threw it in all at once. So we made different combinations to see if the staining sequence has an influence on the output, on the result of your staining. And we found that the colocalization ratios do not really change in this case a lot. So in this case, the staining sequence has no influence. However, this is only valid for these special dyes. So you might have different kinetics with other dyes, and you would have to try this out in your special case. What you also get as a result is this colocalization plot that you see here. So you get red or green dots for all the single positive particles, and you get these blue axes for the double stain or for the double positive particles. But what's really interesting to you, of course, is the percentage of colocalization. We then did a lysis control with NP40, and we would expect, of course, that no more fluorescence is there when all the EVs are destroyed. And this was also the case. You can see in this red line, which is on the 0 of the x-axis, that we have no more fluorescence. The peak that you still get in scatter mode here is due to the PEC. Yeah, this is, again, it's not the best way to purify EVs. Another control we did was double stainings. So if we do a staining on Pacific Blue and Alexa Fluor 488, these are the antibody stainings, and we omit the cell mask, then we would not expect any colocalization ratios in the two channels which contain the red laser. And this is also exactly what happened. You can see here you got blue axis on the 405, 488 colocalization channel, but nothing on the other two. And of course, we also did it with the other combinations, and we can see that this is also working for the other combinations. We get one false positive here on the 405, 488, but basically the controls in this case worked. So to summarize, our instrument, especially with our new software suite, is even faster than it was before. So you get around 2,000 particles analyzed in, yeah, it says as little as 30 seconds. As I said, together with cleaning and injecting and calling up the SOP, it's about 5 minutes per measurement. You get the new intuitive CETA navigator software, which has a lot of advantages over the old model, and especially the colocalization module, which is new and which only works also with the CETA navigator software. And a quick overview about the different models we have. We have 4 mono models, so you can have a violet, a blue, a green, or a red laser here. The twin models, all combinations of these 4 lasers are possible. And with a quad, you get all 4 lasers, and all instruments can be optionally equipped with CETA potential functionality and can be optionally equipped with colocalization functionality. And in each case, we work with a filter wheel which has up to 11 positions for filters, so you could also have customized filters if you want to go for special staining experiments. You can always upgrade from a mono to a twin or to a quad, and you can upgrade from a no colocalization to a colocalization module, or you can at a later date also upgrade to CETA potential. So it's quite flexible. You can start out with the smallest version, and yeah, depending on your budget, can upgrade at a later date if you want to. Yeah, so that's all about me. Yeah, one thing is we also do not use any consumables, so you don't need any chips or kits, which are expensive. The only thing you use is some syringes for rinsing and for injecting your sample, and that's it. And the other stuff you already have in your lab. So thank you very much for your attention. We have the instrument outside. It's running, so we can do you a live demo if you want to. Just come in the lunch break, and if you have any questions, let's do it outside.
Thank you very much, Ingrid. Remember that they will be part of the school tomorrow. So couple of information. First of all, we will start again at 2:30, not at 2 o'clock, because basically we need to digest a little bit. And then, one thing, we have to vote for the posters. So what we thought is we will give you stickers. So these stickers have different colors. The dominating is the smiley or no smiley. That means that please, while you are eating, stop by the posters and stick on them, okay? So I'll see you later. So thank you very much, and I see you in an hour. Thank you very much.
And use your time to go to the poster, to go to the
sponsor booth.
The sponsor.
Yeah,
so use this hour as you more prefer. Okay. Hello, everybody. We are ready to start this second section with me and Professor Mebarek. That is is a big pleasure. Tomorrow she will participate in the school, and now we start with a section with a keynote. That is very important people. Is Professor Alighieri, that is from University of Brescia, is associate professor, and she is a deputy president and founder of the Italian Society of Extracellular Vesicle. It's a big pleasure that she is here with us today. She is currently the leads alongside Paolo Borghese of the Biogenic Colloid Surfaces Interface Lab at the Department of Molecular and Translational Medicine in the University of Brescia. So please, we are ready. And now she speaks. I don't want to wrong like Massimo. The title.
No, the title is
sorry,
unconservative. Unconservative.
The title of this keynote is Beyond the Surface, delving into biomolecular corona and biogenic nanoparticles.
Okay. Very happy to be here. Thanks for the organizer for inviting me to give this lecture that actually will have will give an open view on what's happening in the field of biomolecular corona on extracellular vesicles. It has an intent also educational. So I come from this beautiful city. It's not actually my hometown where I was born, but it's very nice. So if you come by to Brescia, you can visit mountains, lakes, and the city itself, which has also Roman ruins. So it's very interesting. And as Professor said, I'm also deputy president of the Italian Society for Extracellular Vesicle, which was founded in 2018, and is a very young society and open to young fellows also. So we have many grants for students, and this was our last symposium in Urbino. And next year we will have the big symposium of the society in the beautiful island of Favignana in Sicily. So maybe if you're interested, you can come by. So I would like to start my talk just by where everything was born, so by synthetic nanoparticles. So the field of biomolecular corona actually started from researchers working in the synthetic nanoparticle field. So as you might know, when a synthetic nanoparticle is conceived, is synthesized, it bears its own synthetic identity. But as soon as the nanoparticle is injected in a biological fluid, immediately it acquires a new identity. That is, the surface of the nanoparticles changes due to many interactions with biomolecules that are present in the biofluids. And this in turn changes also the physiological response that this nanoparticle exerts onto cells. So the interaction with cell membranes then change inevitably. So this concept has been this phenomenon has been explained with the concept of the biomolecular corona. That is the absorption of biomolecules onto the surface of nanoparticles, which happens in all biofluids. And depending on the bondings that these biomolecules have with the nanoparticle itself, we can have weak bondings or tighter bondings, so we can have a hard or a soft corona, so a different corona formation on the surface of the nanoparticle. And this in turn changes the biological identity of this nanoparticle itself. So in the recent years, actually from the 2018, it has been discovered that the biomolecular corona happens also for extracellular nanoparticles, that is, natural nanoparticle as
EVs as lipoproteins and other biogenic nanoparticles. So this has been discovered actually from the group, the first one was actually the Edith Buccia's groups from Hungary. But could we ask, in Italian we say, is the discovery of the acqua calda, so it's just something so new, this rediscovery of the wheel that actually also natural nanoparticles have a biomolecular corona. And actually, yes, it is the discovery of the acqua calda because vesicles and other nanoparticles have other biological system, and as synthetic nanoparticles are colloids. And colloid is an entity that belongs to the nanometer size. So colloids are like viruses, vesicles, synthetic nanoparticles. And what do these nanoparticles, tiny things, have in common? So they have a peculiar feature, which is the large surface to volume ratio, which gives them a high interfacial free energy. So they are very prone to interact with other particles and with other biomolecules. So, for example, in our, and they tend also to interact with what's in the secretome, in the biofluid, which we know that it is very crowded. And actually this morning I saw the presentation of the matrix vesicles, which I'm very new in the field, so I feel this is much more crowded than other compositions, etc. And actually vesicles, in our lab, we analyzed, we did this study, and we have shown that if you consider, for example, small nanoparticles, that is exosomes, so the most part of the proteins that the exosome show are present on the surface, not in the bulk. So the most interesting part of an exosome is not what's inside in terms of protein content, but it's on the surface. And so, given this, there are many proteins and biomolecules that have been known and been to associate on the surface of EV. This review made by this researcher Halal shows and resumes the biomolecules that have been found on the surface of vesicle, and they call it associated membrane components or interacting particles. And as you see here, we can find many proteins, of course, but also nucleic acids, but also other nanoparticles like lipoproteins. So actually we can ask, who crowns who? Who is the corona of who? So are vesicles the corona of lipoproteins, or it's vice versa? And this interaction is being studied right now by some laboratories, actually the interaction between EVs and lipoproteins, and it actually promises to unveil some new roles of EVs and lipoproteins in themselves. So it happens that maybe they hypothesize that this interaction between lipoproteins and EVs can mediate the transcytosis of the vesicle across the endothelial barrier. So it's a very interesting field that is being explored right now by some researchers. And by the way, also lipoprotein bear a biomolecular corona. So it's a mess. So and this biomolecular corona gives these lipoproteins many new properties. That is, so they can have a role in the antioxidative, anti-inflammatory roles, or in hemostasis. So many new functions are being found by in this field. So what are the main features of this corona on these natural nanoparticles? So the first thing is that it's the surface environment interface which determines the composition of the corona. So the corona depends on the composition of the surface, so how is the nanoparticle made, but also of what it encounters on the biomolecules that it encounters in the environment. So if we think about the huge heterogeneity of vesicles, we can have exosomes, we can have microvesicles, and every microvesicle can be generated in many, many different ways. We can think about the fact that every vesicle can have a different corona. And also, if we think about the fact that vesicles have different physicochemical properties, so their surface changes inevitably. So we can have really different ways of formation of this protein corona. And another thing about, I said surface environment, about environment is that we need to keep in mind that every time we culture, so if many of us work with cell lines and work with vesicles produced by cells, so every time we change the culturing condition of our cell line, then we change the protein corona identity. So here is just an example that shows that cell cultures cultured in 2D or 3D then have a different protein corona on their vesicles. So just, this is one feature of the corona. Another feature that actually Professor Milan today has also mentioned is the fact that not all the biomolecules that are present in the biofluid will be found on the corona. So there is a specific enrichment of selective partners, let's say, on the surface of these nanoparticles. And this is because not all the biomolecules can interact with the surface of these vesicles. And this happens also for synthetic nanoparticle, of course. So we got into this field by studying a protein that is normally known as a free like protein in plasma, which is antithrombin. So antithrombin is among the principal inhibitor of local coagulation, and it's a serine protease family. Its secretion process is well known, secreted by the liver. It's a very highly glycosylated protein. So we found, together with other groups actually, that antithrombin is not only freely present in the plasma, but is also transported by extracellular vesicles, and it's localized on a V surface. And we found this by performing some biochemical assays as dot blot assay and trypsin treatment. So we found that this protein is actually adsorbed on the surface of the vesicle. And what we found most interesting is that not all the glycoforms present in circulation of these proteins are present also on the vesicles. So by 2D electrophoresis, we show that these are all the glycoforms spots referring to different glycoforms of the protein in the plasma. But if we analyzed the glycoforms on the AVs, basically we don't find the same spots. Okay, so not just because of the quantity of the protein that is in there, but for the quality of the type of these glycoforms. So we can say that there's a selectivity in absorption in terms of the type of proteins that gets attached on the surface of these vesicles. To sum up then, so the potential factors that influence the absorption of the biomolecules onto natural nanoparticles, we have to consider the biofluid composition, the fact that there's a crowded secretome, so they are very prone this interaction to happen, the biomolecular and physicochemical property of the nanoparticle itself, and also we have to consider that if the biomolecule, the protein, has a modification, has a mutation, has a post-translational modification, then the corona will change. Maybe this protein won't be able anymore to be present on the corona. And so by observing this, actually, by knowing this, if we think about a pathological condition, if we think about a change in the pH or a protein concentration, so we can think that the corona can really become a new platform for biomarker discovery and for pathogenesis studies. Indeed, in the work of antithrombin, we asked if could this antithrombin adsorption specificity be exploited for deficiency diagnostics. Indeed, there are some deficiencies of antithrombin which are hardly diagnosed because of the lack of tests. And so, briefly, to make it very short, we found that if we compare antithrombin-free plasma glycoprotein profile and antithrombin bound to vesicles, we see more differences in the vesicle-bound form of antithrombin instead of the free plasma. So we think that by adding a new dimension, that is the absorption capacity of a protein, we can add a new tool to discover pathologies. Okay, so in conclusion of this diagnostic part, we can say that AV protein corona interaction, selectivity, and environment dependence opens a new perspective for biomarker studies. And the protein corona composition is influenced by, I say, the glycan profile, but it can be any post-translational modification. And also the study is extendable to all the pathologies that are related to post-translational modification, that is cancer or glycosylation diseases. So, of course, the topic of the biomolecular corona was born and has been discovered in the field of drug delivery, okay, because people were trying to see how this drug delivery system, why they couldn't actually deliver what they should have been, what they were actually synthesized for. So, we have to know that due to its feature, EV corona may impact not only the diagnostic field, but also the application of EVs in therapy. And this may, in terms of EV administration in vivo, in vitro, and also during the EV surface engineering process. So it has been shown in this work from Dirk Strunk laboratory that he took the secretome from placental expanded stromal cells, and he separated vesicles by different cycles of tangential flow filtration and then size exclusion or ultracentrifugation. And basically what he saw is that as soon as like going on with the separating steps, then he progressively removed the corona from the vesicles. And this in turn mediated a loss of function of the vesicle itself. So that could partially be reestablished with actually with applying some albumin or some factors, but not all. So I think it's a similar observation that also professor found. So we need to be aware that every time we culture the cells in some ways using FBS, using 2D or 3D condition, and then when we separate vesicles with some certain methodology, then we inevitably change the EV biomolecular corona composition and therefore its function. So in our lab, we asked if we could modulate EV function by leveraging on the formation of the EV corona. And actually, we got into this field while we were working in a project in which we were trying to engineer vesicles for a targeted therapy. So we actually work with the red blood cell-derived extracellular vesicles. And we tried to engineer them with the antibody cetuximab directed, which recognizes the EGF receptor, which is highly expressed in triple-negative breast cancer cell lines. So this was the target of our study. So we decided to explore two edge cases of EVs modified with this antibody. The first case was by a covalent binding of the antibody onto the surface of the vesicles. So just by click chemistry, we bound the antibody on the surface of the vesicle. And the second strategy was just by incubating the antibody with the vesicle, so just by physisorption, that is by formation of a protein corona, on cetuximab corona on the surface of these vesicles. And what we saw is that if we when we detected the amount of antibody that was bound on the surface of this vesicle, basically it was the same for the two formulations. So the click chemistry ones and the Physi one had the same amount of antibody bound on their surface. And then we asked if these chemisorb and physisorb vesicles worked in the same way. And we did these experiments by performing surface plasmon resonance. So by using a sensor chip that had immobilized the EGF receptor. And so we found that also the KD for EGF receptor was the same for both samples, for both formulations. So on a molecular level, basically these two, they work the same. But then when we went in vitro, so we tried to see the uptake of these EVs in MDA MB231 cell line, which is a TNBC cell line, we saw that when we were in more a physiological condition, that is in the presence of FBS, we could see that the click one won actually in respect to the Physi one. And we believe that this is because when we administer vesicles that are clicked with our cetuximab, basically the cetuximab is there and then binds to the EGF receptor, and this mediates the uptake of the vesicle inside the cell. While when we administer a physisorb formulation, then the antibody is in equilibrium with all the proteins that are present in the surrounding medium. So the corona exchanges, and then we lose the ability of the vesicles to bind to EGF receptor. But this was our case, because in other cases, actually corona and by performing a strategy of pre-coating EVs with albumin actually can enhance the uptake. So it depends on the type of vesicles that you have, the type of corona that you have, and the type of cells that you are performing the experiments with. And so by these experiments, we got really interested in studying how these exchanges on the surface of the vesicle happen. And so we asked, how could we study the dynamics of this biomolecular corona in situ, given the fact that every time that we try to separate vesicle from their biofluid, then we change the biomolecular corona composition. So we never know how it will be composed. So we tried to face this by applying fluorescent correlation spectroscopy to study this plasma biomolecular corona in situ. So our system was built by using the cetuximab. Actually, we got that in Italy. We don't have much money, so we reuse our ideas, actually. And so we modified our cetuximab. Actually, it was a fluorescent cetuximab. And we put it in contact with two different vesicles, type of vesicle. The first one are mesenchymal stromal cell EVs, and the other ones are red blood cell EVs, which we believe are the most interesting vesicle that have a future in terms of therapy. And we basically created a biomolecular corona of cetuximab on their surface. And then we measured the fluctuation of these vesicles in a confocal volume. And by this fluctuation, you can actually extrapolate an autocorrelation function, which gives you the idea and the numbers in terms of size and in terms of concentration of the object in solution. So this all happens in solution. So we don't have to change anything in the biofluid. And then we spiked these vesicles with plasma proteins, and we tried to measure how was the size of these particles in solution. And basically, this is a resume of what we did in allegorical presentation. We submitted this for a cover. So this is our nanoparticle illuminated by a laser, okay? And this is the corona which exchanges. Okay, this was the first result that we got. So just by incubating and analyzing the vesicles in solution just with the cetuximab, we could see that a more abundant fraction of cetuximab is associated to mesenchymal-derived EVs than to red blood cell-derived EVs. So there's a specificity of binding. Indeed, here in the red blood cell-derived one, we have a display, a larger fraction of free cetuximab. So there's let's say a preference for association to mesenchymal-derived EVs than red blood cell-derived EVs. And second, what we saw by spiking plasma protein to the sample, we saw that both Rev-Physi and Mev-Physi display a significant detachment of cetuximab. So we see that the plasma proteins physically detach the cetuximab from the surface of the vesicles. And this is different in a different way between the two samples. And you can see also here by measuring the hydrodynamic diameters, that is the formation of the corona by the plasma proteins. And this happens in a different way between if we compare Mev-Physi or Rev-Physi samples. And finally, we tried to evaluate how is this corona forming, how is this corona forming on the surface of the vesicles. And first we tried to perform a stoichiometric calculation. And so we tried to calculate the nominal hydrodynamic diameter of the vesicles. But every time we tried to make this calculation, then we in turn could see that the experimental diameter was always bigger than the one we calculated. And this was true for all plasma concentration tested. So what we believe is happening here is that we don't have a formation of regular layers of corona on the surface of vesicles, but rather a formation of these like protein monolayers, which actually are then accompanied by protein aggregates. And actually, another work from Edith Boutzas showed that maybe it's this was happening. So single vesicle analysis showed that these aggregates can happen on the surface of vesicles. So in conclusion, we believe that, I hope I've convinced you that this biomolecular corona impacts the function of the vesicles, and its dynamic can be monitored by non-disruptive methods as FCS. Its formation and evolution may be influenced by the EV origin, and therefore it gives also a huge heterogeneity. And its architecture seems consistent among the different EV subtype studies, so it happens for everyone, every type of vesicle, despite different dynamics as we have shown. So what we are doing in our lab is to develop, we are developing new methods to handle these studies in terms of all different things that you can see here, learning from the synthetic nanoparticle world. And so to do this, we are, as Massimo mentioned today, we are organizing this workshop in February, and we will bring together experts that, like the founder of the corona, Kenneth Dawson, and other experts in the field of synthetic biomolecular corona, with other experts in the V field to get together and to learn a little bit more. What we're doing now, experiments running with the fluorescent cross-coloration spectroscopy to check how these interactions between EVs and lipoproteins happen in situ, so not by after separating them. But I see that we can explore also many other interactions, also from the studies I saw this morning. And I thank you. This is my laboratory, which I co-lead with Professor Paolo Bergese, and the experiments that I show here, they were performed by Angelo Musicò, Andrea Zendrini, and Silvia Lacqua, and we have a poster by Selene Tassoni here in our lab. And these are our collaborators, and thank you for your attention.
Thank you. Very, very interesting. I have a question, but first of all, there are questions from the public. A lot of, probably. Please.
Thank you. So nice presentation. Just a question, because the ISEP Society, I remember we spent many years to try to remove lipoproteins from EV, because they are, in particular for people working with plasma, the big contaminant. No, for EV. So with your presentation, you showed that this link between EV and lipoprotein can modify the biological activity of EV and their interaction with target cells. But you think, in your opinion, that could be also the lipid composition of EV affect this interaction? It can be possible the lipoprotein and V can exchange lipid and cooperate together in the biological function of V.
Yeah, these are all cool questions. Well, first of all, yeah, ISEP actually has to move to other ideas and new ideas. So yeah, if we thought in the past that we need to have a very pure preparation of vesicle, I think it's impossible. So because the bondings that keep together a vesicle are the same one that are formed between the biomolecular corona components. So it's impossible to remove them. And we need to be aware that every time that we separate vesicle, in some ways we will have these lipoproteins if they are attached in some ways. And this all, actually, I agree. I agree that they also depend on the composition. Indeed, what we show here is that the composition of the corona. So the corona doesn't cover all the surface of a vesicle. So many lipid parts. If we think that a vesicle is composed by layers, lipid rafts, whatever. So maybe there are some free zones of exposure in which lipids can interact, and maybe there can be an exchange of lipids. It's something that we should explore. I think it's very interesting because lipoprotein actually exchange.
Exactly. This is and lipids are very dynamic, so yeah, could be a very great possibility. Yes. Thank you for your answer. Thank you.
Yeah.
What is an easy there is an easy method to remove corona, just to compare the effect of the vesicle with and without?
Yes, okay, it depends. Okay, you know, every time, many people, we shaved, for example, with trypsin. Okay, this is another, but you know, sometimes it depends on the bondings that keep together the protein corona components with the vesicles. So maybe just a simple wash with some detergent, like mild detergent, can detach some protein. Or just immersing a vesicle, immerging, whatever, in water can detach the corona. So it really like just a change in the pH can detach the corona. So it depends if you are talking about there is this hard corona, maybe this needs like a trypsin treatment, and maybe the soft corona bound proteins they just need maybe a soft method.
Trypsin may modify also transmembrane protein of the vesicle.
I mean, you have to know which are the transmembrane protein first, and then exclude them from the analysis. It's not so obvious. I mean, people are are working on this. So it's
mild detergent might be better.
It depends on the component. Maybe you don't have to destroy the vesicle, though.
Thank you, Professor Ariglieri.
That's it.
We can go on. We will see in Brescia in February.
So now it's time for the section 4 on media vesicles, and I have the opportunity to present the next speaker, Maria Elena Repici. She got her PhD degree from the University of Torino, and postdoctoral studies spend time at the University of Lausanne, Switzerland, and the University of Paris. Before, sorry, I can't read. I have a problem with my eyes.
Okay.
Sorry.
And
before moving to the UK in 2010, where she worked at the University of Leicester. She joined after Aston University in Birmingham in 2019, where she currently runs her own group with a strong focus in molecular mechanism of neurodegeneration. So the title is Regulates. No, it's
no, because they moved.
Oh, yeah.
Actually, the first part of the title, so you laugh a bit, is under here. Is like my affiliation. The right title is Parkinson's-associated protein DJ1 regulates intercellular communication via IV in oxidative stress. So this is why I'm not Parkinson's disease affiliated in any way. I am an academic at university. Okay, so thanks a lot, first of all, to Professor Bottini and all the organizers for inviting me here to share with you the work that has been done in my lab since when I became an independent PI. So I am a neurobiologist by background, and I work on Parkinson's disease. So just, obviously I could talk for Parkinson's forever, that is not the aim of this conference, so I just set the context. For the ones of you who are not so familiar with neurodegenerative diseases, Parkinson's is the second most common neurodegenerative disorder after Alzheimer. It is characterized by the loss, do you see the mouse cursor, no? Where's there? Yes. It is characterized by the loss of these specific neurons in the substantia nigra pars compacta of the brain. In the remaining neurons, we have the formation of these halo structures, okay, that are aggregated of mainly alpha-synuclein. There are motor symptoms. We all know that Parkinson presents with gait, stiffness, and I'm not a neurologist either, so I am a basic scientist working on molecular mechanisms. So we know that there are sporadic and familiar forms of Parkinson, and as basic scientists, we use the familiar forms to understand a little bit more on what happens on the disease, okay? And this is exactly what I'm trying to do. So I work on one specific protein that is called DJ1. DJ1 is responsible for autosomal recessive forms of Parkinson's disease. So Vincenzo Bonifati, who is a big Italian neurologist, identified DJ1 protein in 2003. This is the first science paper that showed that individuals with DJ1 mutations develop Parkinson's at about 40 years of age. What do we know about DJ1? Again, I cut the long story short. This is the structure of the protein. It's a dimer, okay? And in this dimer, there are some specific residues that are very important for DJ1 function in the protection from oxidative stress. And oxidative stress is a key player in Parkinson's pathogenesis, okay? So this is well known. Now, the real question is how? And no one knows exactly how DJ1 can trigger Parkinson's disease pathogenesis. We just know that if we knock out DJ1 in C. elegans, in Drosophila, in millions of different cellular models, we have an increased sensitivity to oxidative stress. This is what we know. Now, what is the link between DJ1 and extracellular vesicles? When I started as an independent PI at Aston University, I had the chance to meet people and my current colleagues who are kind of EV experts, and they are immunologists. And then I started reading about EVs in neurodegeneration, and there were some publications indicating that indeed DJ1 can be found in exosomes from biological fluid of Parkinson's patients more than age-matched controls. So my question was at that time, what is the meaning of this? Is this relevant to Parkinson's pathogenesis, yes or no? Now, in the neurodegeneration field, we know that EVs can play an important role. So we know that, for example, if we think of alpha-synuclein being present in an exosome or an EV released by a degenerating neuron, this can be transmitted to a normal neuron to start the pathogenesis process. However, we also know that the same EVs carrying abnormal aggregated protein in neurodegeneration can activate the other two cell type of the central nervous system, so astrocytes and microglia, to activate and contribute to the neuroinflammation process. Okay, so this is what triggered my interest when I started this entire story. Now, to set the context for the model, so we started using neuroblastoma SHSY5Y cells. Obviously, I'm not selling these as a model of neurons because they are not. They are similar, and I'm very, kind of short. You need to say things as they are. I will have iPSCs derived data as well to convince you that what we saw is real. So neuroblastoma cells can be differentiated in vitro using retinoic acid and BDNF into what I call dopaminergic-like cells. Okay, so these cells, they have, as you can see, some neurofilament H, which is a neuronal marker, some dopa decarboxylase, which is a specific dopaminergic marker. So we have our ongoing model for these kind of dopaminergic-like neurons. And then we use wild-type cells and CRISPR knockout cells for DJ1, so cells without DJ1. And then we looked at the EVs produced by the wild-type, by the DJ1 knockout cells. Now, because of this context, I think that is important to tell you how we isolated the EVs. So we harvested the medium, then we centrifuged down to eliminate dead cell and cellular debris, then we concentrated using Amicon filters with a cutoff of 30 kilodalton. We used ISOQ EV column to obtain the EV fraction, we concentrated again, and this is what we used in our experiments. Now, to define the EV population at the beginning, we used this staining with BodyP FLSE, overnight 5 micromolar concentration, and we used the system that has been described. I mean, here I talk to experts, you know, a million times more than me, so I think I don't need to explain, but we use the beads to calibrate our Cytoflex system so that we could identify large and small particles here. Okay, so large more than 200 nanometers diameter and small less than 200. And then we use this gating in flow cytometry to look at the population originated from differentiated SHSY cells. And this is what we obtained. So first observation is that we didn't really have that many large EVs. We had very many small EVs. And we started looking at what happened in control conditions. So we are not triggering the system as it happens in PD, just in control. And to be honest, I did not expect to see any difference, but we saw some differences. So here is the result we have here. So these are very solid data for a model that has its limitations, obviously. But you can clearly see, first observation is that by differentiating these cells, we increase the number of EV released in the medium, which is obviously what we could expect if we think that there is more intercellular communication in differentiated cells. But second of all, when we look at the difference between PARC7 is the DJ1 gene, and wild type, we saw that we obtain more EVs from PARC7 than from wild type. And this total is reflecting what happens for small EVs, mainly, okay? We proved in collaboration with the University of Warwick and Saskia Bakker that by using cryo-electron microscopy, what we were looking at were real EVs, and these are some of the features in terms of shapes that we obtained for the EV. And then we triggered the system just by oxidative stress. So we used rotenone. People who know a lot about Parkinson's know that rotenone, paraquat, they are typically used for inducing some oxidative stress and mimic a part of the Parkinson's disease pathogenesis, obviously in an in vitro model. So rotenone is an inhibitor of mitochondria complex 1, and this is what we obtained. So first of all, we can see that when, I don't understand why this one doesn't work. Okay, so if you look at what happens at 5 nanomolar concentration, you can see that we don't have any increase in the small EV per microliter here with 5 nanomolar from the DJ1 knockout cells. But we do have from the wild type cells. When we look at what happened at 10 nanomolar, the situation is exactly flipped, so it's the opposite. So this is telling us that if the cells respond to oxidative stress via EVs, and if EVs have a physiological role or versus pathological, this means that the absence of DJ1 is making the cells respond via EV in a different manner. Okay? Then, so these were kind of interesting data, and then we focused our attention at 10 nanomolar, because this is the only concentration that is statistically, thank you so much, statistically significantly different. Which one is this? Yes. So 10 nanomolar is the only concentration for which there is a difference between the two genotypes, but also between each genotype and its respective control. To understand that something was really happening, because obviously we checked, and I don't have time here to show all the studies and everything, but these cells were not dying, so we checked for necrosis. There was no necrosis, and this was not the aim, because we just wanted to stress the cells in a similar way to what could happen in PD. There was no apoptosis either, but there is a mitochondrial damage. So here we evaluated the mitochondrial healthy state of these cells in control condition or in rotenone condition for wild type cells and PARC7, so DJ1 knockout cells. And you can clearly see already that when we use mitospy, that is a dye that labels healthy mitochondria, so when the mitochondria are losing their polarization state, there is less labeling. We saw indeed an effect that is much stronger for PARC7 knockout cells, and this is nothing new. We expected that. As well as when we reconstructed the average branch length of the mitochondria, we observed the same. Okay, so we have this difference at 10 nanomolar in terms of EV produced, and we know that there is a mitochondrial damage. What about the cargo? So we did, I think again, for this context it's important to know that in this case, when we did mass spectrometry to analyze the proteomic signature of these EVs, we pulled together, for each N is the medium pulled together from 5 T75 flasks, so you needed really tons of these, and my PhD student was going mad. But we managed to somehow do some nice mass spectrometry here, and you can clearly see by just looking at the heat map. So these are the 3 samples PARC7 knockout, so EVs from, we are only comparing here rotenon condition for PARC7 and for wild-type cells. This is the Vulcano plot showing our data. We identified in a significant way 116 proteins, 50 overexpressed in the EV from DJ1 knockout, and 66 overexpressed in the EV from wild type. And interestingly, because obviously if science is science, things need to work in a certain manner, when we did the gene ontology analysis against the human prot databases for cellular component, we identified as the most higher in terms of percentage of genes. We have cytoplasm, exosome, lysosome, and plasma membrane. So this is another confirmation that we are really looking at EVs. And then, okay, here I cut this because, again, I don't have time, but Tom did, with my PhD student, did cluster analysis based on fold change, and we identified 5 different clusters. The cluster 1 is represented by only one protein that is completely absent in the EV from DJ1 knockout, and then we have other different clusters. And interestingly, I think the message from this slide is that all these proteins are involved in intracellular signaling and trafficking, okay? Stress response, inflammatory regulation. So this is already super interesting to us from a disease perspective. Now, if I can't have a readout, this is all pointless to me, studying a disease. So I needed to have a readout, and as a readout, we used THP1-derived macrophages, and we evaluated the ability of the EVs obtained upon rotenon treatment from wild-type and DJ1 knockout cells to trigger the migration of these macrophages. How did we do that? With a migration assay here. So this is a transwell. In the upper part, we have our THP1-derived macrophages. In the lower part, we put our EVs from rotenon-treated control or wild type or DJ1 knockout cells, and these are our results. So this is the control, negative control, which means here we have only medium, and we observe that in control condition we have more migration that is induced by the EV from the wild type cells than DJ1 knockout. However, when we did rotenon at 10 nanomolar, this is exactly the opposite. Okay, so this is again telling us that not only there is a difference in cargo, but there is a difference in functional readout. We have some preliminary data, because obviously my aim is to move up to models that are more closer to my disease of interest. So I use primary human astrocytes. So again, these are EVs obtained from neuroblastoma cells with and without DJ1 upon rotenon treatment. And we looked at the morphology, just to start with, of primary human astrocytes by looking at GFAP. This is just the first immediate marker. There are so many more we could look at, but this is just preliminary data I'm showing you today. And you can see that while in area we didn't see any differences, when we look at mean intensity fluorescence, this is just an n equal 2, okay? So we need to repeat this. However, the EV from control neuroblastoma are having a very different effect from the EV from rotenon or from not EV, which again is suggesting a real important physiological role. Because I had a very, because exactly in my field, if you don't go to iPSCs, no one considers your work fine, we moved to iPSC-derived neurons. So we took, Mark Coxon in NIH gave me, and kindly gave me the iPSC cells, isogenic control, and DJ1 knockout. We transformed these cells. These are the rosettes, and this is my DJ1 that is not present in the neuronal precursor cells. And then we differentiated these cells in neurons. Okay, so these are neurons at 12 days in culture upon differentiation. And we again triggered the system with rotenon, and we again saw a difference response in terms of number of EVs, which seems to suggest that this is not only in my neuroblastoma model. So to conclude, I think that we are currently investigating our candidate proteins, and for these candidate proteins I have two possibilities. The first one is that the EVs we are looking are a snapshot of what is happening in the cells. The second possibility is that we have a selective loading of cargo into the EVs that is obviously dependent on the oxidative stress condition. And I finish with my graphical abstract. What I think is happening is that EVs play an important role in the intercellular communication in the CNS. We don't know that much about this, but in control condition, they have a specific cargo that is modified in mild stress or in high stress conditions, and these EVs act on the other cells, such as astrocytes and microglia, to orchestrate the final degeneration of the neurons. And with this, I conclude, and I think I need to thank a lot Thomas Page, who brilliantly had his viva, so he became doctor at the beginning of November, Clara and David for the help with the iPSCs, Dr. Ivana Milic for the help with mass spectrometry, and Professor Andrew David for his expertise on EVs, and my funders. Thank you.
Thank you very much for your presentation, very impressive. So, any question? No question? Because it's very impressive, I think.
There are two possibilities. Yeah. The other one is I was crap. So,
have you any data on the specific content of the Viva mass spectrometry?
Yes, so I can
some
tell
suggestion
in terms of what I think.
Yes.
So I still don't understand all the story about. So the one only cluster is RUBL2, which is a protein that is involved in nucleosome remodeling. So this could be something that is important in the reaction of stress. All the other candidates are synaptotagmin 11. Lysosome trafficking protein, so all proteins that are involved in the lysosome, and I think that this is really in agreement with the idea of the field now, that what is happening in Parkinson's disease is a failure of the proteostasis. Yeah. In terms of markers of EVs, we know, for example, that I can see CD45, CD63, CD81, and they are all depleted in DJ1. In EV from DJ1 knockout, but what does it mean for their physiological function is very complicated. So I need some years to study more about it.
Maybe I a little lost, but you mentioned about the neuronal system, neurons and the astrocytes. What about the glioma cells? If they
the microglia. No, I mentioned microglia.
Okay.
So microglia is also interesting because microglia are the macrophages of the central nervous system. The problem is that you need to know all the three components isolated and then put them together, which is something that I would like to do in my next grant. So taking EVs from neuronal population, putting on astrocytes, and taking EVs from astrocytes, and then you need to have the stress stimulus, so it becomes really complicated. But this is the only way we have to really understand what is happening.
Thank you for your presentation. Just a question, how long was the treatment with rotenol?
24 hours.
The cell's still alive?
Yeah, perfectly fine.
Okay.
So we measured necrosis, apoptosis, and nothing is significant. The only thing, and this is a very good question, is that we started seeing an increase in nuclear circularity upon rotenol, that to me means probably very, very early phases of apoptosis. But the cells were still there, and we normalized all our EV data aside from the oxidative stress condition, because in oxidative stress you can have some detachment, but all the other ones are normalized by number, and the iPSCs, because when you work with iPSCs, you seed some, you don't know how many cells you have at the end. So we did tiling at confocal microscopy to evaluate all the possible cells that were on my tiny cover slip.
Okay.
If it makes sense.
Did you check for autophagy? because you know mitochondrial dysfunction and mitochondrial stress are strongly associated with induction of autophagy.
I think autophagy definitely plays a role.
Autophagy can modify the secretion of EV. So I was just thinking if you check this type of process.
Yeah, the problem is that the data on autophagy and DJ1 are very controversial in the literature. So sometimes some papers show the DJ1 increases, some papers show the DJ1 decreases autophagy. So this is why I didn't look at, but what I really think is happening is that maybe because we overload with oxidized proteins, autophagy can't cope anymore, and maybe these cells produce more EVs as a way to get rid of the oxidized protein. This is my idea at the moment, but obviously we need to do experiments to prove it. But definitely, autophagy plays a role in this story for sure. Thank you.
Thank you.
Okay, thank you again.
Thank you.
After. Okay. So the next speaker is Dr. Simeon. He got his PhD in the University of... yes, it's difficult to pronounce. So Chief Pescara in 2013. During his PhD, he focused on cancer biology and signaling pathway relating to neoplastic transformation. Then he worked at Oncox Biotech on developing monoclonal antibodies targeting TROP2 for oncological clinical application. In 2014, he joined the cytomorphology lab at the University of C.T. Pescara, learning multiparametric flow cytometry to study rare events, antigen-specific lymphocytes, and EVs. So the title is...
Okay. Thank you all. Thank you to the organizer to give me the opportunity to present our preliminary data on the extracellular vesicle from Treg as a novel approach to immunomodulatory therapies. Let's start. Obviously, some concepts that have been exposed many times today. So, but according to the definition provided by International Society for the Extracellular Vesicle, the term extracellular vesicle refers to small particle released by all the cell, surrounded by a lipid layer that lacking the ability to replicate independently, as they don't contain a functional nucleus. Traditionally, EV are classified into exosome, microvesicle, and apoptotic bodies, and these classes of EV have very different biogenesis processes. Exosome range in size from 50 to about 100 nanometers, originate from endosomal system, and are released following fusion with the cell membrane. Microvesicles, also known as ectosome or macroparticle, vary in size from 100 to 1000 nanometers and barred directly from cell membrane. Finally, apoptotic bodies are released by the cell undergoing programmed cell death. To define these entities, ISEV recommend the use of generic terms EV and operational extension of this term instead of inconsistently defined and sometimes misleading terms like isosome or microvesicle. These later terms are often associated with biogenesis pathways and are challenging to demonstrate with precision. Furthermore, it has been observed that subclasses of extracellular vesicle exhibit significant overlap in terms of size, and commonly used preparative techniques and protocols produce heterogeneous population vesicles. The current preferred classification categorizes EV into small EV with a size smaller than 200 nanometers and large EV if larger than 200 nanometers. Extracellular vesicle mediate intercellular crosstalk. It has been demonstrated that most EV contain important biological molecules like RNA, including small non-coding RNA, microRNA, long non-coding RNA, mRNA, DNA, lipids, and proteins. Scientific interest in EV increased significantly once it was established that this vesicle can functionally transfer molecules between cells ever over long distances. Moreover, interest in their role has grown further with the observation that EV uptake by cell may be mediated, at least in part, by receptors, suggesting specificity to certain subclasses. What are the advantages of extracellular vesicle-based therapy? Extracellular vesicles are considered a promising delivery vehicle for various genetic therapies. They are relatively inert, non-immunogenic, biodegradable, and biocompatible because are produced by animal cell by the human cell. EV are stable in the bloodstream, protecting their cargo, and numerous studies have demonstrated their ability to overcome physical barriers, such as the blood-brain barrier. EV can specifically interact both local and distant targets, and they can be designed and engineered to carry a delivery therapeutic molecule, including proteins and RNA, and can be designed and engineered to target specific target. However, a deeper understanding of basic biological of EV is still required, including cellular production mechanism, cellular release, and systematic distribution. This knowledge is crucial to efficiently use these endogenous cellular nanoparticles as next-generation delivery tools. For example, to establish with precision dosage and frequencies, or establish the administration route, the right administration route. And last but not least, a proper GMP protocols. In the recent years, in my lab, Laboratory of Cytomorphology of University of Chieti Pescara, we developed and patented an innovative method for cytometric analysis and FACS-based separation of extracellular vesicles. These methods can be applied to many biological fluids, blood, urine, cell culture, supernatant, and is formed of two reagents, a lipophilic cationic dye that label intact EV, and was chosen between many candidates, and a fluorescent phalidin that have the ability to identify non-intact EV because bind the F-actin. For example, in atypical protocols applied on peripheral blood, we identify in flow cytometry a platelet-free area using platelet as a reference dimensional material, and analyze these events for the positivity to phalloidin and negativity, the positivity to lipophilic cationic dye and negativity to phalloidin. These events are intact EV, could be analyzed for other, but the presence other markers, for example, CD45, CD31, and CD41. In general, we use CD45 for EV from leukocytes, CD31 for endothelial EV, and CD41 for the platelet EV. The advantages are several. We need only small volume to analyze. For example, for peripheral blood, only 5 microliters. Is fast protocols, about 45 minutes of staining time. We don't need lysis or washing steps. With this method allow a versatile flow cytometry analysis for characterization, absolute count, and instrumental sorting. To go on, I have to introduce what's regulatory T cell. The regulatory T cell, formerly known as suppressor T cell, are a subpopulation of T cells that modulate the immune system, maintain tolerance to self-antigen, and prevent autoimmune disease. T cells are immunosuppressive and generally suppress or downregulate induction and proliferation of effector cell. In general, in flow cytometry are defined as event at the subpopulation of T cell, CD4 plus, CD positive, CD25 positive, and FOXP3 positive. For this reason, the co-administration of regulatory T cell alongside conventional T cell in HLA haploidentical transplantation has emerged as a promising strategy to improve transplant outcome, in particular in acute leukemia patients. Tregs, which are immunosuppressive, help to prevent graft versus host disease by reducing the immune response that conventional T make direct against the recipient tissue. This is critical for reducing the complication of GVHD. Simultaneously, the conventional T retain their ability to mount an immune response against residual leukemia cells, preserving the graft versus leukemia effect. In this way, we obtain a dual benefit of reducing the GVHD complication while preserving effective leukemia eradication. Treg can be isolated from peripheral blood, umbilical cord blood, leukapheresis, or GCSF mobilized peripheral blood by magnetic cell separation or flow cytometry sorting use GMP-grade closed system. Isolated Treg are then ex vivo expanded using anti-CD3 CD28 magnetic expander bit or artificial antigen presenting cell in the presence of interleukin 2 or without rapamycin. We aim to isolate extracellular vesicle produced by Treg using the cytometric method described in the previous slides in order to improve therapeutic strategies based on Treg. What was the question? May Treg-derived EV have significant immunomodulatory activity and, given their raw toxicity, could represent a promising therapeutic strategy for GVHD? Treg-derived EV were characterized using this novel flow cytometry patented method and isolated through FACS cell sorting. After isolation, both morphological and dimensional analyses were performed using various techniques, including atomic force microscopy, electron microscopy, and NTA analysis. The characterization revealed that Treg-derived EV had sites within the small EV range, under 200 nanometers. Following the isolation, the EV protein cargo was characterized using proteomics and Western blotting to confirm their vesicular nature. As shown in the future, as you can see in this figure, the vesicle display classical EV marker such as CD63, CD81, flotilin, and the preparation was free of contamination, as can be demonstrated by the absence of cytochrome C, that is a typical contaminant of the preparation, EV preparation. Unbiased bioinformatic analysis revealed that Treg EV cargo proteins were associated with vesicular events, forming cluster of protein critical for immune system function, and other cluster was linked to potential immunomodulatory activity. Different amount of EV were used to treat CFSE stained and activated autologous T effector cell to test immunosuppressive function. The different concentration of EV showed that a direct correlation with the percentage of T effector cell inhibition, reaching a plateau at higher EV concentration. The results show that after 24 hours of treatment, the optimal EV concentration to achieve the best inhibition profile on 100,000 autologous T effector cell, about 97-98% of inhibition, was between 4-6 million of EV. Nevertheless, we select 3 million of EV as working concentration, given that the measured inhibition effect produced by this EV concentration overlap is very similar to that induced by Treg parental cell in 1:1 ratio, and which represent the gold standard for such an inhibition assay. The total protein content carried by derived EV is linear to the number of EV. Moreover, the inhibition effect of Treg EV were comparable to, if not greater than, those induced by parental cell. With our surprise, we found that EV purified by FACS exhibited a threefold higher inhibition capacity compared to the same EV obtained through ultracentrifugation. To investigate the possible immunomodulatory activity in vivo of extracellular vesicle produced by Treg, we use a mouse model of GVHD based on NSG mice. This model lacks functional TB and natural NK cell. And this condition of immunodeficiency prevents the rejection of human cell. So in this way, an SG mice can be engrafted with the human immune cell, PBMC, or for example T effector cell, to create a model that mimic immune responses, including GVHD. The first data on the immunomodulatory role of Treg-derived EV demonstrated that effectiveness in vivo models. For administration of 3 million of Treg-derived EV, successful delay the onset of GVHD in an SG mouse model in a comparable way to the Treg cell. To conclude, Treg-EV can be efficiently characterized and separated by fluorescence-activated cell sorter. Treg-EV display a strong immunomodulatory effect. These EV have a strong potential as new therapeutic agents in many different hematological diseases, as GVHD, display low toxicity, low immunogenicity, and relevant functional potential. And finally, preliminary data demonstrate how Treg-EV display immunomodulatory effect in vivo. Thanks to my lab, in particular the flow cytometry unit of my university, and the group of hematology unit of the Pescara Hospital, the oncology unit, the general pathology unit of my university, the University of Perugia, and proteomic unit for the analysis of protein content. Thank you.
Thank you very much for your presentation. Question?
Thank you for your presentation. Just a question about flow cytometry analysis that you perform. So you discriminate EV particles from not intact EV by staining EV with the lipoprobes and non-intact EV with phalloidine.
Yeah.
But what about other non-EV lipid particles?
Non-EV?
Yes.
Yes. We demonstrated, for example, we applied these protocols, for example, in blood sample, okay? And we demonstrated that if you use, for example, antibodies, we distinguish this EV from, for example, lipoproteins. So when you sort these EV and compare the contamination of FACS sorter EV and, for example, ultracentrifugation, We published a paper in 2020 and demonstrate that the contamination from apolipoprotein, for example, fall down for, I think, 5 times more. In particular,
so you have to combine the lipoprobes together with the specific antibodies.
Yeah. In general, when we use, for example, supernatant from cell line, you don't need of antibodies because in general you have a more homogeneous preparation. Okay,
it depends, because if you use serum or the plate serum, it's
obviously but if you have the blood is a mess, so it's a compulsory to use antibody. Okay, if you have obviously you need, but it's not so compulsory. Okay. If you have the blood sample or, for example, plasma or serum, it's compulsory to use antibodies, because obviously you have a very heterogeneous preparation. So if you want to isolate some part of that, you have to need to use antibodies.
Thank you. Thank you.
When you treat with the vesicle your cells, how many EV per cell you use?
Allora, in this contest we use 3 million for 100,000 cells. 3 million of Treg EV, okay, for 100,000 effector T. Okay. We try with 4 or 6, but about the million for 1 to 10, about
1 to 10.
Yeah.
Other question? So thank you for your intervention. And now we are ready to start with the last, but the best section of this day, that is dedicated for the young people, called Rising Star. We have 8. Please remain in the time. 8 presentation, and we start with Dr. Belade. Yes, and she have a presentation titled "Evaluating the Stability of MCCVS for Oral Delivery."
Okay. Hello everyone, my name is Mona. I'm a final year PhD student at King's College London and National University of Singapore, and my talk today is about evaluating the stability of MSCEVs for oral delivery. I just want a disclaimer that this data is very new. There's a manuscript in preparation, but it's not been published yet. So mesenchymal stem cell-derived extracellular vesicles, or MSCEVs, have regenerative properties and immunomodulatory properties, which make them promising therapeutics for wound healing and inflammatory conditions. They're most commonly administered by injection. However, there are instances where oral delivery would be more advantageous. Oral delivery is more convenient for patients. It can also reduce side effects associated with systemic administration, and it can also help to directly target the site of action for conditions that affect the gastrointestinal tract, such as inflammatory bowel disease, which is the chronic inflammation of the gastrointestinal tract and typically affects the intestines. So by delivering EVs orally, we can target directly the site of action, such as the large intestine or the colon, and we can deliver an effective dose directly to the site of action to reduce the inflammation. However, there are also challenges to oral delivery, and the EVs would need to survive the harsh gastrointestinal environment and the extreme pH levels, and also the digestive enzymes, which break down proteins, lipids, and sugars. So researchers have investigated the effect of stability of the milk EVs and plant-derived EVs in gastrointestinal tract, but there is very limited information on the stability of the MSC EVs, and that's what I'm going to talk about today. So just very briefly, the EVs were isolated from the culture medium of mesenchymal stem cells derived from the human bone marrow. We isolated the EVs using ultracentrifugation on sucrose cushion, and then the EVs were characterized for particle size, concentration, and the presence of EV markers such as CD81 and THG101 and ALEX and so on. Now, to investigate the stability of the MSC EVs in the gastrointestinal tract, we collected the EVs and then incubated them in gastric simulated fluids. So we've got the gastric fluids, the intestinal fluids, and then the enzymes as well, pepsin and pancreatin. We incubated them at a ratio of 1:1. And then we collected the EVs and we measured the changes to the EVs using NTA, a nanoflow cytometry, and cryo-EM. So looking at the NTA data, we can see that there are changes to the EVs in terms of particle size. So when the EVs are in water, we see that the mode particle size is 137 nanometers, so most of the particles are within this range. However, when you incubate the EVs in gastric fluids and in pepsin, the enzyme that breaks down proteins, we can see that there's a reduction in particle size, where the most particles come into the range of 77 nanometers. And this is also the same thing when the EVs are incubated in intestinal fluid and pancreatin, where the mode particle size reduces to 67 nanometers. So this suggests that the EVs are broken down because they're not stable in the gastrointestinal fluids. We also selected two EV markers, CD81, which is a transmembrane protein, and THG101 as an internal marker, to measure for nanoflow cytometry. So when the EVs are in water, we can detect the presence of the EV markers because the EVs are intact. So we have THG101, some EVs have CD81 only, and then some EVs have THG101 and CD81. However, when you incubate the EVs in gastric fluid, there's a significant decrease in the EV marker detection. And that's because the EVs, once again, are not stable. And we can see this as well in the intestinal fluids. So this supports the data that the EVs are broken down because they're not stable in the gastrointestinal fluids. And here we have a selection of images which shows the state of the EVs at different stages of digestion. So when you look at the image on the left, this shows intact EVs. You have a membrane, you have circular shape. However, after incubation with the gastrointestinal fluids and with the digestive enzymes, pepsin and pancreatin, we see structural changes and deformities to the EVs. So we can see here, for example, rupture of the membrane. The EVs are no longer circular. And then the image on the top right here, you can see just a membrane structure because the EVs have just been destroyed and it's just no longer circular. So we can see here from this data that the EVs are clearly not stable in the gastrointestinal fluids. However, if we want to deliver them orally to target the inflammation in the intestine, for example, is there something we can do to protect the EVs? This is where formulation comes in. So we developed a double coating formulation to target colon delivery and protect the EVs to increase their stability in the GI fluids. So how we did that, we used chitosan coating and udogate coating. So it's a double coating formulation. So first we add the EVs to the chitosan to form a single layer. The EVs and the chitosan have opposite charge, so they attract each other and they form a coating. And the reason we use chitosan is because it's very commonly used in the formulation for drug delivery to the colon. It's also mucoadhesive, so it helps the EVs to be delivered to the injured tissue. And then we add udogate to the mix. So udogate S100, we add it to the mix stepwise and we stir, and this creates the formation of white particles, and these are the coated EVs. So the reason for the double coating is to target the delivery to the colon. So we start off with the double coated EVs straight after oral administration, and then the udogate S100 doesn't dissolve until pH 7, so it's actually stable in the stomach and in the beginning of the small intestine, and it's only when it reaches the final stage of the small intestine where the pH increases above 7 that the udogate dissolves, and we only have chitosans coating the EVs, and that's what enters the colon. And then the bacteria in the colon breaks down the chitosan and releases the EVs at the site of action. Right, so we've characterized this new formulation, and when we coat the EVs with the chitosan, we always see an increase in size by almost double, as we can see here from the results of the NTA and then the cryo-imaging as well. And there's also a change to the charge, so the EVs are normally negatively charged, but when we coat them with chitosan, they become positively charged. And then when we add on the udogate coating, we see these white particles forming. So we image them using scanning electron microscopy, or SEM. And we can see here that from the images, that actually the udogate precipitates around the EVs and it groups them together. But we can still see here individual EVs coated by the chitosan. So we evaluated the stability of the EVs, of the coated EVs, and we did that using scanning electron microscopy. So we incubated the coated EVs in the gastric fluids in the presence of the enzymes, and what we see here is almost no difference. So the coated EVs remained stable, they remained intact, there was no change to the structure, we could still see the white particles forming, and we can still see here like the individual EVs coated. So the coating was stable and it protected the EVs from degradation. So we also looked at the stability of the EVs in the nanoflow cytometry, so the detection of the EV markers. So if the coating was stable, then we would not detect any EVs released in the supranatant. But if the coating was broken down, then we would detect EV presence in the supranatants. So in the water here, for example, there is no presence of the EV markers, the same ones as before, so CD81 and THG101. And that's because the EVs are inside the coating, so they have not been released from into the supranatant. We still have those white particles. And then when the EVs are incubated in the gastrointestinal fluids, we also see no markers present, and that's because the EVs are also inside the coating. That the coating protected the EVs from degradation, and we don't see the release of the EVs or the presence of the EV markers. And it's only when we when we add the coating EVs to the colonic fluid, which has a pH of above 7, and this dissolves the udogate layer, and then with the chitosanase present, which kind of mimics the bacteria degradation of the chitosan. So the chitosan then breaks down, and this releases intact EVs into the colonic fluid. And this is supported by the presence of the EV markers that are detected using nanoflow cytometry. So the coating, just to summarize, the coating here protects the EVs, and it can release the intact EVs into the colonic fluid. Okay, so just to summarize what I've just presented today, so the MSC EVs are not stable in the gastrointestinal environment, and if we want to deliver the EVs orally, we need to protect them from degradation using an oral formulation. I just explained that we developed a double coating formulation for the MSC EVs, which stabilized the EVs and then protected them from degradation and targeted the colon delivery by releasing the EVs in the colonic fluid. However, there's an important question that remains is, are the EVs stable? Do they have therapeutic activity when they reach the colon? And that's something I'm going to talk about at the ISEF annual meeting, hopefully, because when we investigated the effects of the EVs after oral administration in vivo, so we investigated the stability. We investigated the efficacy of the oral formulation in mice, and I'm going to present this data hopefully at the ISEF annual conference. Spoiler alert: they do work. So just my acknowledgements. I want to thank the organizing secretariat for inviting me to the conference and for sponsoring me in Aquarella as well. I want to acknowledge my supervisors and the colleagues who have helped me in this work, and I want to thank everyone here for listening. Thank you.
We have time for one question. Please.
Congratulations for your talk.
Thank you.
I was wondering, what is the effect of the chitosan on the surface of the vesicles? I mean, does it get attached? That when you solubilize it, when you degrade it, just then simply goes away, or you have still traces of the chitosan and
thank you for your question. So the chitosan interacts with the EVs that they have electrostatic attraction because they have opposite charges. So they form a coating, but it's not like a strong coating. The chitosanase. So normally the chitosan is targeting the colon. People use it in formulations for the colon, and what it is that in the colon you have bacteria that breaks down chitosan. So when the chitosan breaks down, it just leaves the EVs, and then the EVs are just released. Obviously, the truth is that some EVs also get broken down because of this. It's just that it's just minimal enough that we still have an effect.
Okay, thank you.
Thank you.
Thank you. The next is Dr. Cavarles, and the talk will be on small extracellular vesicles, propagation, neurodegenerative mRNA in Alzheimer's disease.
Hello everyone, my name is Maria Cavarles. I'm a third-year graduate student in the Chen Lab at Sanford Burnham Prebys in San Diego. Our lab does a lot of genomic sequencing, so sorry if it's a little boring, but we're still interested in small extracellular vesicles, and we're particularly interested in how they propagate neurodegenerative mRNAs found in AD. Some background on Alzheimer's disease. It's a progressive disease with no cure. The symptoms are loss of memory and cognition with difficulty performing basic tasks. And its pathology is characterized by the reduction of brain size and the presence of neurotoxic proteins causing neuroinflammation. The hallmarks of AD are amyloid beta plaques and tau neurofibrillary tangles, which start to aggregate at one portion of the brain and over time will spread throughout the brain as it advances. A lot of AD is familiar with the familial form of this disease, and it accounts for 5% of cases due to the PSEN and APP mutations. However, sporadic AD is the most common form, and it arises from a combination of environmental factors and genes, presumably due to somatic events occurring in postmitotic cells such as neurons. Current therapies targeting these AD hallmarks exist but are not very effective to restore cognition. Therefore, we're interested in looking at other mechanisms that propagate neurodegeneration. And we can see here that EV contents are altered in disease states. SEVs are released upon depolarization in neural cells, and increased ATP and microglia in astrocytes will release the vesicles. In disease states, you can see that there's pathogenic peptides and lipids and nucleic acids that are upregulated in these EVs, causing aggregation of these pathogenic molecules that enhance cell death and stimulate an inflammatory response. EVs also, from the periphery outside of the brain, can cross the blood-brain barrier, and can influence the neurodegeneration as well. And a lot of interesting AD research focuses on these hallmarks, such as APP. In this proteomics study, they show that there are 88 proteins in healthy patient brains, and disease and healthy patient-derived EVs share 852 proteins, while the AD subset has a unique subset of proteins that are related to the AD SEVs. And we see an increased load of these neurotoxic proteins, especially the amyloid beta 42 and phosphorylated tau in these diseased vesicles. And it's been shown that the EVs propagate tau protein as this prion-like mechanism. In this study here, they injected tau EVs from patients into mouse brain, and the mice brain actually used the tau as a template to start misfolding. But when we think of vesicles, they are kind of reminiscent of viruses. Viruses are exported using the escort-dependent pathway via shedding or budding. They contain structural proteins such as capsids containing the viral genome that are DNA or RNA, and they have retroviral capabilities. SEVs are exported with the escort-dependent or independent pathway. They contain structural and soluble proteins, and of course they contain DNA, microRNAs, and mRNAs, which I will be talking today. And the effect of this could be inflammatory signaling, or possibly these nucleic acids could be exogenously processed for direct translation. So we proposed that SEVs containing these AD mRNAs can spread this AD phenotype, starting with the diseased neuron propagating these SEV mRNAs, specifically APP, that can be taken on by a healthy cell and eventually express the neurotoxic proteins, whereas a reactive microglia will contain SEV mRNAs propagating an inflammatory response and affect a resting microglia to switch to a reactive form, which can be pathological as it will attack neurons as well. And in our lab, we have isolated SEVs from the human brain. We take slices of postmortem brain tissue, and we dissociate the tissue with collagenase, and then we do successive spins of ultracentrifugation, and then we do differential gradient ultracentrifugation to collect the different fractions. And when we characterize these human brain SEVs, we find that fraction 2 contains the EV markers flocculin, TSGM101, CD63, and CD81, and they're depleted in cell debris markers, making this a very clean preparation. And with the TEM, we see that the morphology of the SEVs is retained, and that by nanoparticle tracking analysis, the vesicles are around 100 to 200 nanometers. So we do short read RNA sequencing to get an idea of the transcriptome of the diseased vesicles and healthy vesicles from these patients. And we corroborate this data with long-read sequencing, which will give you the intact form of the mRNAs, making them more likely translatable as well. And you can see the differential expression of the non-diseased and AD patient-derived vesicles. AD vesicles are enriched for transcripts related to regulation of kinase activity, regulation of type 1 interferon production, and regulation of defense response, implicating an increase in an inflammatory response in the brain. While non-diseased vesicles are enriched in synaptic signaling and brain development, indicating a healthy brain functioning. And some of these transcripts that we found were related to AD disease causal transcripts such as ApoE, APP, PSEN1, and MS4, which is expressed in inflammatory microglia. But because we did the total brain transcriptomics, we wanted to do a cell-specific analysis of these mRNAs, so we produced mouse primary brain cells. We took vesicles from neurons, astrocytes, and microglia, and we compared their transcriptomes to other vesicles of other cell types and to their cells of origin. And we find that the microglia vesicles and whole and their cells share a lot of transcripts, while astrocytes and neuron vesicles cluster together but away from their cells of origin. And on the right side, you can see that many of these transcripts for the different cell types are related to Alzheimer's disease. And more interestingly, we found that the human brain SEVs are enriched for transposable elements. In the differential expression analysis of the bulk brain tissue versus SEVs, there is a unique subset of transposable elements only expressed in the SEVs, such as Lyme1HS. Lyme1HS is the youngest retrotransposon, so part of the Line1 family, and it is capable of retrotransposition, and it accounts for 80% of the total reads. So the fact that these SEVs contain this transposable element and can deliver RNA make this potential mechanism for retroinsertion into a recipient genome. So some ongoing developments we have in the lab to follow up on this amazing work is to do transferable EV assays. So we have moved on to develop isolating vesicles from mouse brain of mouse models, and to do this, we use size exclusion chromatography, since we find that the morphology of the vesicles is retained, it's better repeatability, and in my opinion, retains their functionality as well. And in this study, this really quick study, we loaded the EVs from mouse neural progenitor cells with an EGFP plasmid. Since we're interested in transfer of nucleic acids into recipient cells, we place these loaded EVs onto naive cells, which will then express the EGFP plasmid, whereas the control condition where the EVs incubated with plasmid alone had 0 signals. So this kind of validates that the SEC method retains the bioactivity of the vesicles to deliver nucleic acid into recipient cells. So today I shared with you that the 80 human brain vesicles contain a distinct subset of full-length mRNAs. There's cell type specific packaging of these mRNAs, and these SEVs contain distinct TEs compared to bulk brain tissue. And for our future interests, we're interested in looking at AD pathology due to this mRNA delivery mechanism, and we want to investigate the role of these cell-specific mRNAs delivered by SEVs. So I'd like to thank my lab and all the other labs that helped me in my research so far, and for CSCV for inviting me and for organizing this event. Thank you.
Thank you very much. Question? No question? Yes, one question.
Maybe it's a naive question, but I'm not a specialist for neuronal systems. So it's possible to compare she mentioned about the Parkinson and Alzheimer disease. The EVs may be similar or not? For these two models, two diseases.
I mean, you can correct me on this as well. PSEN is also related to Parkinson's as well. Some people use it for AD, but we focus on amyloid beta and tau. So with PSEN, there's a different. Yeah.
Exactly, yeah. So it's very. I think they are different. However, if there are common molecular mechanisms that maybe you can have in neurodegeneration, they will be there for both diseases. But I have another question that is, how, when you looked at your miRNA from the mouse model
and from your human brains, how similar, because the big problem in AD field is that we can never reproduce, right, the animal data in humans. So how often did you find the same interesting mRNA candidates?
Yeah, so we do see the same mRNA candidates in the human model, the healthy human models, and with the primary as well. In terms of isoforms, the transcripts, how different they are, I'd have to ask to remember what that is, yeah.
Thank you. Now, Dr. Florio from University, D'Annunzio University, will be speak about exploring antitumor activity of B7H3 CAR-T derived extracellular vesicles in pancreatic cancer cell. Please.
I thank the organizers for giving me the opportunity to speak in this meeting. In Professor Kama laboratory, together with several collaborators, we are studying the anti-tumor effect of extracellular vesicles derived from B7H3 CAR-T cells on pancreatic cancer cells. Pancreatic cancer is one of the most aggressive tumors with poor prognosis and high mortality. At present, conventional treatment strategies for pancreatic cancer show limited clinical efficacy. Moreover, the immunosuppressive tumor microenvironment causes a lack of response to current immunotherapy approaches. Therefore, novel therapeutic options against pancreatic cancer are urgently needed. Chimeric antigen receptor cell therapy emerged as a promising immunotherapeutic approach to cancer treatment, particularly in the management of hematological malignancies. However, the use of CAR-T cell therapy in solid tumors is still limited because of the absence of tumor-specific antigens, the immunosuppressive tumor microenvironment, tumor barriers, antigen escape, and the occurrence of severe toxicity. The use of extracellular vesicles derived from CAR-T cells in the treatment of solid tumors has several potential advantages when compared to parental CAR-T cells. Extracellular vesicles are considered cell-free immunotherapy agents that easily cross the physical tumor barriers. Furthermore, CAR-T extracellular vesicles are resistant to an immunosuppressive tumor microenvironment, and they are associated with the low collateral toxicity risks. This study aimed to explore the question of whether extracellular vesicles derived from CAR-T cells have the potential to improve pancreatic cancer treatment. Like in other solid tumors, the choice of CAR-T antigen target is critical. Among several CAR-T target candidates, B7H3 protein is overexpressed in pancreatic cancer with limited expression in normal tissues. For this reason, this target appears one of the most promising candidate targets in pancreatic cancer. This is a flowchart of our experimental procedures that include transduction of CAR-T cells, isolation of extracellular vesicles from CAR-T cells, characterization of these extracellular vesicles, and analysis of killing activity of these extracellular vesicles on pancreatic cancer cells. First, we generated CAR-T cells targeting B7H3 antigen. The transduction efficiency of CAR-T cells was generally greater than 60%, as assessed by flow cytometry using the B7H3 ligand. Flow cytometry analysis confirmed that all tested PC cell lines displayed high levels of B7H3, although with a certain degree of difference among cell lines. For our initial study, we chose L3.6PL cells, which has an intermediate level of B7H3 expression. We investigated the anti-tumor potential of B7H3 CAR-T cells. L3.6PL cells were co-cultured with B7H3 CAR-T cells and untransduced T cells at different effector to tumor cell ratios. killing assay was assessed by flow cytometry using the 7ADD assay. Results showed that B7H3 CAR T cells significantly affected cell viability on target L3.6 PL cells in a dose-dependent manner. Then we isolated extracellular vesicles from B7H3 CAR T cells and untransduced T cells by a flow cytometry method patented by our collaborator, Professor Lanuti. This method allowed to identify, count, and isolate intact extracellular vesicles. These extracellular vesicles were first characterized by atomic force microscopy for the evaluation of their size and integrity. Most of these extracellular vesicles displayed a globular shape and were in isolated form. Diameter and size distribution of extracellular vesicles were also measured by nanoparticle tracking analysis. Extracellular vesicles had an average size of 114 nanometers. Further characterization of these extracellular vesicles by Western blot analysis confirmed the presence of canonical ISEV marker CD63 and flotilin-1 and confirmed the absence of cytochrome C, which is present only in control cells as expected. These results were in line with those obtained using flow cytometry analysis. The expression of CAR molecules on extracellular vesicles and on their parental CAR T cell surface was measurement by flow cytometry analysis. Notably, extracellular vesicles derived from B7H3 CAR T cells expressed CAR on their surface. Moreover, considering that extracellular vesicles has a surface 3 to 4 orders of magnitude smaller than their parental CAR T cells, they had a higher density of CAR on their surface. Granzyme B and perforin are known effector of CAR T killing activity. Thus, we evaluated the presence of this enzyme using flow cytometry analysis. Our results showed that B7H3 CAR T derived extracellular vesicles carry high level of granzyme B and perforin. Here we investigated the anti-tumor potential of B7H3 CAR T derived extracellular vesicles on pancreatic cancer cells. L3.6 PL cells were treated with B7H3 CAR T derived extracellular vesicles, and then we performed a killing assay using both MTT test and 7ADD assay. MTT results show that B7H3 CAR T derived extracellular vesicles significantly affected cell viability in a dose-dependent manner compared as a control extracellular vesicles. Furthermore, the 7ADD assay showed that B7H3 CAR T derived extracellular vesicles exerted a time-dependent killing activity against L3.6 PL cells. In particular, at 24 hours of treatment, the killing was 14%, and at 48 hours, the killing was 22%. In conclusion, we generated B7H3 CAR T cells that revealed killing activity on PC cells. B7H3 CAR T derived extracellular vesicles expressed CAR molecules on their surface, and they carried granzyme B and perforin. B7H3 CAR T derived extracellular vesicles killed PC cells in a dose and time dependent manner. Further, in vitro and in vivo studies are needed to deepen the potential of B7H3 CAR T derived extracellular vesicles in pancreatic cancer therapy. I would like to thank colleagues and all of you for the attention.
Thank you. And some questions? Not sorry. And after coffee.
Thank you for your presentation. Just a technical curiosity, how did you concentrate your sample after sorting for the downstream biological effects of your EV?
After sorting, we used a different centrifugation and ultracentrifugation binary protocols, and then we choose the better one concentration for our test.
Okay, now because the good things of sorting, I think that preserve the integrity of EV after the isolation. So combining ultracentrifugation can affect the quality of your EV isolation. So maybe
no, because in the with nanoparticle tracking analysis, the extracellular vesicles are intact, and
okay, thank you.
I have only a general question on the experiment that if you have analyzed the toxicity also in non-tumoral cells, healthy?
No, we didn't perform this experiment, but extracellular vesicles are not able to proliferate, and they have a limited lifespan, and are cleared rapidly for the circulation.
Okay. Other suggestion? Okay, I suggest to have a break time to a coffee. 10 minutes, yes, okay. Only 10 minutes, but it's needed. It's okay, yeah.
So it's time to begin. So the next speaker is Dr. Fanelli from University of Rome, Tor Vergata, and the title of the seminar is Evaluation of Myeloid Activation Marking Blood Cells and Circulating EVs as a New Tools Monitoring SARS-CoV-2 Infection and Long COVID-Associated Sequelae.
Okay, thank you. I want to thank the scientific committee for this opportunity to present our data regarding the evaluation of myeloid activation markers in blood cells and circulating extracellular vesicle as a new tool for monitoring SARS-CoV-2 infection and long COVID-associated sequelae. CD169 is a adhesion molecule expressed on the surface of macrophages and monocytes and can bind granulocytes, erythrocytes, B and T cells. Different work highlight the function of CD169 positive macrophages in human disease, such as in immunotolerance, in tumors, in autoimmune disease, and particularly in viral infection. In fact, they have act a two-rule antiviral rule or proviral rule in viral infections, such as in HIV. Very important is the early response of the innate immunity to the infection. Indeed, during the viral infection, the dendritic cell release interferon alpha and beta and induce monocyte to production of CD169 on their membrane. On the other hand, during bacterial infection, the dendritic cell induce natural killer cells to production interferon gamma and stimulate neutrophils to production of CD64 on their membrane. Those two markers are very important to link the innate immunity and adaptive immunity. In this work, those two markers were used from emergency unit to discriminate patient with bacterial infection that express CD64 on the surface of neutrophils and the patient with viral infection that express CD169 on monocytes. COVID-19 disease is a complex disease caused by SARS-CoV-2 and induces initially a hyperactivation immune system characterized by a cytokine storm, and after lead to a lymphopenia, a depletion of CD8 T cells and exhausted, and lead to a multi-organ dysfunction. Different work highlight the function of CD169 positive macrophages in COVID-19, and in this context, our group, in collaboration with Department of Research and Development, Beckman Coulter Life Science Immunotech, tested the use of CD169 and CD4 as marker to discriminate viral versus bacterial infection, and particularly to the early detection of SARS-CoV-2 infection. We use a particular ratio RMFI calculated from CD169 mean fluorescence intensity ratio between activated monocytes and lymphocytes, and we define a cutoff value. Over this cutoff, we define the positive of SARS-CoV-2 infection. I report our work in which we define and demonstrated that CD169 RMFI is a good marker for early detection and can discriminate healthy donors from COVID-19 patients. And also, this ratio reflects the immunophenotype disruption and oxygen need in COVID-19 patients. COVID-19 is a complex disease and is characterized by different detrimental conditions, particularly the alteration of immune system and also different symptoms. And after the resolution of infection, those symptoms persisting, and we know the post-acute sequelae of SARS-CoV-2 infection. That the name is PASC or long COVID individuals that are characterized by multifactorial characteristics, and those conditions such as psychiatric, cutaneous, gastrointestinal or systemic symptoms are linked with a persistence of immune dysfunction. And some works define that there is a chronic inflammation, alteration of different subpopulation of naive B and T cells, and also the exhaustion of CD8 positive T cells.
In this context, in collaboration with the Department of Infection Disease from Policlinico Rome Tor Vergata, we enrolled healthy donors, COVID-19 patients and PASC individuals. And we demonstrated that CD139 RMFI is a good marker of viral infection and was significantly higher in COVID-19 patients. And also we demonstrated that the percentage of monocyte, the high percentage of monocyte positive CD169, and particularly the percentage of CD169 HLA-DR positive monocytes was significantly higher in COVID-19 patients respect to the healthy donors. And these characteristics persisted the persistence in PASC individuals or long COVID individuals. And also we demonstrated the downregulation of HLA-DR positive monocytes. It's very important to characterize the markers, the expression of markers at the level of the cells, but it's very important to know the role of extracellular vesicle because it represents an important mechanism of communication cell to cell. And it's important to know that the extracellular vesicle reflect the cell of origin. And in this context, in COVID-19 disease and in PASC syndrome, some work define that the role of extracellular vesicle as anti-SARS-CoV-2 effect because extracellular vesicle show on the membrane ACE2 receptor and can bind SARS-CoV-2. But on the other hand, some work define a negative role of extracellular vesicle as a mediator of SARS-CoV-2 because carrier a virus, SARS-CoV-2, also RNA or protein. And in one work describe extracellular vesicle in PASC individual and those extracellular vesicle containing a residual RNA and spike protein and end protein, and this represent a possible reservoir or SARS-CoV-2. In this context, we characterize a subgroup of our enrolled cohort and COVID-19 patient and PASC individual and healthy donors. And through flow cytometry, we analyzed the markers CD169 and HLA-DR activation marker on the different subpopulation leukocytes and extracellular vesicle isolated from plasma. I report here a gate strategy that we use for characterize extracellular vesicle. We use a gigamix that are characterized from beads with a specific dimension from 0.1 to 0.9 micrometers, and we use this gate to characterize extracellular vesicle. And starting from unstained sample, we define the expression of these markers HLA-DR, CD169, and the double expression of this marker on extracellular vesicles. Firstly, we analyzed the leukocytes of population and we confirmed the persistence of circulating CD169 positive monocytes and double positive with HLA-DR and the downregulation of HLA-DR in COVID-19 patient and LC or PASC individual. And also we demonstrated the high and significant percentage of HLA-DR positive lymphocytes in COVID-19 patient and LC. After we analyze the extracellular vesicle and we define also the percentage and also the number of extracellular vesicle from microliter of plasma expressing the different markers analyzed, HLA-DR, CD165 and the double expression. And we demonstrated the high number and the high percentage of HLA-DR positive, CD169 positive extracellular vesicle in COVID-19 and in LC. We use the gate strategy and arbitrarily we analyze 3 different subgroups of EVs considering 3 different dimensions, and we demonstrated that the extracellular vesicle double positive, HLA-DR positive and CD169 were significantly higher in COVID-19 patient and LC respect to the healthy donors. We analyze also the possible correlation, and in COVID-19 patient, the number of EVs double positive correlate directly with CD169 RMFI and with the percentage of HLA-DR positive, CD169 positive monocytes. In long COVID patient, we demonstrated a positive correlation between the positive EVs of HLA-DR with the percentage of HLA-DR positive monocytes. And we demonstrated that EVs double positive correlates with coagulation factor and inflammatory indices in COVID-19 patient, and in LC individuals those correlation were lost, but persisting the correlation with dimer. In conclusion, we demonstrated that CD169 RMFI is a reliable marker of acute infection in COVID-19 patient, and the low expression of HLA-DR represent an immune dysregulation in COVID-19 and also in PASCO or long COVID individuals. We demonstrated the high percentage of monocyte double positive HLA-DR and CD169 was significantly higher in COVID-19 patient and the persistence in PASC individual. And also we characterized the extracellular vesicle, and particularly the double positive extracellular vesicle HLA-DR CD169 was significantly higher in COVID-19 patient and in LC on PASC individuals. CD169 is a good marker in early viral infection and is important for monitoring immunological dysfunction. I report a recent work in which CD169 is used as a possible antiviral therapeutic target. I want to thank all of you and my group, particularly Professor Claudia Matteucci, Dr. Antonella Minutolo, and the whole collaboration. Thank you for your attention.
Thank you very much for your presentation.
Thank
Questions? I have a question. How did you isolate your extracellular vesicles? You used flow cytometry?
Yes.
Yeah. And you isolate from plasma?
Yes. From plasma with a centrifuge 13,000 RPM.
Okay. And okay. Thank you. Question?
Okay.
Thank you.
Thank you. So we now is the time for Dr. Protto from National Institute of Health, that presented the HSV infection induces phosphorylated tau propagation among neurons via extracellular vesicles. Please.
Yes. So, hello to everyone, and first of all, I want to thank you, the organizing committee, for inviting me and for giving me the opportunity to presenting you this study. So, HSV-1 is a neurotropic virus that is very diffuse into the population, and it first makes contact with the host, with the orolabial mucosa, and then it infects the neurons of trigeminal ganglia, where it established a lifelong latency. Several stimuli could reactivate the virus, which can migrate again to the site of primary infection, but it can also enter the central nervous system, when it can trigger a massive immune response, which is called the herpetic encephalitis. However, we know that now the HSV-1 can also enter the brain and entail mild infection, and many studies in the last 20 years correlated this mild infection with an increased risk for the Alzheimer's disease onset. So the Alzheimer's disease, we all know that is this neurodegenerative disorder, and from a molecular point of view, It is characterized by the presence of the accumulation of the beta peptides, that is the results of the processing of the amyloid precursor protein, and the beta aggregates outside of the cells in amyloid plaques. While intracellularly, there are aggregates that are called neurofibrillary tangles that are the results of the accumulation of the tau protein, which is hypophosphorylated and which detach from the microtubules. These two aggregates and accumulation induce trigger the activation of the microglia and the immune cells, resident cells, that try to clear these aggregates. And this could induce a neuroinflammation that could increase damage and produce a lot of oxidative stress that again could increase the stress. As we now know also from the really nice previous presentation, the extracellular vesicles in Alzheimer's disease are also important because they can pack this neurotoxin protein, such as beta and phosphotau, and this could are related to the dissemination of the pathology among brain cells. So where the HSV1 is involved in all these molecular pathways? Here I try to sum a lot of pathways that are related to neurodegeneration that are results of many in vitro studies, and we performed some of that in vitro, and I to sum up that here HSV1 it is known that HSV1 could induce the APP phosphorylation and the amyloidogenic processing, could favor the tau phosphorylation, could alter the epigenetic alteration and increase the oxidative stress. And how I will show you shortly, it also is involved in the exosome release and damage propagation. So we also set up an in vivo mouse model in the last years that we use here, and I will show you briefly that we set up an in vivo mice model of recurrent infection. So we usually infect the mice through the lips here, and then we wait for the establishment of latency. Then we induce the activation of the virus, performing thermal stress. We induce the activation of the viruses, and we perform this cycle for several times through the lives of the mice. And after the several cycles, the mice develop an AD-like phenotype. Indeed, we can see in these mice that there is accumulation of beta and aggregates, phosphorylation of tau, neuroinflammation, and also cognitive deficits that we measured by behavioral tests. Using this mice model, AD-like phenotype, we also characterized others neurodegenerative pathways. So here are some papers. There's some studies about the oxidative stress that is present in the brain of these mice, the epigenetic alterations that we can measure, and also deficits in the neurogenesis process that we know it is important for the memories, memory. So how the story with exosomes started? When we were analyzing the brain of these mice, we noticed that the increase in the tau phosphorylation that we were analyzing was not restricted only to the cells that were positive for a viral marker, like GB, that is the viral glycoprotein GB, so this is the cells that show the active replication of the cells. So we hypothesized and we asked ourselves if in HSV1-induced neurodegeneration, the HSV1 could promote the dissemination of tau among brain cells through extracellular vesicle, as occurs in Alzheimer's disease. So first of all, we analyzed the species of tau that are inside cells or that are released in primary cortical neurons. So here we chose to analyze these three sites that are tau 217, tau 205, and tau 181. which are strongly related to Alzheimer's diseases. As you can see, the modulation of these three sites are really different inside cells. So in primary neurons, we have a strong increase inside cells of 2117, while 205 and 181 slightly decrease and migrates toward the nucleus. And we confirm this also by Western blot. When we analyzed by LSSA the content of tau protein inside of the neurons and outside of the cells, so we measuring the tau outside of the cells, we noticed that inside we had a slightly decrease of the protein that is paralleled by a strong increase of the tau that is released outside of the cells following HSV1 infection. So we are, and this is more dependent. So this is a clue that tau could be indeed released outside of the cells by the HSV1 virus. So then we started to isolate exosomes and extracellular vesicles, indeed, and we started to set up the isolation with neuroblastoma cell line, which is a human neuroblastoma cell lines. So we did the isolation by ultracentrifugation, and these are the population that we isolated. So the larger vesicles here, and this is the size distribution, and this is the small vesicles that are more homogeneous. And then we characterize this also by the marker that we all know, that are ALIX, TSG, and flotilin, and then we analyze the content of tau and phosphotau in these vesicles that we isolated. So following HSV1 infection, we have a strong increase of tau 181 in the large vesicles in particular, but also tau 205 and 217, also in the small vesicles, the exosomes, also at high molecular levels, which are compatible to the forms that are partially aggregated of tau. Next, we confirm this data with a different model that are primary culture of neurons. And in this case, these are the population that we isolated that are a little bit less homogeneous. This is the size distribution. Here are the macrovesicles and the large vesicles, and here are the small vesicles. And again, we characterize the presence of the markers, and this is the content of tau and phosphotau inside of these vesicles. So here we have again an increase in the microvesicles and exosomes that are derived from HSV1 infected neurons, and also an increase in tau 181 in HSV1 microvesicles derived from HSV1 infected cells at high molecular level. Then we ask ourselves if if HSV1 could induce the transmission of this tau that we found increase in these vesicles among neurons. So we performed a recipient assay. So we transfected the neuro-2A cells to a neuroblastoma cell line with a recombinant tau that is a human tau tagged with the protein GFP to track the protein. Then we infected the neuroblastoma transfected cells, and we purify the extracellular vesicles of these cultures. Then we treated the vesicles with ultraviolet to inactivate the virus that could potentially hide inside the extracellular vesicles, and then finally we layered these vesicles on recipient cells that we chose to be tau knockout neurons. So how can you see here, this is the neuroblastoma that are transfected with the recombinant protein. As you can see that the infection does not alter the expression of the recombinant protein, but also in this case the infection increased the level of tau that is phosphorylated, in particular in the site 205. Then we follow the protein inside the vesicles, and here we have the large vesicles and the small vesicles. Also here you can see that the tau can be packed inside these vesicles, and that the vesicles that are isolated from HSV1 infected cells contain higher level of tau 205 and 181, in particular in the large vesicles. Finally, this is the analysis of the recipient tau knockout neurons. As you can see here, the cells that are exposed to the cells, to the microvesicles, in particular to the microvesicles derived from HSV1 infected and transfected cells, uptake higher level of tau tagged with GFP, and this tau is phosphorylated higher level at tau 205. And we confirmed this also by confocal microscopy. As you can see here, these are the puncta that show us the uptake of the exosomes and the vesicles that we put on the recipient neurons. So finally, we wanted to isolate the vesicles from our in vivo model that I showed you before. So we took the brain of the mice that are MOC and HSV1 infected. Here we did this by ultracentrifugation, but then we added a sacrum cushion to separate the vesicles. We obtained 3 fractions. The first of all was the first fraction on top of the layer was the purest one from the characterization both of the marker of the intracellular, the absence of the intracellular markers. And then here you can see the analysis by electron microscopy that confirm that are the vesicles that we are searching for. And here you can see the content of tau that again, it was at higher level. The content of tau was higher in the vesicles that were isolated from HSV1 infected mice. And this tau, you can see that here is at higher level phosphorylated in 205 and 181. So in this study that recently published, we show that HSV1 infection in neurons is able to induce the release of extracellular vesicles that are packed with neurotoxic peptides such as phosphorylated tau, and this extracellular vesicles could be uptaken by recipient neurons, and this shows us that HSV1 infection can indeed promote tau propagation among neurons of the extracellular vesicles, thus supporting the hypothesis that repeated these cycles and the repeated activation of HSV1 may concur also by this pathway to an increase in neurodegeneration. So I want to thank you all the groups and the researchers that take part on these studies, and you for the attention. Thank you.
Question?
Hi, great talk. It's really interesting to see the propagation of tau in your EVs. I was wondering, what do you think the interaction is between the HIV and the phosphorylated tau? How do you...
The HSV and the phosphorylated tau?
Yes.
Yes, I show you is that phosphorylated tau is a really difficult protein. There are a lot of post-translational modification. We focus on these 3 sites that are really related to Alzheimer's diseases, and we think that they are modulated differently. And of course, as I showed you before, the 217 was really higher levels, so tau is really high phosphorylated. We don't know which kinases, we are studying about this a little bit, which kinases could phosphorylate it so much that sites, but there, if you can see, the 205 was less inside neurons. So maybe we think, and we hypothesized, we don't have really proof about this, but we think that different sites, phosphorylated sites from HSV1 infected cells, are modulated differently. So maybe 205 was directed outside of the cells, while 217 differently. And also it was really interesting to see that some of the sites are migrating toward the nucleus, and this could be another neurodegenerative related properties and pathway.
Yeah, it's interesting. There's a localization guidance mechanism.
Yeah.
Thank you.
Thank you.
Thank you for your presentation.
Thank you.
I was thinking in maybe this mechanism of propagation could be a mechanism of clearance. So have you measured the levels of EVs with phospho-tau or phospho-tau in this model of the animal in plasma or cerebrospinal fluid.
Okay, so you think about the clearance of the tau outside of the protein. Yeah, didn't wonder. We want to move through the plasma. We know that in Alzheimer's, some of these sites, all three of these sites are used as biomarkers, but we didn't check yet. We have some pilots, experiment about this, but we haven't the data yet. But it is, of course, possible. I don't know if it is a method of clearance about this, but for sure, HSV1 and the virus infection have a really difficult environment to study because there are really a battle between the virus and the cell, and there are a lot of hypotheses. For example, the increased uptake of the exosomes could be due to an increased ligand that are HSV1 related. So this could be one of the hypotheses about the higher uptake from the recipient cells. But I don't know about the outside and why this is. There is another hypothesis from another paper, and that was it's really interesting about a beta and also maybe p-tau, that these peptides could have antimicrobial effects. So in this by this point of view, the cells could maybe send signal to other cells to activate some antiviral states. We know this about other STING and inflammatory related protein, and they are trying to correlate this. We know about this beta, but not with phospho-tau yet.
Thank you.
Thank you very much.
Thank you.
So, the next speaker is Dr. Colaci, and the title of the presentation is Use of Atomic Force Microscopy Nanoannotation for Extracellular Vesicles Detection and Biogenesis Analysis, a novel MATLAB-based approach.
Okay, good evening everybody. My name is Federica, and in my today's talk I'm going to show you a novel MATLAB approach for the study of extracellular vesicles by atomic force microscopy. I am a research fellow at the Institute for Structure of Matter at the CNR, and this work is within the scope of the PRIN paradigm. So let's start with the topics of this talk. We'll start by talking about the extracellular vesicles and their biogenesis and methods of analysis. Then I'll briefly introduce the atomic force microscopy. Then I'll show you our results and future perspectives. So, as few speakers already said, extracellular vesicles are lipid-bound vesicles secreted by cells into the extracellular space. There are mainly three kinds of extracellular vesicles, but we focus on the microvesicles. Microvesicles range in size between 100 up to 1,000 nanometers, and they are released by pinching or budding of the plasma membrane of the cell. Both microvesicles and exosomes could be very useful for cancer therapy, so a better understanding of their release and their biogenesis could enhance the research in this field. So, here's the typical two kind of analysis that we can make, and we'll focus on the physical properties of the extracellular vesicles. In particular, we can have NTA analysis, DLS, but also the microscopy. And in the microscopy, we can find electron microscopy and atomic force microscopy. Both these techniques could reach high-resolution images, but with the atomic force microscopy, the sample preparation is very easy. So the atomic force microscopy can work both in a liquid environment and in air, so the sample can also be alive. atomic force microscopy has the typical setup shown in this figure right here, and it consists of a tip that is placed at the end of a cantilever. On the back of the cantilever, a laser beam is focused, then it is reflected onto a photodetector. The photodetector is connected with a controller that, by a piezoelectric, is able to move the sample plate. By atomic force microscopy, we can work both for an imaging mode or for an indentation mode. In an imaging mode, we can have high-resolution morphology images, but with the indentation mode, we can also have some information about the mechanical properties of the sample. So, let's focus on the indentation mode, and for each point of the sample, we can have the curve right here. And in particular, in the first step, we can say, we have the tip and the sample that are not in contact with each other. Then the tip is pushed against the sample, it indents the sample itself, and then it is retracted from the sample. We can have an approach curve, that is the one right here, and that retraction curve. From the approach curve, we are able to estimate some particular mechanical properties, such as the Young's modulus, that is an estimation of the rigidity of the cell, or in this case, of the extracellular vesicle, and by estimating the slope of this part of the curve, while working on the retraction curve, we are able to estimate some particular properties, such as the adhesion force, the energy dissipation, and also the viscoelasticity of the sample. So this one right here is a typical curve for a biological sample, but for extracellular vesicles, we have a particular trend that is the one represented here. You can see a peak, we can say, that is quite particular. So in this case, it is reported in the literature that this trend is a typical trend for the AFM indentation and extracellular vesicles, and we can see three dots, 3 points of interest. The first one is the contact point between the tip and the sample, in this case the extracellular vesicles. By this point, the slope of the curve increases. Then we have the second part of the curve in which the reaction force of the sample of the vesicle is softer. Then we have another increase until the last dot. This last point is the most important one because two different events may happen. In fact, the first one could be the rupture of the membrane of the extracellular vesicle, while another kind of event could be the touch between the two lipid layers of the extracellular vesicle. Which one of these events is the right one, we actually don't know, but we focused only on this trend, so for our work, it doesn't matter. We wrote a software in MATLAB that is able to identify only the curves that have this particular trend. So in this case, we are able to estimate the presence, the concentration, and also the mechanical properties of the sample. Our software consists of few steps. The first one is the selection of the starting point. This point is chosen right here. And by this point, the software calculates the number of flexes of the curve. And in particular, the software selects only the curves that have at least 2 flexes at this point of the curve. In this way, we are able to acquire all the curves, but we are able to identify only the curve with this particular trend that are the ones of the extracellular vesicles. Until nowadays, non-indentation with AFM has been used only for the mechanical properties, but with our software, we are also able to estimate the presence and the concentration of extracellular vesicles. So here's some examples of the application of our software. These two images right here are the typical images from the AFM analysis. The first one is the elasticity map of a specific region of the sample, while the second one is the topography map. For the elasticity map, each pixel represents the Young's modulus value of that point, while for the topography, each pixel is the estimation of the contact point from the tip and the sample. So for this reason, we can have an high resolution image in this case. But the most important information in this slide is that in some cases, we could have two different kinds of images and information for the extracellular vesicles. In fact, on the left, you could see that two vesicles could be present. But in this image right here, one vesicle seems to be disappeared. So which one is the right image, and how many vesicles are there in this kind of region? From this image right here, we can say anything, but with the application of our software, by matching each curve with a white pixel, we are able to say that in this region, only one vesicle is present. And in this case, the topography 1, we can say. But here's another example in which the topography is not clear at all. So only seeing the elasticity map, we can be confident to say that there are two vesicles, but with the application of our software, we can recognize two different vesicles in this region. Here's another example in which the elasticity map is not clear, and also in this case, the topography 1, because the black and white image matches perfectly with the topography of the region. At this point, we decided to apply this software also on cells. And in particular, we studied the MCF7 cell, that is a cell line derived from breast cancer. And in this case, we can see that the curves are the pink dot right here. The curves are present mainly in the peripheral part of the cells. In this case, we have two different cells near to each other here. And here's another example in which we have only one cell, and also in this case, we have the curves placed in the peripheral part of the cell. So, remembering that the biogenesis of microvesicles could be by blebbing or pinching of the plasma membrane, and knowing by literature that this kind of blebbing is present mainly in the peripheral part of a cell, we can easily conclude that this kind of curves detected on cell could be the blebbing of the plasma membrane. So, our software could be useful both for the study of extracellular vesicles and for the study of cell. And in particular, for the extracellular vesicles, we are able to estimate the presence, the concentration, and to have clear results about the mechanical properties of the extracellular vesicles. While, on the other hand, studying the indentation on cell, we are able to follow the dynamic changes of the membrane for the budding and the release of microvesicles. To conclude, I'll show you briefly our future perspectives. As you already know, there is the artificial intelligence, and our future perspective is the application of artificial intelligence at AFM. And in particular, we are going to use the neural networks. Neural networks are computational models composed by a few layers of neurons that take in input and information such as an image, or in our case, a curve, and give us as output and information of the classification or a prediction. So in our case, I trained a network that is able to acquire the curves from the AFM non-indentation and give us as output a classification of a curve. And in particular, we have two classes right here that are non-vesicle-like curve and vesicle-like curve. This is just an example of the confusion matrix of our training pathways, but in general, we reach an accuracy of 94%. So it's quite good, but we'd like to make it better. And in particular, we can focus only some few features, such as the mechanical properties, as I already said, so by the estimation of the Young's modulus of the curves, by the number of flexes, or some other information, such as the, I don't know, the mean of the data. So this is a project that is still ongoing. To conclude, I'd like to thank Dr. Simone Dinarelli, Dr. Marco Girasole, and Dr. Giovanni Longo from the ISM of the CNR. I'd like to thank Professor Bottini and Dr. Geta Corti from University of Rome Tor Vergata. Last but not least, I'd like to thank Professor Lanuti, Professor Simeone, and Dr. Francesca Dascanio from the University of Chieti and Pescara. And then I'd like to thank you for the... You're welcome.
Yes.
for the attention. Thank you.
Thank you very much. Any questions? I have a question. What is the resolution of this technique? Do you have a limit for the size?
Yeah, but we have always some limitation with size with the AFM, so we can have a little image with a high resolution also by the curves, because we have more pixels in a specific area. In fact, with the cell, you can see only one dot. That is the resolution for that point, because we have nanometric scale, but only one pixel for size. So it could be better to have high concentration for the vesicles.
Okay.
But do you have compare with the other techniques?
We are comparing it right now, so it's a project still ongoing.
Ah, okay.
No question? Thank you very much.
Thank you.
Thank you.
So now, Dr. Zanotti, from University of Tor Vergata, that present a work titled Cabbage-derived exosome, an innovative purification method and potential application in pharmaceutical and cosmeceutical fields. New aspect of the of the macrovesicle, please.
Sì. Okay, thank you. So, good afternoon to everybody. I'm Clarissa Zanotti, a PhD student in the doctoral course of cellular and molecular biology here in Tor Vergata, and I work in the laboratory of plant physiology of Professor Mauro Marra. And I would like to thank the organizer to give me this opportunity to talk about plant-derived exosomes today, and in particular of cabbage-derived exosomes. So a little introduction about plant-derived exosomes. And these type of exosomes are similar to mammalian exosomes for their size, dimension, content, and maybe properties. They present diameters with values between 20 and 200 nanometers, and they are composed by phospholipid bilayers and are made up of proteins, lipids, nucleic acids, and secondary metabolized. And thanks to these natural compounds, they can exert natural anti-cancer, anti-tumor, and antioxidant, anti-inflammatory, and wound healing effect, because they can be isolated by streamless varieties of fruits and vegetables, and they can maintain this unique and powerful therapeutic effect. And in plants, exosomes participate in many physiological processes. For instance, in developmental processes such as cellular proliferation and differentiation, and moreover in the plant immune response, so against plant pathogen infections. And in particular, plant exosomes, thanks to their component and thanks to their bioactive compounds, they can participate in the intra or intercellular communication, so deliver these bioactive compounds, imparting new properties or function to receiving cells. And now is well reported in literature how plant exosomes can interact with mammalian cells, and so suggesting a putative cross-kingdom communication. And for this reason, plant-derived exosome could exploit a novel technological platform for loading and delivery bioactive compounds, but moreover exogenous nutraceuticals and drugs. First of all, for their biocompatibility, given by a green and biological production that give also higher bioavailability. And moreover, for the abundance of plant resources, we can have a large scale of production with cost efficiency. And moreover, they present promising biosafety and therapeutic application for the treatment of various diseases. And indeed, in the last 10 years, increased the interest for plant exosomes, and despite many types of isolation methods are currently available, all of them are unsatisfactory in terms of yield and purity for plant-derived exosomes. So this research project, that is a PON research project, have an industrial application. So the aim is to isolate plant-derived exosome from natural source for an industrial application, in particular for cosmeceutical and nutraceutical fields. So we start to the selection of natural source, and we decide to use Brassica oleracea spruce. And then we test at the beginning different type of isolation methods, such as the ultracentrifugation one, but moreover the polyethylene glycol-based approach. And from this type of isolation protocol, we obtained the first exosome solution that were analyzed with dynamic less scattering analysis that give us information about hydrodynamic diameters with values around 100, 200 nanometers, moreover about the polydispersity index, and from this type of analysis we assess the better storage condition for our samples to still maintain their biological activities. And moreover, we obtain negative zeta potentia values, suggesting that vesicles in solution have a net surface charge properties, and so we wondering if exosomes can be isolated with anion exchange chromatography. So after a lot of studies and try, we developed this protocol in our laboratory, starting from the homogenization of the plant tissue, performing differential centrifugation, and then filtration and concentration step. And these pre-treated samples were then purified with the anion exchange chromatography in an FPLC system, so in fast pressure liquid chromatography. And so at this point we have the highly purified cabbage exosomes that were then characterized. So for the characterization of our plant exosomes, we start with a qualitative preliminary quality analysis with fluorescence microscopy to assess if our exosome solution are purified with this process, and we assess a reduction of contaminants that generally occurs in the plant extract, and then we assess the use of TET8 protein as exosomal protein marker for plant-derived exosomes, and TET8 protein is the analog of CD63 as mammalian exosomal marker, and we assess the presence of exosomes in exosome-enriched fraction of FPLC, and then we perform transmission electron microscopy, which we assess their rounded shape and confirm their dimension. But in particular, we perform nanoparticle tracking analysis in which we compared exosome solution purified with FPLC and not purified with our protocol. And what we can observe is significant increase in the purification given by a reduction in the peak area of the histograms, and moreover a reduction in the intensity in the density plot on the right side. Moreover, we can observe how the mean dimension of vesicles analyzed before the purification process have values around 200 nanometers, and then after the purification process in FPLC, the values are around 113 nanometers. And here I have the videos of this analysis, but no. It didn't work, it's not matter. And so we have the videos of the samples that we analyzed with the relative concentration, and we have at the end of this purification process cabbage-derived exosomes with concentration in the order of 10 at 10 particles per milliliters. And at this point, with these highly purified cabbage exosomes, we were able to perform proteomic analysis to better characterize their content. We identified 314 proteins that were firstly analyzed with string analysis to perform a protein-protein interaction. And then we assess a functional enrichment for this protein, and they were clusterized in different clusters. For instance, we identify protein associated to the proteasome complex, but moreover to the extracellular vesicle biogenesis, such as annexin, clathrin heavy chain, but moreover heat shock protein that were more abundant in plant exosomes and plant extracellular vesicles. We identify moreover protein associated to metabolic process, to apoplast function, such as yakalin protein, but moreover protein associated to intercellular communication, such as the presence of translin family protein or DNA binding protein that suggests a putative role of these exosomes in the intercellular communication. And with highly purified and better characterized exosome, we were now able to perform biological assay to assess if exosome could have an effect for biological activities. In particular, for the future cosmeceutical application, we evaluate the effect on skin regeneration. So we first of all set up the condition and we analyzed if exosomes have any cytotoxic effect, and we test different concentration of cabbage exosome, purified and non-purified in FPLC, and for all the concentration and the sample and the exosome type that we used, we didn't observe any cytotoxic effect. And so at this point, we perform a wound healing assay to assess if exosome could have a role in skin regeneration. And we perform a scratch wound on ACAT cell monolayer, and then we treated cells with different concentration of exosomes. And for all the concentration that we test, we observe a strong wound closure after the treatment of exosomes, compared to the control samples, but also to the positive control treated with TGF-beta. And so from these results, we suppose a strong regenerative effect upon the treatment of cabbage exosomes. So to better investigate in which way cabbage exosomes are able to induce this wound healing effect, we evaluate the expression of genes associated to wound healing. For instance, genes associated to proliferation, such as AGFR or Ki67 as marker of proliferation, and we observe a significant increase in the expression of this proliferative gene, similar to the positive control and more than the control one. And then we try to investigate also in the expression of genes associated to extracellular matrix remodeling, and we obtain significant results for collagen type 4 gene. And we know that wound healing is a sophisticated process in which occur many physiological processes, such as also the immune response. So we're wondering if exosome could have any anti-inflammatory activities. And so we firstly perform an in vitro assay using raw cell line. We stimulated the raw cells to induce the immune response with LPS. And these cells were pre-treated with cabbage exosomes. And then what we observe is the significant reduction in the nitric oxide content for specific concentration of highly purified cabbage exosomes. And this reduction is around 60%. So then we treated ACAT cell line to recapitulate this inflammatory response under LPS stimulation. And we analyzed the expression of pro-inflammatory genes such as toll-like receptor 2 and interleukin-1 beta. And we obtained significant reduction in the expression of this pro-inflammatory gene, suggesting that cabbage exosome could have an anti-inflammatory effect in this in vitro test. And at the end, we started to validate also if exosomes, cabbage exosome, can have a role also in basal condition, also for the same aim to have a cosmeceutical application. We use human dermal fibroblasts, and we treated with these cells in basal condition with different concentration of cabbage exosomes, and we analyzed the expression of proliferated gene, and also in this case we obtain increase in the expression of Ki67, GDF11 and EGFR, suggesting that also in basal condition cabbage exosomes maybe have a role in the modulation of basal proliferation. And now the main question is which is the players in this biological processes, and we are now starting to perform microRNA sequencing from cabbage-derived exosome to better understand if specific microRNA are correlated to these biological activities. So in conclusion, we can say that we were able to isolate cabbage exosome from brassica oleracea using this innovative purification protocol based on the anion exchange chromatography in FPLC system. We characterize the vesicles in terms of dimension, sulfur charge, sample concentration, their shape, and we perform also the proteomic content analysis. And we assess some biological activities, in particular a strong wound healing effect and a putative role in inflammatory processes, in particular as an anti-inflammatory effect. And at the end, I would like to thank my colleagues here in Tor Vergata and Artera Bioscience to give me the possibility to perform the biological assay, and the Andrea Scaloni group for the support for the proteomic analysis, and all of you for your kind attention.
Thank you. Suggestion, question?
Yes.
Okay, please.
Thank you. That was very interesting study. I never thought about plants making exosomes. Do you have an idea how efficient is plant exosomes uptake by mammalian cells?
So we didn't perform this type of analysis, but from data reported in literature is well known how mammalian cells, human cells are able to uptake plant exosomes. And maybe through this process, plant exosomes deliver their bioactive compounds and exert these biological activities.
Can I have another one? I have two more, but I will have another. So you think that microRNAs delivered by plant exosomes, they exert the biological activity. But I would rather think also about plant-derived metabolites, which are unique for plants. Do you have any metabolomic data, or maybe something from the literature, that what are the plant-specific metabolites that affect metabolism of human cells, specifically from exosomes?
So we perform metabolomic analysis from cabbage exosomes, and we are now validating the results. But we know that sulforaphane from broccoli is a mediator of some antioxidant effect in this biological activity. So maybe also metabolites have a role in this way. But for us, the major players are the microRNA, because in literature are well reported that microRNA from plants and from extracellular vesicles of plants can regulate some target human genes.
Thank you.
Thank you very much.
Thank you for this very interesting talk. I was just curious why you chose to work with cabbage and not tomatoes or lettuce or cucumber, for example?
So the choice first for the quantities. We choose to use sprouts of brassicoleraceae, so broccoli, because we can obtain a large amount in few days, and maybe also from tomato is possible to isolate plant-derived exosomes, and also because cabbage are well reported in literature have an anti-inflammatory effect in some inflammatory disease, and some clinical trials are now testing the effect of broccoli exosomes. So for this reason.
Thank you.
Thank you too. Thank you.
So the last speaker is Dr. Barbieri from Italy, and the title of the presentation is Small RNA Carried by Outer Membrane Vesicles, a novel complex mechanism to regulate the infection process of HLYF, positive Zika toxin producing Escherichia coli strains.
Thank you for the presentation. I know that you are all tired, me too, but I'll ask you for a little more patience. So now I will show you the preliminary results of my PhD project that regards the potential involvement of small RNA within outer membrane vesicles produced by Zika toxin producing E. coli. And but first, I would like you to present this guy. So, who is Zika toxin producing E. coli? Probably you've heard about stec strains as foodborne pathogens frequently associated with foodborne outbreaks, like the recent outbreak in America of October 2024 due to the consumption of cheeseburgers from the famous fast food, and due to the consumption of contaminated onions in these cheeseburgers. But also another example is the contamination of organic carrots that caused another outbreaks in America. These strains, Zika toxin producing E. coli, are in fact zoonotic pathogens that usually cause gastrointestinal infections in humans, also with severe complications, whose main natural reservoir is represented by the gastrointestinal tract of ruminants. In fact, once ingested by these animals that carry stec strains asymptomatically, these bacteria are released in the environment via feces, and these events correspond to the consequent contamination of water, food, so of the environment devoted to food production. So the infections in humans occur through the ingestion of this water and food contaminated. So the infections due to Zika toxin producing E. coli represent a public health problems, as they can lead to serious sporadic cases, but also to outbreaks of large dimensions. In the pathogenesis of stec infections, two events are fundamental. The first is the gut colonization, through which stec strains can win the competition with the resident intestinal microbiota and, above all, adhere to the intestinal mucosa. And the second is the elaboration of Zika toxins. Zika toxin is a potent cytotoxin that is encoded by the STX1 and STX2 genes carried by lysogenic lambdoid bacteriophages that are integrated in the genome of stec strains. And this toxin has a tropism for the endothelial epithelium. So once arrived at the level of the enterocyte, there is the internalization and the translocation of Zika toxin through blood circulation, and then the binding to target cells, where it induces cell damage, above all on the microvascular endothelial of renal cells. And this cell damage could correspond to different symptoms, ranging from mild diarrhea to the most serious hemolytic uremic syndrome, that is the main cause of pediatric renal impairment that can also lead to neurological complications and, unfortunately, sometimes also to death. So, among the different factors, both of the host and the pathogens that are involved in the infections due to stack strain, there is also the possibility, and in fact it has shown that also outer membrane vesicles could contribute to this, to the progression of the infections in humans. In fact, it has been shown that in the human host, these vesicles are absorbed by both intestinal cells and microvascular endothelial cells, and also that these vesicles possess proteins and different virulence factors, including the Shiga toxin, that can be released into host cells. But the knowledge about their acid nucleic and RNA content, above all small RNA content, is still scanty. And another thing, another aspect that I would like to know is that in last decades, stack strains harboring virulence determinants typical of extraintestinal pathogenic E. coli strain are emerging in Europe as a cause of severe disease in humans. And this characteristic is principally due to the presence in the genome of these hybrid strains of a plasmid that is called PR44A plasmid that carries the HLYF gene that is associated with an augmented production of outer membrane vesicles. So, considering all these aspects and considering that these strains are emerging as highly virulent strains, we selected two of these strains, that is an O80 serotype and O26 serotypes of stack hybrid strains, both producing Shiga toxin subtype 2 and possessing the HLYF gene, to investigate about the production of these vesicles and above all to focus on the small RNA content and their role in the infection. So the strains were grown in Luria-Bertani broth and then were subjected to two differential steps of ultracentrifugation to collect two pellets, so two fractions of outer membrane vesicles. The vesicles were then studied analyzed with electron microscopy, both scansion and transmission, to confirm the presence of pure vesicles, and then analyzed through nanoparticle tracking to study their size and concentration. At the same time, we performed the whole genome sequencing analysis of these strains to identify in the genome of the strains under study sequences or regions of the genome encoding to some small RNA, so to identify putative region encoding small RNA. And starting from this information, we design specific real-time PCR parameter pairs. Then, after the confirmation of the presence of this small RNA in the vesicles produced by these strains, we also quantify the presence in the both fractions obtained, and then finally we also study the function of the identified small RNA. And these are the results of our microscopic analysis of our preparations of outer membrane vesicles. So in general, we can say that using these two differential consequential steps of ultracentrifugation at two differential speeds, the first obtained at 100,000 G and the second at 200,000 G, allows us to obtain different size distribution of particles. We can identify a main population for both strains of about 20 to 100 nanometers, but we can also say that in the O26 strain we observed a wider population of larger dimension. In general, we observed in all fractions, so for both strains, a higher number of vesicles of 40 nanometers, so very little. But we also observed differences between the fractions. For example, in fraction 1, the population of 100 to 180 nanometers was more abundant. But in general, we observed dimensions that are very similar to those already observed in literature. Then we proceed for the with the nanoparticle tracking analysis in both strains, in both fractions. In general, we observed these uniform peaks of size distribution of particles with a modal value of 70 to 120 nanometers. This result could also could be slight difference from the measures previously observed from microscopic analysis, but it could be due also to the different handling of the sample and the different technique, of course. But in general, we obtained high concentration of vesicles for both strains, in particular in the order of 10 to 7 particles per ml for O26 serotype and 10 to 9 particles per ml for O80 serotype. But then we proceed when we focus our attention on the small RNA. So we perform this whole genome sequencing analysis, not only on the 2 strains under studies, but also on 3 additional HLYF stack strains, so these hybrid strains, in comparison with non-pathogenic E. coli strains, so not possessing these genes, these characteristics. And selecting the sequences encoding small RNA of dimension of lower than 20 nucleotides. So first we identify 27 putative small RNA produced in the genome of these strains. 10 of these were also identified in the sequence of the plasmid that I said before. So presumably they are carried by this plasmid. And starting from this, we design these real-time PCR primer pairs. So then we move to the small RNA within the outer membrane vesicle. So after the RNA extraction, DNA treatment, and retrotranscription, with this, the above mentioned primer pairs, we identify within the vesicles 10 small RNA produced at least by one of the 2 strains under study. 4 of these also identified in the sequence of plasmid. And an interesting thing is that 1 small RNA was present in both strains, that is SNC100 target. Then we proceed with the quantification of this RNA to see if there were differences in concentration between the fractions. In fact, for O26 serotype, we identify a high abundance of allele Ifasi and Staxi small RNA, and with respect to the other small RNA, but in this small RNA we also observe differences in fractions. In fact, while for allele and Ifasi small RNA we observed a higher abundance in fraction 1, for Staxi small RNA we found a more abundant concentration in fraction 2. For O80, we identified the presence of only 4 small RNA. Group 2, D1, D47 was the one with the lowest abundance, but in general the other targets had higher concentration, and again with different concentration between the fractions. And another thing that I want to highlight is that STNC100, that is present in both strains, was higher, whose concentration was higher within a O80 serotype with respect to O26 serotype. Then we proceed our investigation studying the function of the small RNA identified with this OMV. For some of these, we identify regulatory functions, in particular in bacterial DNA replication, transposition, integration, and also involved in the management of stress response. We also found that some of these encompass all the domains of life. Then we search for homologous regions between the sequences encoding for our small RNA and the genome of other genera. In fact, we found a similarity between the sequences with respect to other genera that are Shigella, Salmonella, Klebsiella, and we can say that are all enterobacteria, so probably they share the same niche, so this could be interesting. And we found homology also against bacteria belonging to genera that had plant host as example, so non-human host, Agrobacterium, Xanthomonas, and so on. And finally, the last thing is that for the target STNC100 that was present in both strains, we found that it was encoded on the Shiga toxin encoding phage, so in the genome of the strain, downstream the STIC2 operon in both strains. And this suggests a regulatory role in Shiga toxin production, and this is the first time that such a role was identified for this target. So, with this study, these preliminary results, we characterized different size populations of outer membrane vesicles produced by these highly virulent STEC-XPEC strains. Since it was the first time we were approaching to these techniques and in general to outer membrane vesicle study, I think that we obtained a good quality and yield using these two differential steps of ultracentrifugation. But then we focus our analysis on the study of the small RNA. In fact, we were able to identify 10 small RNA produced by at least one of the two STEC strains under study, and we can affirm that these small RNA are released through these outer membrane vesicles, and that they have putative role in the regulation of bacterial DNA replication, integration, and so on. And we also saw that this encompasses all the domains of life. So, considering that outer membrane vesicles are known to be a kind of bridge of communication for interspecies, intraspecies, interkingdom in general communication, and if we focus on the physiology and mechanism of pathogenesis of STEC, the production of these vesicles probably could be involved in the crosstalk with the host, but also with the other members of the intestinal microbiota. And finally, we found that the STNC100 small RNA target had a complementarity region with the operon of the STIC2 Shiga toxin in the sequencing of the genome of the phage that is integrated in the genome of the STEC. And thus, this means that this target, small RNA, may be involved in the regulation of Shiga toxin production that is may be mediated by outer membrane vesicles delivery. And I want to thank you all my lab colleagues, in particular the European and National Reference Laboratory for E. coli, in particular Shiga toxin producing E. coli, and also my colleague Serena of Sapienza, and all of you for your attention and patience.
Thank you very much. Question? I have one question. Depending to the strains, you have different type of vesicles. Do you know why?
With different types of vesicles.
Yeah.
I think that O8 serotype produced more vesicles. So, in fact, we saw the results from the NTA analysis.
Yeah.
And I cannot say that the vesicles are so different. We have a different production.
Yes.
But we need to deepen the investigation to say that the vesicles are diverse. We have a different concentration of sites distribution.
And
probably
the
you
content
say
is different because we have seen that for some vesicles you have small RNA.
Okay.
Different.
In this case,
yes, I have seen in your presentation.
You are referring to the small RNA.
Yeah.
Okay.
It's
okay.
different.
Yes, I understand the question. I think that we have to investigate more to say why there are these differences. Because in general, if this RNA as allele and IFASI are found also in other genera or in other kingdom of life, probably they are useful for the fitness of the bacteria in this case. And in other case, we have we may have a selective production of vesicles and of RNA in those vesicles, probably that could help the fitness of the bacteria, both with the interaction with the host and with the interaction of the other members of the microbiota.
Yeah, thank you.
So we have finished.
Yes.
Yes. in time. I would thank the young researchers and please, Claudia, Massimo, if you want to wait a moment. Yes. 10? Ah, okay.
We need just 10 minutes to, gather together the permission for so we ask you, just to wait for us.
So just to make the final selection with the committee to give the awards, and then to give the awards
and the conclusions, and then, you're free to go. But it's yawning.
Okay.
So you stay inside and we go outside. It's the opposite. You stay inside. Andiamo fuori.
Andiamo dall'altra parte?
No, perché loro stanno qui e passano e gli stringiamo la mano. Chi è che si è portata mezza casa tanto?
Mi dispiace perché non li vedo.
Ma niente,
Li ho visti qua in giro. Erano tutti PDF. Tu in mezzo alla tua roba sei sicuro di no, eh? Io ti avevo guardato le robe nella tua cartelletta. Io avevo questa busta qua di quelle di Evita però solo, quindi vabbè, mi spiace, ma va bene.
Allora, quella è Protto che domani viene alla scuola, no?
Sì, domani gliela cambiamo. Ok. Andiamo, dai. Siamo tutti qua, scusate, cosa facciamo? Stiamo tutti qua?
Sì.
Ok, aspetta, fammi vedere, no, volevo vedere una cosa. Okay, so now it's time to give the awards and also close these very nice days that we enjoy together. I'm sure that was interesting because we could see from your interaction and your interest that this was really an effective day together. So first of all, we want to give an award to be part as a keynote speakers of our workshop. To one for Professoressa Ratigliere. This is from Tor Vergata and from the rector and I mean this just a recognition by our university.
And another one to Professor Milan. That hopefully you like it and you put in your office. Congratulations.
Okay, so now it's time to give the award for the oral presentation. We will have two from Evita and one from Zanichelli for the oral presentation, the same for the poster.
Yeah, it was very tough because you guys are amazing. So every presentation was really very, very, very good.
Yes, it was difficult to decide. It's all very, very interesting.
Yeah, well, we ended up with this. So we are very proud to have this, to give this award. And Evita actually is very proud to support young students for this presentation. The first one is to Mona Belaid. Congratulations. And the second one, congratulations again. The second one to an Italian actually, to Virginia Protto. Maybe we then take a picture afterwards. Congratulations, bravissima.
Okay, should we?
Sorry, some suspense. So the winner is Dr. Fanelli, and you received the Nick Kelly Awards.
But we will print you out because we will send you the PDF. So the Gianni Kelly Award is really. Actually, yesterday was in your lab. Oh, here, we found it. But we have to cut in half.
Tomorrow. We
Tomorrow.
forgot to say
You have-
that
yeah.
the awards are for
We have
Etica
it.
awards are.
You can trust us. We have it.
No, I just forgot to say that Evita awards are 250 euros for the oral presentation each. Okay, so you get some money, and the Gianni Kelly one. is,
150 euros.
Okay.
You can use to purchase books and things, in on the website.
And so please afterwards come to me and just give me your. Okay.
Posso now for the poster.
Yeah, for the posters also. So the first winner for the best poster is Francesca Dascagno.
Hola, hola.
Congratulazioni. And the second one is Lina Pineda, who got a very high hit. So thank you for applying the sticks on the. Thank you. And these were awarded with 200 euros each.
We found it.
And the third is Tassoni Selene for the prostate. It's the Gianni Kelly Award.
Thank you.
So,
I would like to thank everybody, and for who is going to attend the school tomorrow morning, 8:30, Finazzi Agro Hall, and Mayra is with the person of interest, basically for tomorrow morning pickup in front of the hotel, who's going to be in the hotel. So for Maria, Mona, so there will be a taxi, and Mayra is the person of interest, your reference person, the lady from ISON, okay? But for the other person like that will just directly come to the university, will be 8:30 in front of the Finazzi Agro Hall. And again, I would like to thank everybody. I think that we have made, a good job today, and thank you very much, for attending the conference.
Bravissimi. Facciamoci una foto, dai. Questo è il premio. Hai preso 1000.